Okay, it's 9 o'clock in the morning. Good morning, everyone, and welcome to the NIH workshop. My name is Oleg Miroshnichenko. I'm program official from Office of Research Infrastructure Program, a division of Program Coordination and Strategic Initiatives at NIH. This meeting comes at an important moment as the biomedical research community continues to adapt to new scientific opportunities, rapidly evolving technologies, and growing expectations for collaboration, rigor, and translational impact. The conversation we'll have today are about more than individual programs or resources. They're about the research infrastructure needed to support the next generation of discovery, including the thoughtful integration of animal models and new approach methodologies to improve the predictability, human relevance, and translational value of biomedical research. We are fortunate to begin with welcoming remarks from Nicole Klein-Stroyer, NIH Deputy Director for Program Coordination, Planning and Strategic Initiatives, and Director of Dipakipsi at NIH. Nicole is internationally recognized for her leadership in developing innovative, human-relevant research strategies that advance public health protection. Prior to her current position, she serves as Director of National Toxicology Program, Interagency Center for the Evaluation of Alternative Toxicological Methods within NIHS. Nicole, please give your introductory remarks.
Thank you so much, Oleg, and thank you to the whole ORIP team and the organizing committee for pulling together this incredible workshop. We have such an amazing agenda planned for today and for the 12th. So I'm really, really excited to see what comes out of this. And thanks to everyone who is dialing in and listening and taking time out of your busy schedules to join us online today. So we're really excited to bring together such a diverse group of experts driving innovation and collaboration in this space. I think, as Oleg mentioned, this workshop comes at a really important moment for biomedical research. NIH has long supported a very broad ecosystem of model systems that are designed to help understand human biology and disease mechanisms and therapeutic response. And this workshop is comes at a watershed moment. So reflecting NIH's growing commitment to accelerating adoption of NAMs, or human-based science, and innovative technologies that are human relevant, predictive, and efficient research tools. So we all know that NAMs are advancing very rapidly, and to ensure their broader uptake and use and translational application, we need to focus on validation, shared standards, infrastructure, training, building confidence in their use throughout the scientific and regulatory community. So the overall goal of this workshop is to support practical discussion towards those aims. So how can new approach methods and human-based science and innovative technologies be incorporated into biomedical research in a rigorous and fit-for-purpose way that advances the NIH mission? So I just have a couple of brief remarks before we jump into the workshop. So first, the workshop was organized by the Office of Research Infrastructure Programs, or ORIP, with representatives from many different NIH institutes and centers and offices, and it's directed broadly to the entire biomedical research community. So NIH-supported research model systems and infrastructure have made major contributions to understanding human disease and advancing therapies. And I think we are all witnessing this very rapid progress in new approach methodologies, expanding the range of tools that are available to study human biology and disease mechanisms. As many of you are aware, NIH recently announced the intent to establish the Office of Research Innovation Validation and Application, or ORIVA, within the Office of the NIH Director and within Dipakipsi. We've also recently made major investments through programs like the Common Fund complementary program to develop and scale research methods that better simulate human biology and produce more predictive models of human disease. And the goal is to really optimize the models that we are using and ensure that they have the highest relevance to human biology for each scientific question, and that the models that researchers choose are the best fit for the research question at hand. So that requires clear criteria around model selection, improving validation and shared benchmarks, and strong links between experimental systems and the biology that those systems are querying and human outcomes. So we have a really large opportunity here to improve rigor, predictivity, reproducibility, and translation across NIH-supported research. So this workshop will take place over 2 different days, and it was organized by 2 ORIP divisions that have very related but very distinct missions. And so together, the 2 days, I think, will really take a creative approach to connecting strategic questions about model integration with technology-focused discussions about development and implementation of human-based science and innovative technologies. So one major focus is how NAMs and other human-based models can be used to study complex disease biology and support better preclinical decisions. And then another focus is how emerging technologies like organoids and tissue chips and in chemical systems and computational models and AI and real-world data can really strengthen human relevant research. So across both days, there's going to be common themes around validation and reproducibility and shared data standards and high quality resources and infrastructure and training and sustained NIH-wide coordination, which is really the remit of both DPKIPC and ORIP. So we really hope that this workshop will engage the broader biomedical research community, will help define practical next steps for building a more predictive, integrated, and human-relevant model systems ecosystem. So again, just want to extend my thanks and my gratitude to all the speakers and the organizers and the panelists and the participants for contributing your time and your expertise, and wish everyone a productive and successful workshop.
Thank you very much, Nicole. Please show our next slides. Now I would like briefly to present our organizing committee. As you can see, we have a nice representation of NIH programs from a number of NIH institutes, as well as representatives from biomedical community. Check marks indicate co-chairs of our 4 sessions. I would like also to acknowledge support of our assistant, Susan, and scientific consulting group, SCG team. Next slide, please. And I would briefly mention several housekeeping items. Audio and video has been disabled upon a try for all attendees except speakers and co-chairs. Q&A session will be held only after keynote speaker presentation. Please submit your questions in the chat. For all sessions, there will be no Q&A after each individual presentation. However, you may submit questions in the chat, and they will be addressed during the panel discussions. Dear presenters, please keep to your scheduled time, and I would like to ask panelists as well to take care of it. The workshop is being recorded, but the recording will be only retained temporarily until meeting summary is completed. We still continue considering maybe we will post the recording if we will get all permissions to do that in the future. The written summary will be made publicly available, and for technical assistance, please email Daniel, and there is a link there. Next slide, please. So we are now pleased to begin our keynote presentation, Complementary Roles for Human Organ Chips, Artificial Intelligence, AI, and Animal Models. It is my pleasure to introduce Donald Ingberg, founding director of the Weiss Institute for Biologically Inspired Engineering at Harvard University, and a professor at Harvard Medical School, Boston Children's Hospital, and Harvard John Paulson School of Engineering and Applied Sciences. Dr. Ingberg is a pioneer in the field of biologically inspired engineering, and at the Weiss Institute, he currently leads scientific engineering teams that cross a broad range of disciplines to develop breakthrough technologies to advance healthcare and improve sustainability. His work has led to major advances in mechanobiology, cell-structure, tumor angiogenesis, tissue engineering, systems biology, nanobiotechnology, and translational medicine. Please join me to welcome Dr. Ingberg. Donald, please share your slides.
Thank you. Let me share my slides here. Can you see that now and hear me?
Yes.
Excellent. Great. So thank you so much for the invitation. And as you can see from the title, I was asked to speak about complementary roles for human organ chips, AI, and animal models. And that's what I'd like to do today. So let's get started. So I think you all are well aware about the passage of the FDA Modernization Act in 2022, where it was for the first time stated that new approach methodologies such as organ chips, organoids, AI can potentially be used in lieu of animal data in IND applications reviewed by the FDA. And although that process was supported, it really hadn't been pursued actively. But this past year, things have really revved up with the FDA announcing a plan to phase out animal testing over the next 3 to 5 years, and NIH prioritizing human-based NAMs in grant applications. And so my group has worked for now over 20 years on development, what we call human organs on chips. I really want to emphasize these are not tissue chips. These are organs on chips, because what they are essentially taking techniques from the microchip manufacturing industry, where you have control over features at the nanometer to micrometer scale, where cells and living tissues live at. We're able to create devices that we call microchips that contain living human cells that reconstitute organ level functions, not cell or tissue, but organ level functions. I would say that organ chips are truly organ chips, where organoids are really tissueoids, but that is at least in the original versions of them, they're now both getting more complex. So the idea when we developed this was to accelerate drug development, replace animal testing, and advance personalized medicine. And to just I'm going to give you a broad feel of what we've done and then place it in context of both animal and AI. So our first breakthrough was in 2010 in science. We called it a human breathing lung on a chip. We don't try to build whole organs. Think of these as a living three-dimensional cross-section to a major functional unit of an organ. And we started with the lung alveolus or air sac, and I think you probably are aware, is where gas exchange, anesthetics are delivered, metastases, pneumonias, COVID, et cetera. And if you go to the electron microscopic level, let me see if I can get a pointer here. At the right, you basically have in cross-section air, a single layer of alveolar epithelium, flexible porous basement membrane, and extracellular matrix, and then a capillary cell and then blood. But what it doesn't show you is that this is incredibly mechanically active. Every time we breathe in and out, you basically have stretching of the tissues, and that we know from respiratory physiology has major effects on functionality. So if you're trying to distill down, like what are the minimal design principles of an organ rather than a tissue? An organ are two or more tissues that come together, new functions emerge. Usually one is a vascular tissue to deliver oxygen and nutrients and remove waste products. We also need dynamic flow, obviously, for vascular perfusion, and we felt you need the mechanical environment, breathing motion. So this next animation shows you how it works. It's the size of a computer memory stick, as shown at the top right. It's made of an optically clear silicone rubber that's flexible if you cut it in cross-section. 3 hollow channels, each less than 1 millimeter wide. Middle channel is cut into top and bottom by a porous membrane, which we coat with extracellular matrix. Then we put human lung alveolar cells on the top, human lung capillary on the bottom, and we just recreated the alveolar capillary interface. We then apply cyclic suction or vacuum to the side chambers, and because it's flexible, it stretches and relaxes at the same rate and degree as when we breathe in and out. We then could put air in the top channel to create an air-liquid interface, and we could flow medium with or without immune cells through the bottom. So if this were to work, it should mimic an organ level response. So imagine you have a bacterial infection. There's a tissue signaling response. Cytokines are put out by epithelium, activate endothelium. White blood cells that were just flowing by will now stick and roll because receptors like ICAM are expressed on the surface of the endothelium, and that's homeostasis. You have clearance of bacteria. So now I'm going to show you imaging in the device. The white blood cells are going to be labeled fluorescently. You can't see the endothelium because it's unlabeled, and the epithelium is behind the screen. So to begin with, it's a healthy endothelium. They just flow by. Then we put bacteria on the opposite side. You get this cytokine response. You get ICAM expression, and in a second, you'll see that you could watch in real time these cells being pulled out under flow. And this is important because the initial binding step is shear stress dependent. So you miss that in a static culture when you add immune cells to endothelium. Now you can do high magnification views. Here's one, actually a neutrophil going through two unlabeled endothelial cells. Here it goes through the matrix-filled pentagonal pore, and now comes out on the other side by phase contrast. And now I'm going to show you the white blood cells in red because the bacteria are in green. And you watch them being engulfed. So you just watch the entire human inflammatory response in this little rubber chip. Now, we have developed over 20 different organ chips, healthy and diseased, over the last 10, 15 years. And we've been funded by NIH, FDA, DARPA, BARDA, NASA, set foundations. I can't go through all of this, but I am going to give you examples of the capabilities we've already demonstrated, which is for drug toxicity testing, modeling human acute and chronic diseases, modeling drug efficacy and development of drug resistance, drug discovery and repurposing, vaccine safety and efficacy, modeling host microbiome interactions, assisting in clinical trials design, advancing personalized medicine, and then I'll end with a couple of examples of combining organ chips with AI and animal models. So drug toxicity testing. So we had that lung chip. We talked to pharmaceutical companies and they liked it, but they said, we're more interested in disease models and drug toxicity models. So we hit two birds with one stone by developing a pulmonary edema model, fluid on the lungs. We perfused interleukin-2, an FDA-approved cancer drug that has pulmonary vascular leakage and pulmonary edema as a dose-limiting toxicity. So we perfuse that like in a patient intravenously through the vascular channel. And at time zero, if you look above with air, that's what the chip looks like at the top right. Now, when you perfuse this through the bottom and you watch over 2 to 4 days, you see a meniscus of fluid filling the airspace, and you basically have pulmonary edema on the chip. Now, we quantified this with a trick from kidney physiology. We used fluorescent inulin, and what you can see with the squares at the top is you have influx of fluid into the airspace, as you just saw, but primarily only with breathing motions, physiological breathing motions. The red are without breathing motions, and it's there, but I don't know if we would have believed it. And we went to an in vivo animal model, ex vivo ventilation perfusion model in a mouse, where the heart's pumping, but you have the lung opened up and you're ventilating. And we actually confirmed that this is true in vivo as well. Now, I'd worked for years on mechanobiology, the idea that mechanical forces are as important as chemicals and genes. And we had discovered that the earliest signal we could see when you stretch a substrate, a flexible substrate with cells adherent, is that forces go through integrins to an ion channel called TRPV4, and that lets calcium into the cell in 5 milliseconds. And GlaxoSmithKline, I knew, was working on an inhibitor. They gave it to us. We completely knocked out pulmonary edema in this model. They, in parallel, did studies in dogs and mice with cardiogenic pulmonary edema due to back pressure, not a drug. And they got the same result. We had back-to-back papers in Science Translation Medicine, and that drug moved on to clinical trials. Now, one nice thing about these devices is because you have a healthy endothelium, you can perfuse whole human blood without anticoagulants, at least for about 15 to 20 minutes, which is what you're seeing here. That little movement is blood cells. And we use this approach to show that we could predict toxicities due to thrombosis that were not detected in animal models. So there was a drug, a biologic developed for lupus that was called basically therapeutic antibodies, HU5C8, for example, that targeted CD40L. And in animals, totally safe, but in clinical trials, thromboses in the lung and other places, clots. And using fluorescent platelets and fibrin, I won't go into the details, we could mimic this beautifully, and we actually could get insight into the mechanism behind this. We then developed a bone marrow chip. Here we take CD34 hemopoietic progenitor cells from circulating blood or bone marrow. This time we put it in a matrix gel, filling the top channel with bone marrow-derived stromal cells. And then we have endothelium in the bottom, and we perfuse only through the bottom, like in marrow. In the bottom here are flow cytometry showing you get near normal neutrophil maturation over a month and near normal erythroid maturation over a month in vitro. And the Giemsa sustain at the right shows you get many different types of bone marrow cells forming. Now, AstraZeneca saw this and came to us because they had a very peculiar regimen-specific toxicity with a cancer drug in phase 1 clinical trials. Over 2 hours, they got neutropenia and anemia. And over 2 days, same dose, they only got neutropenia. And they were having a hard time modeling this in animals. Now, they had measured the pharmacokinetics, how drug levels change over time, which is shown in gray in the clinical trial. And because we have flow, and this is really important about the chips as opposed to organoids or other NAMs, we can mimic the drug exposure profile precisely. That's the circles and black lines. And when we do that, we mimic this regimen-specific toxicity beautifully. Now, probably the most dramatic and impressive, convincing results with toxicity predictions of organ chips being better than animal models is a study done by Emulate, which I will disclose at the end, as a company I formed that commercialized this technology. Started in my lab, moved there, so I was part of this study. We did 870 human liver chips with primary cells from 3 different human donors. Now, these chips have primary hepatocytes in the top channel in a matrix gel, and then the bottom has primary human liver sinusoidal endothelial cells, Kupffer cells, and stellate cells, all primary in the right locations. Now, we tested 27 different hepatotoxic and non-toxic drugs, that most of which were promoted by the IQ Consortium, which is a pharmaceutical consortium. It was published as a list of benchmarks to see if you can predict a NAM is better than an animal model, because they knew the results in animals and humans. And what we found is that these chips predicted small molecule drug induced liver injury, DILI, with 87% sensitivity and 100% specificity. This was 7 to 8 times better than animal models for the same drugs. And we did an economic analysis, and that estimated that just preventing late stage failures due to DILI would save the pharmaceutical industry $2 to $3 billion a year. Now, these data were included as the first organ-on-chip technology accepted in the FDA's iSTAND program, which is their program designed to basically approve NAMs of any type. And that is now in its very final stages. It is being repeated by 2 companies with a commercial product. And if those results look as good as we hope, it would be approved, and then any company in the world could include data from this liver chip about DILI with a small molecule, that specific context of use, in lieu of animal data. Now, the other thing in terms of interaction with animal models is that Moderna was started to use these chips instead of non-human primates. And as you know, non-human primates are very hard to come by after COVID, very, very expensive. And they found that not only could they get results that were more human relevant, and this was down selecting lipid nanoparticles for non-fibrotic, for fibrotic toxicities, essentially trying to identify ones that don't induce fibrosis. And they found that not only were they more human relevant than non-human primates, it only cost 1/10 the price, and they did it in 1/4 the time. And my hope is that this is the type of finding that would get the industry to really take this over in a bigger way, as well as NIH in terms of funding research programs. So now I'm going to move to disease modeling, drug efficacy, and drug resistance. So drug companies said, well, the alveolus chip was interesting, but they're more interested in chronic disease like asthma and chronic obstructive pulmonary disease, COPD. So we made those affect larger airways. And so we made a chip now with bronchial epithelium and endothelium, and viewed from the side, you see beautiful cilia moving in a directional way. At the bottom right, these are fluorescent microparticles showing you mucociliary clearance, a clearance in the mucus due to the cilia motions, and it's the same rate as in all of our lungs at this very moment. We then developed a cigarette smoking robot. This is a cigarette lighter from a car, a Gatling gun with real cigarettes, and we actually can control smoking parameters to mimic human pulses and pauses and breathing, and have real smoke go to the airspace of the chip, not extract. And this is just a couple of examples. If we look at IL-8, a major inflammatory cytokine, Healthy chip, smoking didn't do much. A COPD chip, chip lined with cells from patients with chronic obstructive pulmonary disease, doubling of IL-8. And at the rate, we did transcriptomic analysis, and I should note, we've done proteomic, metabolomic, glycomic, transcriptomic with these chips. You could do anything you could do in animals or in vitro with these chips. 3 different human donors, healthy people, looking at changes in genes after smoking versus before. And then the 9 at the left are from a clinical study of healthy people who smoke cigarettes. And we were really excited that the top quarter versus bottom 3 quarters, very close resemblance. Paper got turned down because, patients are more variable. Then we realized that we actually could do this type of study better than a clinical study, because this is matched comparative modeling of the same patient before and after a stimulus we're interested in, smoke. And so these changes are due to smoke. The ones at the left clearly are due to smoke, but also family history, working history, home environment, you name it. And it finally got accepted. And when we were asked to do a commentary on modeling, matched comparative modeling in vitro. NIH and DARPA both came to us in 2017 because it was the centennial anniversary of the influenza pandemic in earlier century, saying, could you model potential pandemic viruses like influenza? So turns out it's a great model. GFP virus at the bottom left, you see infects the cells. You see breakage of ZO1 junctions, loss of cilia. Because we have endothelium, you could even see endothelial damage. If we looked at the viral load and we compared different strains, H1N1, H3N2, H5N1, we get different virulence, similar to seen in patients. And now we could also explore comorbidities, which you don't explore in animal models usually, but comes to haunt in clinical trials. You can see that a COPD chip infected with these viruses has a 10-fold higher viral load. And these patients are known to be much more sensitive to viral exacerbations. And I should note that this shows you the importance of primary cells or adult stem cells, if you use organoids, because they retain epigenetic signature, which I worry you will lose with iPS cells. We also could show you can measure what is effectively a cytokine storm, as we now call it. These are different viral strains, IL-6, IP-10, Rantis, interferon beta, MCP-1, IL-8, and this matches very much the virulence responses that's seen in patients. At the bottom, you see neutrophil clearance of virus, as I showed you in the video. And the bottom right, you see drug testing. So oseltamivir is also known as Tamiflu. That is the first-line drug approved by FDA, but it's only approved for the first 48 hours after influenza infection because it loses activity. When we tested it, beautiful inhibition, but only for the first 48 hours on chip, just like in patient. And then my postdoc, Long C, did a really interesting experiment. He asked, could we predict evolution of spontaneous variants of concern? So here we have a lung chip infected with virus influenza, and we gave it amantadine, antiviral drug, but only at about 80% efficacy level concentration. And then after a couple of days, we mimic coughing by having a droplet of fluid go through the airspace and pass to the airspace of another lung chip, treated the same way, and we did this repeatedly. And after 8 passages, we got virus that was completely resistant to drug. When we did whole genome sequencing, we found all of the mutations that were previously described clinically in patients who became resistant, but we identified a couple of others that no one's ever seen before, which raises the possibility of being able to identify mutations before they happen for vaccine and drug development. Now, I've worked in cancer since the 1970s, and in the cancer biology research in the 80s, people came up with orthotopic models where you put a mammary tumor in a mammary fat pad or a prostate tumor in a prostate gland, and it looked a little bit more like in humans. So we had these different chips, so we said, could we make an human orthotopic cancer model? So we got human non-small cell lung cancer cells, adenocarcinoma form, H1975, that clinically emerge in the distant bronchiole, a small airway, but they preferentially grow throughout alveoli, and that's how people die. And so we plate the cells, GFP-labeled tumor cells, at a low ratio or high ratio, small number to healthy. So it's in a host environment. And then we quantify growth. And this is really interesting. Top right, the gray circles show you the growth of these tumor cells in a plastic dish in serum-containing medium, the way they've been cultured for 50 years, exponential growth. The white circles show you the same cells in the same dish, but we're using the medium that we use to feed our chips only through the lung channel. The vascular channel, because there's air in the other channel. These tumor cells don't grow practically at all in this medium on a plastic dish. So all the growth I'm going to show you is microenvironment, human microenvironment dependent. So in the airway, where they form in vivo but don't grow very quickly, they don't grow at all for 10 to 12 days, and then they grow slowly. But in the alveolus, where they grow in vivo rapidly, they grow exponentially from the beginning. And then we did an experiment where we did plus or minus breathing motions, because we're interested in mechanics, and it turned out breathing suppressed growth by 50%. And it's very easy to measure invasion in these chips, because you can watch the GFP cells go through these pores, as you can see, and breathing inhibited invasion by 50%. Turns out breathing also inhibited therapeutic response. So if you have no breathing where the tumor grows steadily, you suppress growth, it flattens. If you have breathing where it grows slowly right through the drug. This is ruxolitinib, it's a kinase inhibitor, and we showed mechanistically this works, that the breathing regulates EGFR receptor phosphorylation. So we've done many chips, as I told you. We moved to intestine chips, and now we take adult stem cell-based organoids from a biopsy or a surgical resection. We grow them like all other organoid people in Matrigel, where they form a ball of cells, they proliferate, you can freeze them, it's wonderful for expansion. But you can't do things like measure absorption, transport, mucus, microbiome very easily. So we break them up, and we put the epithelial cells in the top channel, and then we put endothelial or stromal cells in the bottom channel to create tissue-tissue interface. And you can see here that you get beautiful intestinal villi viewed from above or the side. When we did transcriptomic analysis and we compared the organoid in the center from a duodenal organoid at the left to I just lost control of well, I'll leave that. The organoid from the duodenum versus the duodenal organoid-based chip versus duodenum in vivo, the chip was significantly closer based on transcriptomic analysis. Now, this paper came out 2 weeks ago in Nature Biomedical Engineering. Here, we made similar intestine chips, but we got organoids from patients with inflammatory bowel disease. And not only did we make organoids from the epithelial stem cells, but we took their stromal cells from the same biopsy. And now we make chips with a stromal epithelial interface. And at the bottom left, you could see healthy villi, or these are colons, so crypts and vertical structures. In Crohn's disease in the center and ulcerative colitis at the right, you're flattening, the cells are becoming flattened, or you're losing goblet cells, which are the cells with the lighter oval circles in their apex. You have increased inflammation, looking at inflammatory cytokines at the bottom right. We can look at mucus production in these chips by just turning them on the side and doing dark field imaging in live cultures. And you can see at the top left, the mucus in healthy compared to the bottom is thicker in the healthy than the IBD. It's well known in patients. We did this plus or minus peristalsis, and we discovered peristalsis is actually a major driver for mucus production, but it's always lower in IBD compared to healthy. And then because we have stromal cells, we can look at fibrosis by looking at second harmonic generation imaging. And here we see that only when you have peristalsis, physiological peristalsis, do you see much higher fibrosis in the IBD patients, as shown at the bottom right. Now, women who have IBD, if they want to get pregnant, have to get the disease under control first, because you can get terrible exacerbations. So we got female IBD chips, organoids and fibroblasts, and we made those chips and we exposed them to pregnancy hormones. And only when they exposed to pregnancy hormones do you see this increase in inflammation at the top, all those reds, and the bottom right, that is fibrosis only in IBD chips, not in healthy. And this may be why they have exacerbations. The other thing you could do with these chips, you could do tissue recombinance, sort of a developmental biology approach. We make chips with healthy epithelium and healthy stroma, IBD epithelium and IBD stroma, or healthy epithelium and IBD stroma or IBD epithelium, and healthy stroma. And what this revealed is that the stroma is the major driver of inflammation. Look at barrier disruption, inflammatory cytokine production. It's the IBD stroma that is the critical determinant of this. And this is important because all the drugs are designed primarily against epithelial products and immune cell products. And this is a whole open area for targeting. Now, the last thing we did is it's known that IBD patients have an increased propensity for cancer formation. So we exposed these chips for 3 weeks to a carcinogen, ENU. And what we found, amazingly, is that only the IBD chips express increased inflammation, early cancer initiation markers like down modulation of E-cadherin and upregulation of nuclear beta-catenin and upregulation of CEA-CAM5. And that also is stromal fibroblast dependent. And then we did shallow pass genome sequencing, and only the IBD chips showed increased mutation and gene duplications, again, consistent with early cancer formation. This is studying human carcinogenesis, the earliest stages in vitro, which I have not ever seen done before like this. So these are a couple of examples of our scores of papers, but it shows you that we realize that organ chips provide literally a window on molecular scale activities inside living human cells within a relevant tissue and organ context. And so this meant it could reveal new disease mechanisms, as I showed you, but also enable drug discovery and provide a way to experimentally test those predictions. So during COVID, we were using influenza as a model because we didn't have a BSL-3 lab at the time. So we were testing influenza in the air alveolus chip, and the postdoc did that breathing experiment, plus or minus breathing, and found that breathing suppressed viral infection dramatically, as shown at the left, and inflammatory cytokines. And he worked this out mechanistically and found that breathing activates innate immunity. It's a potent inducer of type 1-3 protective interferons, which are known to be antiviral. By the way, they're also anti-cancer, which may explain our cancer results. And in the middle, when he did transcriptomic analysis, there was this one gene, S100A7, that was screamingly high, that also we confirmed is screamingly high at the protein level. And it is a ligand for RAGE, or the receptor for advanced glycation end products, which is highly expressed in lung and major mediator of inflammation, which obviously in COVID was a major problem. We found a drug called azilurigone that is a RAGE inhibitor. It was in phase 3 clinical trials for Alzheimer's, oral drug, safe in 2,000 patients, missed its mark in Alzheimer's. We were able to get the drug from the company. We completely knocked out cytokine storm at the top right. Those very data were included in a pre-IND application of the FDA by Cantex that owns the drug to start COVID-19 trials, which they moved on into. Then my own group postdocs did a siRNA screen looking for genes that may mediate host tolerance to infection. Rather than targeting the virus and having to worry about mutations and becoming resistant, why not increase the host ability to protect itself against viral infection? And they did a siRNA screen, and they found a bunch of RNAs that worked. And then they made control RNAs against the same target, and they didn't work. And what we found out is that all the RNAs that worked had the same structure. They turned out to be short, duplex, double-stranded RNAs that turned out to be potent inducers of type 1 and 3 interferons, interferon alpha, beta, lambda. I'm showing you beta in the middle on chip, airway, epithelium, and alveolar, potent induction. But they turned out to be broad-spectrum antivirals. So they work against in vitro here, I'm showing you N1 influenza, H3N2 influenza, common cold virus, it's a coronavirus, MERS coronavirus, SARS-CoV-2 coronavirus. And based on this, we did an animal model in a mouse SARS-CoV-2 model in a BSL-3 lab, and we got a 5-log reduction in viral infection using this, which that lab had never seen before. And based on this, we have an ARP-H grant now to look at both virus, but primarily cancer because of the antiviral effects in cancer. And now we're delivering a newer version of this drug, and we've engineered the structure of these RNAs so that we've got a 100-fold increase in potency now. And this is not even the most potent one, but we deliver it with lipid nanoparticles as they would be in humans. And here you're seeing dose-dependent inhibition of cancer growth, GFP at the bottom, quantification in the graph, that is really quite beautiful, and we've confirmed this in mice as well. We've also developed, with funding from FDA and BARDA, human organ chip models of radiation toxicity, acute toxicity, and this was asked of us because animal models don't show you the same radiation dose sensitivity at human or the same manifestations. We get the exact same dose sensitivity as humans in manifestations. So we've done lung, bone marrow, intestine, and now lymph node. I'm not showing here, where the bone marrow is most sensitive in the 2 to 4 gray. The lung is most resistant in the sort of 10 to 20, and the intestine is in between. But we've been developing drugs against radiation countermeasure drugs because pretty much the only approved drugs worked for bone marrow. And so we use CRISPR screens for this. We basically have a whole kinome scale CRISPR screen. We take, in this case, endothelial cells, because mechanistically we discovered the endothelium is the major target of radiation toxicity, and others have as well. And we basically screen for which cells survive when you're exposed to radiation. And then we can identify the kinase, and then we can find known kinase inhibitors, so that we identified CLK2. We found a drug, TG003, and a couple of drugs, and they beautifully predict radiation toxicity. Here, you lose intestinal villi in the small intestine chip, and you get it back with villus height, as you can see at the right. And this same drug, because it targets endothelium, works in intestine and lung. And this, we hope that this may provide a new path to radiation countermeasure drugs. So now, modeling host microbiome interactions, we created a hypoxia gradient using a chamber where the oxygenated medium goes to the endothelium and nitrogen gas is on the other side in the upper channel. So we can get it, and we have oxygen sensors in this, so we can get down below 0.5% oxygen in the lumen of the epithelial channel, so you could have obligate anaerobes survive, but the gradient is enough so that the human endothelial cells survive and aerobes survive closer to the epithelium. And using this, you could literally take a stool sample, dissolve it, and put it in the lumen of the chip, the epithelial lumen, and you get, over at least 5 days, you get similar complexity, over 200 different types of microbes surviving on these chips, as you see in stool. And the right, all those little speckles between the villi, that's how dense it is. And actually, if it's a healthy microbiome, you get better barrier function. If it's dysbiotic, you lose barrier, get inflammation. A Gates Foundation came to us and said, could you model kids with environmental enteric dysfunction, EED? This is due to malnutrition in low-resource nations. It has multiple contributors: malnutrition, dysbiosis, microbiome, pathogens, immune, etc. And so we got organoids from kids in Pakistan with EED and then healthy, and we mimic nutritional deficiency, an environmental contributor, by taking out nicotinamide and tryptophan out of the medium. And what you can see is you get villus blunting, whether a healthy chip or ED with nutritional deficiency, and that's seen in patients. But when we compared healthy versus ED with absorption, I'm just showing you fatty acids, but we've done many other things, you get much higher loss of fatty acid absorption in the EED chips. And we were lucky that they did transcriptomics on the biopsies of the duodenum from these kids. And what you see at the right are comparisons of the left lane is the clinical data, things that went up in the EED kids versus down in blue. I'm not going to go through all these, but what we found is only if you had the chips lined by organoids from kids with EED and nutritional deficiency did it come close to what you see in the patients. That's the second lane, red at the top, blue at the bottom. If you have EED organoids and complete medium, you don't see this. If you have healthy organoids on chip with nutritional deficiency, you don't see this. And then Jeff Gordon was able to give us a microbiome that he had obtained from kids with EED. And now you add that, and now you have a different environmental factor, and now you get much higher inflammation in red, and you get higher intestinal permeability, meaning breakage of the barrier when the microbiome is there, and nutritional deficiency rather than either alone. Now, a lot of people say there are no, good NAMs for lymphoid for immune system, but years ago, and now a series of papers, that one of which second is in press, we developed basically a lymph node model. We take B and T cells from blood, from apheresis colors, put them at high density in matrigel in one channel, and perfuse through the other channel. And if you vaccinate with a commercial flu zone vaccine, influenza vaccine, and you have dendritic cells in there as well, you get germinal center-like structures spontaneously self-assembling. They express AID, they form plasma cells, and they undergo antibody class switching. You get high affinity IgG against the antigen. We can collect effluents from the channels and basically measure cytokines like a blood sample. We were lucky to have clinical data from people who were vaccinated with Fluzone, and we got very similar profiles on chip as in patients. We've now used this for adjuvants. We confirmed that there's somatic hypermutation, so that you're really getting antibody, reorganization, chromosome reorganization and production. And we've done this with multiple types of vaccines. We've also modeled Lyme disease and cancer as well. Finally, personalized medicine and clinical trials. A year ago, we published a paper with a clinician in McGill, Lorenzo Ferri. He treats patients with esophageal cancer. He's gotten surgical resections. He makes organoids of the tumor epithelium, and he collects stroma from the same tissues. First thing he did is compared the mutational expression, and that circle at the bottom right is showing you that although organoids are much better than other types of culture for retaining cancer-specific mutations, the organ chips actually were better. And then he asked, could he predict patient responses using these chips? Because his patients become resistant to therapy. It's a combined therapy, and he has to choose new drugs, and he has to basically wing it. And could he use this sort of model? So if you try to mimic the clinical drug exposures, 2 days with the right doses in organoids, they die because you're just bathing them in it. But if you perfuse them through the stromal channel of our chips, like it's coming through the interstitium, like an IV drug, you beautifully predict patient responses. Those are the patient responses are blue, but the red is what the chips predicted. If you give it to the epithelial channel, it's like the organoid, they die. So you have to give it to a physiological distribution pattern and a clinically relevant dose. And he's hoping to use this to select drugs for his patients in the future for personalized medicine. And then when I was first funded to do this, I actually had written a review in 2012 where I said, because we have an endothelium line channel, it's not just one channel, it's two, it's an organ chip, not a tissue chip, we could connect them potentially and create a human body on chips. Imagine an oral drug through the lumen of the gut chip, watch it be absorbed, see it metabolized by the liver chip, watch it pass to the kidney and be passed out, and see what it does to heart, lung, etc. And we were funded by DARPA to do this, and we do this with a robotic sampler and not tubing because there's too much dead space. We could do one drop between chips and then one drop for mass spec analysis for drug levels or cytokines or metabolites. And in this published studies, we did gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas. We kept them alive for a month. And we used a single blood substitute medium because we had endothelium in all chips, and we could perfuse the other channels with other medium as well. And then the reason DARPA wanted us to do it was to carry out in vitro to in vivo translation of PK, pharmacokinetic parameters. Could you predict drug pharmacokinetic parameters using just human cells on chips and computational PK/PD modeling, which would shortcut clinical trials design because you know what doses to start or what regimen to start with. And what you really want to predict is the Cmax, the maximum plasma concentration, and the T1/2. And so to do this, we simplified. We did a first pass model of gut, liver, kidney, and we put an AV arteriovenous reservoir so we could take samples from it, like a phlebotomy sample, rather than a distal vein, rather than after one of the organs. And that worked unbelievably well. It was also published in that same issue. The top, we have oral drug oral drug nicotine. We put it through the lumen of the gut chip. We use mass spec to measure its levels and its metabolites in every chamber. On the bottom, we used cisplatin, an IV drug. We put it into the AV reservoir, and we did the same thing. At the right, the dotted lines are computational PK/PD predictions of pharmacokinetics of these drugs. We did this with CFDRC. It's a company that has done computational PK/PD for pharmaceutical industry. They knew tissue masses, blood flow rates, so we could scale from our tiny little chips up to the human body. And the dots with error bars are from clinical studies. In the top, nicotine snus is chewing tobacco or gum. In the bottom, IV cisplatin, 2 different infusions. And you can see we precisely predicted drug PK parameters. And so to end this part, drug companies now will spend hundreds of millions to develop a drug, 15 years, 10 million on clinical trials for a few years. They usually fail. They then have a statistician see, is there a subgroup that responded better? If they're lucky, they do a targeted, genetic subpopulation and they get approved for a narrow application. With organ chips, organoids, iPS cells, you can make, you can flip it on its head. You could do 50 to 75 patients that are very similar, Hispanic women with COPD, and you test their lung chips and find 50 that work well, and then you test their other organs and go down to 40 because of toxicities, and use those 40 patients in a small trial. It'll be faster, be cheaper, be more likely to succeed, and I think this would just flip the paradigm. And then to end, I want to show you how we actually integrated AI with organ chips and animal models to get really powerful results. So we developed our own machine learning AI-based platform called NemoCAD that also has gene network analysis capabilities hooked into. You start with transcriptomic data from patients, any disease you want versus healthy. You basically are asking, how do what thousands of genes do you flip to go from disease to healthy? It accesses all the databases for FDA-approved drugs. It also has gene network analysis and gene drug network analysis. It sort of maximizes all this, and it gives you a list of drugs that should normalize a disease state. And during COVID, this came up with a prediction that a subset of statins, but not all statins, should increase survival in COVID. We didn't know what to do with that during COVID, but a couple of years later, we collaborated with Stanford that did a retrospective study of 4,000 patients, and it turns out this to be true. And we now do this and we validate in organ chips. And so one example is another DARPA's program called Biostasis, where they wanted to develop drugs, injectable drugs, that would basically induce a state of suspended animation that would slow metabolism at all levels. And you're trying to mimic things like torpor, hibernation. And so we did this using transcriptomics from animal torpor studies. And now we're using a novel animal model, which is the Xenopus tadpole, which has a much higher developed immune system in CNS than Drosophila or C. elegans. And we screen in these tadpoles. And here you can see that when you give this drug that was identified, they stop moving, they slow, but it is totally reversible. We used network analysis to figure out what's going on. We tested in organ chips to validate slowing of metabolism. We then moved to a pig model, a heart transplant model, where it's whole hearts taken out in a device where they're perfused, and we showed beautiful slowing of metabolism, decrease in oxygen consumption, lactate, lower inflammation, higher reactivation with beating, and that is actually now funded by ORIP to move to human hearts. Having this system, and someone came and basically said their child has a disease called Rett syndrome, a daughter, which is a form of autism primarily in girls, repetitive motion, seizures, and at least one form of it, there's a known gene. So with Xenopus, you can make CRISPR models of disease models in 2 weeks. And we did that. And on the right, what you see is, on the far right is a control. These tadpoles that are normal, they move around the edge in a consistent way. When you knock out this gene, you see these repetitive motions, and they just stop moving, which is actually a seizure because then they start moving again. And we then found a drug using this AI-based approach. That drug, it turns out, an animal model that was used to move the one drug approved for the clinic, trofinetide, is much better than that drug in the same mouse model. It also works after the symptoms start, whereas the other drug you have to start before. And these kids, the parents are more worried about GI symptoms rather than nervous symptoms, because that's what really bothers them. And this drug also addresses that. And that drug, we formed a company called Unravel to commercialize it. And that just started clinical trials a couple of months ago in 2 different neurological conditions. It's actually been used by physicians on a compassionate basis with a couple of kids, and it's worked beautifully. So a take-home message from all this is that organ chips are more than potential animal replacements. They're also mechanistic drug discovery tools and test beds for validating newly discovered therapeutics. The power of AI is much greater when you combine it with really good human relevant models such as organ chips. And then I think, clearly human organ chips, animal models, and AI are synergistic approaches that we're going to be able to use to confront major challenges in drug development. We're going to be using all of these for a long time. I hope that more and more we can reduce animal tests, but I think, this is where we are today. So with that, I have to say you can't believe anything I tell you, because I hold equity, sit on the board, and chair SABs of Emulate and Unravel, and invite you to the website if you'd like to learn more. So thank you so much, and I am happy to answer questions if there is time.
Thank you very much, Donald, for very comprehensive review of the field. We have a couple of questions we would like to ask you. We have time for that.
Yes.
So question number 1. So someone is asking, I believe the alveolus microstructure includes type 1 and type 2 pneumocytes, alveolar brush cells, alveolar pores, fused basement membranes, alveolar macrophages. Could you remind again what this lung chip includes? And I mean, it's a general question, if any type of cells are missing or they are not on the right stage of the development, how we address this question.
So the alveolus chip has type 1 and type 2, and we have paper in review with macrophages as well. And we've obviously showed you can put, circulating immune cells as well. You got to realize most people approach this field that they're trying to mimic the complexity of the organ. We approach this trying to get the simplest model that mimics the clinical phenotype that we're interested in, because that's what a model is. You're trying to simplify so you get the fundamental features. So this model is obviously a good model for influenza infection and pulmonary edema, but if you want to model pulmonary fibrosis, there are no fibroblasts, so it's missing. Now, we've integrated fibroblasts later when we looked at fibrosis, so you can do that. But it depends what your question is. So you don't want to put everything in there. You want to put the minimal in there that use clinical data, clinical response as your benchmark. If you mimic it, it's simpler than you thought. So, we have found again and again, for example, that you don't need as much complexity as people think you need. People think of COPD as an inflammatory disease, right? Immune cell based, but I just showed you no immune cells. The epigenetics still gives you a lot of the disease phenotype. So you really have to design these to be models. They're not exactly the same, but you have to. This is why FDA emphasizes context of use. You have to, that liver chip is great for small molecule induced lung injury. It may not be, it's probably not, has to be redone for monoclonal induced drug injury.
Thank you. 2 questions which are related. Which models have incorporated lymphocytes? How do you address histocompatibility and limited viability in vitro? And related question, lymphoid organ chip can mimic antibody development following antigen exposure. Do you have a chip that can similarly mimic the process of T-cell development and maturation in response to antigen stimulation?
We published that you can use naive antigens with that chip and get, full immune response. We've modeled Lyme disease where you get hypergammaglobulinemia, T-cell suppression. We've done, we've made a muscle chip for intramuscular vaccination and then link it to the lung chip. Yes, we link it to the lymph node chip and show we can get the full vaccination response with self-replicating vaccines. We've vaccinated the lymph node chip with influenza and then connect it to the lung chip and then infect the lung chip with virus and show that we have protection. We have B cells for a month, B and T cells for a month in these chips. So, Yeah, I mean, we've done all of those things. Is it perfect? No. Can it be optimized more? Yeah. And we hope to do that, but it's been pretty good at what it's done so far.
Thank you. One more question. Can you say something about sample size and power for those studies? In many published papers, the sample size are very small for NAM studies. Experiments run on plates may have wells that do not produce useful results. How do we get to a real assessment of the utility of those models?
So that's a good question. I mean, the emulate system has been an instrument that does 12 chips at a time, and you could put about 3 in an incubator. So, 24 to 36 at a time. So usually we do triplicate or quadruplicate. And then we repeat it in, multiple donors. But the study with the bone marrow chip where we looked at drug toxicities for 5-fluorouracil, so we had 6 different donors, and the reproducibility was incredibly good. I would say the reproducibility with the liver chips for the iSTAN study is certainly as good as animal models. But the animal models are inbred. So like the lymphoid chips are obviously have a greater variation because of immune response. And so you need to do more. You need to do more donors for that. But Emulate is now came out with a 96 chip instrument that is an incubator with imaging, and that's going to give us much higher number, and that gives better reproducibility as well. So and there are other commercial organ chips that now have higher number also. So I think we're moving in the right direction, but that is a challenge. You have to do the downside that pharma doesn't like is they don't get all the beautiful reproducibility of an inbred mouse or a established cell line, but then they go to a clinical trial and they fail all the time because of variability. And so you can look at that variability with chips. You can select subpopulations to focus on to move to the clinic.
Thank you. The next question is, any chips in your plan for eye diseases in the field of ophthalmology?
Years ago, I can't remember his name, but I collaborated with someone at NIH who was developing IPS, retinal pigmented epithelial cells, and he went to the clinic with it. And we started to make a chip with endothelium, optimal, eye endothelium and RPEs, and it looked good, but I just don't have funding to do it. But it's definitely doable.
Very general question, kind of maybe naive. If no one ever used your system, where to start?
If no one ever used our system,
I mean, yeah.
If you wanted to start using that,
yeah.
I mean, it really depends on the quality of cells that you have, and we always start with the same culture medium that's used for 50 years. You know what I mean? And when you have different cell types, sometimes you have totally different media. And there, sometimes we have to do like, when we have a lung and we have different cell types, we only have one channel with medium. So then we have to do like a 1-to-1 mix. But for the most part, you need to get matrix coatings that will get the cells initially adherent, but then they put out their own matrix. You don't have to get the matrix precisely right. You need to get the medium right. And we always start by mimicking the physical environment of in vivo. So if it's an intestine, we'll look at the literature and find out how much stretch and at what frequency. The lung the same, the kidney the same. And with that, we've been incredibly lucky. I mean, we haven't had a chip we couldn't build.
Yeah, so in principle, what probably you're also saying, if system, even if it's organ on a chip, it's still becoming more complex and complex, right? Quite sophisticated. So maybe for such people, it will be better idea try to get already ready to go chip, right? Which is prefabricated for them. What can you say about
You mean, I think the long range goal of all these companies are to sell chips with cells on it already. If that's what you're intimating, I think that would be great. That certainly has been thought about, but that requires a lot more funding and development. But Emulate sells a liver chip kit, an intestine chip kit, and a brain chip kit. The cells have been qualified, the medium has been qualified, and their methods are, published and on the web. And that's what these companies around the world are validating for iStand, so that they're not using their own cells for that. That is a commercial product. But they still have to plate them. They're not already on the chip.
Okay, so the last question, big one. How do you see everything in 5 years?
How do I see everything in 5 years?
Yeah, AI, NAMs, human systems, maybe some animal models for certain application. How do you see this in general? What are you dreaming about?
I mean, I see AI bringing in more like electronic health record data, clinical data, and counterpointing it with the sort of AI discovery algorithms, and now coming up with predictions, but then iterating with experimental models like organ chips. And then the other is like this emulate system has constant fluorescent microscopic imaging of these 96 chips over time. So now you're going to have a huge amount of data for AI to analyze that's morphological, or if you have reporter constructs. I think integrating more sensors into chips will give you even more data. So I think that's when AI will really be valuable. I think the animals, are going to, the more we cross-validate, the animals with AI and organ chips and human data, the more we'll see, maybe a subset of animal models are really good, but a lot of them are not. And maybe we will stop using the ones that are not good and start, reducing what we use. That's how I kind of hope to see it. And of course, I hope to see, my dream would be that one company does what I say. They use chips to design a drug for a small subgroup, and they go to the clinic and get it really fast, because I think that'll flip the whole industry.
Okay, thank you very much for your excellent presentation and nice discussion. Right now we are moving to a break, so we will come back at 10:20. Thank you.
Thank you.
Welcome back. We are moving to our first session. The title of the session is The Need to Complex Integration of New Technologies to Study Biological Mechanisms of Human Disease Progression, Complementary Use of Animal Models and New Approach Methodologies for Preclinical Studies and Therapeutic Development. Our co-chairs are Rebecca Roof and Jason Shepard, and let me briefly introduce them. Rebecca Roof is a program director at Division of Translational Research within NIH, where she oversees the innovation grants to nurture initial translational efforts program. This program supports early stage therapeutic development as well as disease model development and validation. This includes both in vivo and in vitro models. Jason Shepard is a professor in the Department of Neurobiology and holds the John Huntsman Presidential Endowed Chair at the University of Utah. His team employs an interdisciplinary and collaborative approach to understand the mechanism of learning and information storage from molecular level through in vivo neuronal networks and how those processes are disrupted in neurological disorders. Please take your microphones.
Thanks, Oleg. And thanks everyone for joining in, and welcome to our first session. I wanted to emphasize that this first session is talking about integration and complementary integration of animal models with NAMs. All right, so our next speaker is Dr. Takanori Takebe, who is a professor and director of commercial innovation for Center for Stem Cell and Organoid Medicine at Cincinnati Children's Hospital Medical Center. His lab seeks to develop and apply novel approaches and tools to recapitulate, probe, and manipulate human liver development and to understand personalized molecular mechanisms that lead to disease. His title is Organoid NAM Guided Precision Hepatology. Thanks, Takanori.
Thanks, Jason. If you can't hear me, just interrupt me. But I'm Tucker from Cincinnati Children's Hospital. And first and foremost, I would like to thank my old colleagues at the Center for Stem Cell and Organ Medicine in Cincinnati Children's and many collaborators, across US. So just to start off, as a way of introducing ourselves, so we are from, CUSTOM, or Center for Stem Cell and Organ Medicine, that was established in 2017 by my close colleagues, Alan Zorn, Michael Helmut, who is going to talk in the next session, as well as Jim Wells. And now we have 38 faculty labs associated with the custom, ranging from the brain down to, other, skeletal tissue model systems. We have aggregated many different sort of organ portfolios. What we think is really unique is having the infrastructure for supporting the dissemination of the technologies as well as de-risking of the technologies, which is oftentimes a problem around the organoid technologies. For example, since 2007, Jim Wells as well as Chris Mayhew opened up a first in-kind, iPS cell stem cell core. This is really vital for us to expand the human pluripotent stem cell portfolios for accessible to many colleagues in institute as well as other institutes as well. And in 2019, we recently launched another sort of infrastructure called the Custom Accelerator Lab, whereby we have free automated sort of manufacturing facilities, including the high throughput screening capabilities. So I think this kind of transitional infrastructure is really essential for us to de-risk the technologies before ready for the commercial transformational cycles. And obviously, we are embedded in a children's hospital, so we have ample access to clinical data sets, whereby we access to the patient's, pharmacology data sets as well as iPS cell lines. So in my lab in particular, is interested in trying to create the human liver tissues from the iPS cells or embryonic stem cells. But oftentimes we, are dealing with the problem of how complex is enough to study your interested biologies. Obviously, from, simplest model of the 2D monolayer directed cell differentiation approach, which is still our sort of favorite systems of, the simplistic model for study some aspects of the biologies, including transwell-based type of approach. But some of the biology, for example, canaliculated tissue architectures, which is actually sort of structures in between the hepatocytes, we need actual 3D organizations for that type of problems. Epithelial organoid model system embedded in matrices in most cases. We now are able to form, interhepatocytic canaliculated architectures, whereby we could, control the polarized, hepatic functionalities. For example, towards the canalicular side, we can sort of eliminate the waste metabolites, whereas the basal or basolateral side, we can secrete many, important protein synthesis ability as well as secretory capacities. But not limited to the epithelial architectures, we obviously need more, complex components, including vascular cells or mesenchymal stromal population, and then down to the immune cell populations. For this type of tissues, we think, iPS cells or pluripotent stem cell type of technology is really powerful in, co-developing the other sort of, intertissue components. So we actually put so much efforts on the multi-tissue type of organoid model systems over the past 10 years. But ultimately, liver is not isolated from the other organ systems. For example, if you have a bioduct sort of connectivity problems, liver is massively damaged. So for that type of, interconnectivity problems, we, actively employ a symbioid type of a system to try to connect the neighboring systems, including, bioduct, pancreatic tissues, or duodenal architectures. So to really study the, ultimate goal of, human disease model, we definitely need to study what would be the minimum components. Because if you increase the complexity, it could be, informative for the physiological perspective, but it's going to be compromising the throughput or, scalable perspective. So we need to choose the right complexity model for the downstream application systems. So, I think not to mention here, this is sort of review articles from EMA's sort of questionnaire-based survey and identifying several, promising areas of in vitro now-based approach. And obviously always one of the sort of exciting areas of investigations, including the safety and toxicologies, as well as drug discovery efforts, as well as efficacy perspective. And, a range of different organ systems is really emphasized, but liver is one of the top sort of, demanding organ systems in the form of 2D in vitro models, organ-on-a-chip type of model, or 3D sort of in vitro model system. But not in the fair future, in spherical model will be really chiming in to really complement these areas of the NAM. So it's really quite exciting times to study, many exciting application areas with the, organoid technologies. So today I am going to talk about two platforms, focus on the safety as well as efficacy platform, mainly based on the IPS technologies, to study the human heterogeneities. In the remaining minutes, I'm going to talk a little about the future sort of, challenges as well as potential future enabler for improving the adoption of these NAM technologies. So really the first part of my talk is really focused on the toxicity side, which we already heard amazing talk from the Professor Don Ingber's. But we actually use the most simplest model to study this, drug-induced liver injury model, because we are interested in studying drug efflux process. So on the left-hand side, this is the sort of cartoons of like hepatocytes inside of the organoid structures. And as you can appreciate here, from the bottom, there is a very polarized, drug intake transporters, including OATPs or NTCP, expressed by outer layers of the organoid tissues. But the inner layers of the tissues, they are actually sort of polarized to express the drug efflux transporter. For example, PGP is one of the major sort of efflux transporters for eliminating the drug into the bioeconomic components. So even with just a simple hepatocytes, if you can control these geometries, we can sort of control the ordered drug intake as well as, elimination process in the defined conditions. So by leveraging this structural advantage, we are actually developing a very simple live biofluorescence dyes to study how, drug is actually perturbing the bioacid elimination efflux process to really identify as a proxy for the cholestatic damage, which is like most predominant form of drug-induced liver damage, or DILI. So using this simple imaging-based readouts, for example, on the panel A is showcasing, a couple of selected very, well-characterized compounds for particular sort of mechanisms of actions. And for example, in the middle, class B panels is actually composed by, very well-known cholestatic drugs preventing the biotransport processes. And with this exposure in physiological dosage, you can nicely appreciate the reduction of the fluorescent bioacid dye internalization process, which is inhibition of the biotransport process. So using this very simple imaging-based readouts, we actually tested more than 200 compounds, and then with the sensitivity, specificity, very compatible to the primary hepatocyte-based assay systems. So even with this, simplest form of the organoids, we can test some of the, toxicity parameters in a relatively higher throughput formats. So we next went on to study what would be sort of potential opportunities uniquely, equipped in the IPS technologies. So to test that, we actually wondered what would be the sort of genetic basis for relatively rare, DILI incidence in the real-world patient cohorts. So to tackle these problems, we actually, in partnership with the Drug-Induced Liver Injury Consortium from UK, as well as NIH-supported US-based consortium, together with the Takeda Pharmaceutical and Shinya Yamanaka, we actually gathered more than thousands of DILI patients' genomics data sets, as well as toxicity data sets. Then we went on to formulate the genetic score across the DILI patients and then identifying what would be the potential sort of, common genetic factors driving the DILI. So I should mention this, but, this DILI patient data set is actually composed by hundreds of different drug-induced toxicity patient data sets. So in this polygenic risk score published in 2020, we actually made a score that is overarching across a different drug causing a cholestatic DILI. So using this polygenic risk score hypothesis, we actually stratified 20 different iPS-derived models as well as primary hepatocyte-based model system, testing 12 different drugs to see if our genetic score can predict the damage response or DILI incidence across the score. So this on the right-hand side is really abstracted sort of data set in this 2020 papers, and it's showing you the genetic score increase variability parameters is reduced. And this trend is actually preserved across the different sort of compound sets, which is known to cause a cholestatic DILI. Although data is not included here, no cholestatic potential drug is not really, linearly sort of correlated with this genetic score, indicating this polygenic risk score can inform the potential sort of sensitivity to cholestatic DILI. So we actually tested this, genetic score in the independent clinical trial data sets and identifying the TAK-875, which is like type 2 diabetic drugs, and failed in phase 3 clinical trials. And we can, identify the patients at risk for developing, this TAK-875 compounds. So essentially, this, human data sets as well as organoid NAM data sets can inform the very complex genetic sort of architectures defining DILI. And results not shown here. We also performed the mechanistic studies in identifying what would be the downstream sort of trigger for this DILI and identifying the mitochondrial sort of abnormalities or dysregulation is really overarching factor for this. So this could be a very promising evidence that, our organoid NAMs or in vitro NAM can inform the patient sensitivity informations in a mechanistic way. So now we are actually putting our discoveries to our neighbors to replicate these results in a more robust as well as reproducible fashions. To do that, our own accelerator lab, led by Dr. Magdalena Cassandra, is now taking another layers of approach to really automate the process of our organoid production systems with a defined genetic data sets. And then she performed the very rigorous toxicity screening in a high throughput format and validating the benchmarking compounds as independent replication studies. So I pretty much believe that, independent replication is really a crucial factors transitioning to the commercial application process. So this is our DILI example. Next application is about more complex disorders called MAS or MASUD or MASH. So MASUD is very sort of broader high burden disorders across the world because 1 in 3 adults is actually affected by simple steatosis. But if you have the type 2 diabetes, obesity complications, you diagnose MASUD. And once MASUD is diagnosed, a very high percentage of the population is transitioning to inflammatory fibrosis state, eventually leading to the cancer formation process. So this journey takes typically 20 to 30 years to move on to the next stage. So we wanted to study this process in a more accelerated ways. Now, obviously, hepatocytes is one upstream factors, but not limited to that. Endothelial cells of liver-specific subsets or macrophage population down to stellate lineages. It is really important for driving the MASUD problems. And the problem is, progression is extremely heterogeneous. So the drug treatment is not always working in the same way as other people. So we definitely need a precision medicine approach. So to tackle this, we actually made an organoid model, intentionally having the multiple cellular components, including macrophage cell types as well as stellate lineages. And once we put the fully fatty acid and high sugar treatments at the end stage of the differentiation process, you can drive down to, accumulate a significant amount of lipids on the left-hand side, for example. And this response is dose dependent to the fully fatty acid exposure profiles. And if you persist the cetotic conditions, you can nicely drive the macrophage-driven inflammatory process, for example, detected by the IL-6, IL-8, and TNF-alpha. If you let the cells to inflame for more than 2 weeks, you can also appreciate the collagen accumulations, which is like replication of the fibrotic conditions. So this indicates the progressive fibrosis is actually going on in this culture model systems. So we next went on, to study again, patient variations. So to tackle this, we actually made efforts of screening the number of different patients, individuals, at the same time. So essentially, we are developing more than 20 patients mixed organoid virages in the same culture plates, and then perturb it with the fully fatty acid exposure and the sugar applications, and then testing their phenotypic association to the genetic associations. In summaries, we actually were able to identify the very well-known MASH-specific genetic factors, including PNPLA3s as well as GCKR, even at relatively smaller sample size. So this would indicate, organoids in culture-based model system can enrich the genetic contributions because of identical exposures as applied in these conditions. And in fact, these genetic factors is really well replicated in the clinical setting, indicating this steatohepatitis NAM can be, at least translatable for capturing the genetic factors. But I need to also appreciate the current challenges around the organoid technologies. For example, if you use the iPS technologies, organoids in general is early postnatal states. And partly because of the limited complexity, because we know that in the normal situations, the tissues are talking to many other organ systems. For example, in the liver case, gastrointestinal stimulation is really crucial for driving nutritional response. And because of this, there are, some limitations around, for example, like blood-borne factor contributions or systemic exposures, like nutrients, or even just a perfusion-driven mechanical contribution is missing in the existing in vitro model systems. But excitingly, as already have been summarized by the previous talk, there are a number of ways of addressing these limitations. So I think this would be the very exciting momentum we can create with the multidisciplinary collaboration process. For example, as Dr. Sergio Pasquer, we actually made a pre-existing tissue composite to make more complexity introduced in the organoid model system as a form of assembly tissue architectures. And Blaine is prominent examples. In the organ-on-a-chip approach, which has been beautifully summarized today's keynote, perfusion is really driving down the number of, key crosstalk, which was very difficult to capture in the organoid itself. But I like to, appreciate the fact that, transplantation approach is really also still the powerful way of getting the tissues to mature. For example, in the bottom panel, this is our example of the human vascularized organoid model systems. After transplantations, we can, permeate, the functional blood vessels in the tissues of the humans in the animals. And the beauty of the system is, for example, we can study the immune interactions or thrombotic interactions, which is very hard to study or stably study, at least in culture model systems. But not limited to that, maturational drive is really, exciting avenues. For example, cytochrome activity is pretty much activated when you meet with the blood exposures. And, live image readout would be also another sort of, emerging frontiers. For example, in the middle panel here, this is the visualize of neutrophil behaviors. Neutrophil is very challenging to isolate ex vivo, but in vivo situations, you can, study how this single neutrophil is actually behaving to target the cells to damage, for example, in the T cells. So this type of in vivo hybrid type of system could really bringing the organoid limitations to the next stage. So I will end two slides. There are a couple of areas we think would be extremely helpful to going forward. For example, I showcase some of the population screening data sets, but obviously, single cell type of technologies is really expensive. And so we wanted to have more multiplexing approach or demultiplexing approach to really make it scalable analysis across the populations. And close organ builder platform, organ to organ, media variations, readout variations, always tricky to study more complex biologies. And third, we now know that, biobank as well as data biobank is really exciting to study human stem cell model over to the clinical phenotype. But, this, data management strategy is always the challenging part. So we need a really streamlined, data biobanking process. And obviously for the computational model is really next frontier, ADMET, PBB, PK, QSP, QST model is really, started being explored, and hopefully we can extend these capacities with many pharmacology folks to study the human pharmacology reactions. So I would end with this slides. We show that, genetics can be studied in organoid model system, and the scalability can be tested outside of the laboratories, and really lubrication is crucial. And our MASH model were, able to study the population scale dynamics and then, probing the personalized differences. But obviously, maturing and connecting to, neighbors and the systemic crosstalk is another frontier. So we need either, in vivo models or in vitro complex MPS type of systems. So with that, I would like to conclude my talk and happy to discuss in a later panel.
Great, thank you so much. I really appreciate the slide where you talked about what's needed next. So I think we'll be talking about that more in the panel. Appreciate you setting that up for us. All right, our next speaker is Dr. Sergiu Pasca at Stanford University. This lab pioneered assembloids, developed regionalized neural organoids, and created integrated human neural circuits. And will be telling us more about their work. So thank you so much. Do we have her? Him? I'm sorry.
She's on. She just is muted.
Okay.
I saw her there for a second, but there she is. We just need to. There you go.
You called Sergio Pasca, so I wasn't sure if I was up next.
Yeah, I think that's who's next.
I'm Diana Mansavais.
Yeah,
Diana
you're fourth on the list. Unless something's changed.
Do you mind to introduce Diana briefly?
Alrighty. Sorry about that. So I'm sorry, you messed up my my order of operations here. I apologize, everyone. So next is Diana Mansavais from Bayer College. She's a faculty member at Santiago Ramon y Cajal, empowered professor at the development of pathology and immunology at Bayer. Her lab focuses on uterine biology, endometrial regeneration, pregnancy loss, and endometriosis using genetic mouse models, human tissues, and 3D organoids. So take it away, Diana.
Okay, good morning, good afternoon, everyone. Thank you to the organizers for the opportunity to present our work regarding the use of mouse models and 3D organoids for the study of endometrial regeneration and infertility. So reproductive function in mammals is controlled by a neuroendocrine system that involves
Excuse
the
me.
brain.
I'm so sorry. Do you need me to share your slides or do you want to share them?
I thought I was sharing them. I can try again.
Okay. Yeah. We don't see any yet. There they come in. You just need to put them in presentation mode. Perfect. Thank you.
Okay, so here we go. So reproductive function in mammals is controlled by a neuroendocrine system that involves the brain's hypothalamus, the anterior pituitary gland, and the gonads. This system is termed the hypothalamic pituitary gonadal axis and functions by the secretion of GnRH from the gonadotropes in the hypothalamus, which then signals to the pituitary and promotes the secretion of follicle-stimulating hormone, which signals to the ovaries or testes to induce follicle or sperm maturation. So genetically engineered mice have been essential to uncovering the systemic and molecular mechanisms that underpin the HPG axis. For example, knockout of the receptor for GnRH results in underdeveloped male and female reproductive tracts, while knockout of the receptor for inhibin ACVR2A, which is a negative regulator of FSH production secreted by the gonads, results in underdeveloped testes and ovaries that are lacking mature corpora lutea. While mice that are lacking the progesterone receptor demonstrate that this gonadal hormone is essential for uterine development and ovarian maturation. So these examples, as well as many other knockout mouse models, have revealed that fertility is controlled by an inter-organ communication system that requires a coordinated and highly regulated mechanisms. So due to their short gestation period and their hormonal profiles, mice have also been essential models for uncovering the earliest phases of pregnancy establishment. So fertilization and implantation of a blastocyst can be carefully traced from the moment of copulation, which is defined as 0.5 days postcoitum, and until the moment of implantation, which occurs at about 4.5 DPC. At 3.5 DPC, the blastocyst enters the uterine cavity and begins to make loose contact with the inner lining of the uterus, which is termed the endometrium. Then the process of attachment begins through a process that involves ligand receptor mediated interactions and is followed by embryo implantation, where the blastocyst actually breaches the luminal epithelium and invades into the underlying stroma. As this occurs, this process of implantation triggers a process of endometrial stromal differentiation known as decidualization, where these endometrial stromal cells become specialized to support early embryonic development prior to the development of a placenta. So this process of early pregnancy is coordinated by the secretion of the ovarian steroid hormones, estrogen and progesterone, which are similarly controlled in both mouse and humans. So recent advances using in vitro culture systems have begun to recapitulate part of this process of embryo implantation in humans. For example, the generation of endometrial assembloids using human endometrial epithelium and stromal cells can be established from endometrial biopsies or menstrual effluent, and then combined to form 3D structures that histologically resemble the human endometrium, as can be seen here. So when these endometrial assembloids are overlaid with a 5-day blastocyst, the endometrial assembloids can be shown to receive and allow for blastocyst expansion for an additional 3 days in culture, showing that these faithfully recapitulate the processes of early pregnancy in humans. Of course, there's technical and ethical difficulties with doing these experiments with human blastocysts. So to overcome these, several labs have started using blastoid and gastroloid models that can be developed from iPSCs and allow the study of these pre and post-implantation events. More recently, some of these blastoid models were tested on an engineered 3D endometrial model that's comprised of endometrial stroma and epithelium. So this endometrioid model was plated on a defined hydrogel of similar stiffness to the endometrium and was shown to histologically mimic the structure of the human endometrium with a spatial organization of endometrial epithelium, glandular and luminal epithelium, and stroma in the underlying layer. Functionally, the model recapitulated recurrent implantation failure when tested in biopsies from women that had experienced more than 3 sequential pregnancy failures. This is termed as recurrent implantation failure. When endometrioid or assembloid models were established from control women with previous normal healthy pregnancies and women with RIF, and then overlaid with blastoids, they could recapitulate that the endometrial assembloid models from RIF patients failed to implant and also failed to outgrow and expand as those observed from the control patients. So to summarize this part, in vivo models have been and continue to be essential to model the neuroendocrine control of reproductive function. These have also paved the way for the development of human 3D in vitro models that allow the study of these special processes for normal and diseased conditions. So animal and in vitro endometrial 3D models have also been applied to study endometrial regeneration. In humans, the inner lining of the uterus, the endometrium, has the capacity to undergo up to 400 cycles of regeneration throughout the reproductive lifespan of a woman. This process of regeneration is controlled mainly by the cyclical action of the steroid hormones estrogen and progesterone, which are secreted from the ovaries, where estrogen drives the mitogenic potential and expansion of both the endometrial stroma and epithelium in the endometrium. And progesterone, which is secreted by the ovary following ovulation, drives the differentiation of the endometrium in preparation for a pregnancy. In the absence of an implanting embryo, the drop in progesterone leads to a pro-inflammatory reaction that's characterized by an influx of macrophages and other immune cells that triggers matrix metalloproteinases and leads to the breakdown and release of the endometrial tissue through menstruation. So this process, as I mentioned, has the capacity to undergo 300 to 400 times in a woman throughout her lifespan. And so it's hypothesized that this rapid rise in regeneration is triggered by the presence of stem cells that are residing in the basalis endometrium, which is a portion of the endometrium that remains following menstruation. And that these stem cells will expand and rapidly heal or regenerate the endometrium within a 24 to 72 hour period. However, the actual identity, location, and the factors that are leading to stem cell expansion have not been very well characterized in the endometrium and are currently an active area of investigation. So although mice do not menstruate, they do have an estrous cycle that's also controlled by the rise and fall of the steroid hormones estrogen and progesterone. And this endometrial tissue will also expand and resorb through the mouse estrous cycle, which is 4 to 5 days long. We can also see a rapid period of regeneration in the postpartum period in the mouse, where after delivery of the embryo, the entire endometrial architecture will quickly regenerate within a 24 to 72 hour period. Lineage tracing studies, as well as classical stem cell assays, have shown that several stem cell markers, such as LGR5, AXIN2, and PDGFR alpha and beta, are essential progenitors that lead to the rapid regeneration of the endometrium, both throughout the estrous cycle and in the postpartum phase. And so using genetically engineered mouse models, we have shown that receptors as well as transcription factors of the TGF-beta signaling pathway are essential for endometrial homeostasis, and that deletion of the receptors such as ALK5 or TGF-beta R2, or the downstream transcription factors that are activated by TGF-beta signaling such as SMAD2/3, results in uterine tumors, as can be seen here by these uterine masses that metastasize to the lungs. So if we generate organoids from these mouse models, we can actually recapitulate the morphological defects in vitro, where organoids from the mutant mice develop these dense and stratified epithelial architectures with increased organoid numbers and increased organoid areas. We could also see that the organoids from these mutant mice develop abnormal expression of squamous epithelial markers such as P63 and cytokeratin 5, which are typically expressed in stratified epithelium such as the cervix and vagina, but not typically expressed in the single columnar epithelium of the uterus, as can be shown here in the controls. So if we profile these organoids using transcriptomics, we can observe that the organoids that are derived from SMAD2/3 conditional knockout mice have altered expression of retinoid signaling, of genes that are involved in retinoid biosynthesis. In particular, we can see that there's excessive expression of the aldehyde dehydrogenase enzymes. So these aldehyde dehydrogenase enzymes are known to convert the activation of the inactive retinol to the active retinoic acid, which is then able to bind to the RAR and RXR, and in many other tissues has been shown to be essential for stem cell maintenance, regeneration, and differentiation. When we do immunohistochemistry of ALDH1A1 in the mouse endometrium, we can see that ALDH1A1 is highly enriched in the crypts of these endometrial glands, which is hypothesized to be a reservoir of stem cells in the mouse endometrium. So to further define whether aldehyde dehydrogenase 1A1 is itself a stem cell marker in the endometrium, we've used the ALDIFLOR assay to sort cells with high and low ALDH activity from the mouse uterus, and can demonstrate that that organoids that are formed from the aldehyde dehydrogenase 1A1 have a higher organoid formation capacity than those cells that are sorted from ALDH low, than cells with ALDH low activity. These ALDH high cells have a higher organoid formation rate, as well as a higher organoid area compared to the ALDH low. So if we perform the same experiment in human endometrium, we can observe that the ALDH high organoids also have a higher organoid formation rate, and can be they have a higher organoid formation rate than the ALDH low organoids, and they can be maintained in culture for a longer period of time than the ALDH low organoids. If we compare then the ALDH high organoids that are derived from eutopic endometrium, so the inner lining of the uterus, and from ectopic organoids, which are derived from lesions of endometriosis, we can see that ectopic lesions of endometriosis have higher percentage of ALDH high cells than those that are derived from the eutopic endometrium. These ALDH high organoids from both the eutopic and ectopic endometrium also have very unique transcriptomes, demonstrating genes that are enriched in categories such as inflammation, neuroimmune signaling, increased ciliated expression, and stemness, indicating that these two aldehyde dehydrogenase cell populations are uniquely different, whether they're located or originating from the eutopic endometrium or from the ectopic endometrium of matched patients. So over the past few years, my lab has been dedicated to expanding patient-derived organoid models from patients that are undergoing treatment for various gynecological conditions in the Texas Medical Center here in Houston. We routinely receive biopsies from patients that are undergoing excisions of endometriosis, of endometriotic lesions, patients that are undergoing treatment for endometrial cancer, or we also receive menstrual effluent from patients across various gynecological clinics. So far, we can take these biopsies and we attempt to establish organoids from each one. And so far, we've established over 120 unique epithelial organoid lines that can then be used for various developmental or drug targeting, drug screening approaches. Here, for example, we can see that we can directly compare utopic and endometriotic organoids derived from the same patient. We can expand them and treat them with various unique kinase inhibitors that have been developed here at Baylor at the Center for Drug Discovery. And then we can profile each patient's unique transcriptomic response to these kinase inhibitors, or to inflammatory stimuli, as we can show here in organoids treated with interleukin beta. Or we could also perform a time course drug treatment and then perform a live dead assay to look at the unique patient's organoid response to drugs, and then qualify viability as well as dead cells in these various patient-derived organoids. So to conclude, I hope that I've shown today that mouse models are essential for inter-organ communication systems that control reproductive function and dysfunction. Endometrial organoid and assembloid models demonstrate that they are very useful tools to discover mechanistic drivers of disease and model early pregnancy in humans. Mouse models have helped identify cell populations as well as growth factors that drive endometrial regeneration in humans. And we've performed studies sorting ALDH high cells and have shown that they form more as well as larger organoids than ALDH low cells. And we can show that ectopic organoids from patients with endometriosis have higher ALDH high organoids and display different gene expression patterns. So I'd like to thank the members of my lab who have contributed to all of these studies, and in particular to the formation of the organoid biobank, our clinical collaborators, from the Center for Drug Discovery, and our funding sources from the NIH and the Burroughs Wellcome Fund. Thank you.
Great, thank you. Again, we're saving questions for the panel, so we'll move along. Next is Manuel Amavea, who is a physician scientist and professor of pediatrics, infectious disease, and microbiology and immunology at Stanford, whose research examines how bacterial pathogens colonize epithelial surfaces and cause disease using animal models, human tissue, and advanced imaging techniques. Welcome, Manuel. Thank you.
Thank you very much. That was an amazing talk by Diana. I'm going to continue with the idea that we need to combine animal models, human tissues, and human-derived microtissues models to find new things. In this case, I'm adding the complexity of microbes and how they actually interact with our cells in a much more intimate way than we think. Just from I'm an infectious disease doctor, but I spend all my time trying to kill microbes. But in the end, you will like this one. So this is a picture of a gland in a stomach in a human. And the green blobs that you see there, I'm going to convince you are bacteria that are living deep in the tissue and that interact even with your stem cells and may be the source of gastric cancer. So the microbe that I'm talking about today is Helicobacter pylori. It's called helical because of its helical shape. It has flagella and it can swim. And it lives in a place where really nobody wants to live in, which is the human stomach, because it's a vat of acid. However, even though it's the only place it can live, it still colonizes about 50% of the world. And it is a commensal in most people, but in about 20% of people, it causes ulcers. And that's how it was discovered. And the discoverers received the Nobel Prize for this because now we can treat ulcers with antibiotics. However, about 1% of people that are infected will develop gastric cancer. And 1% of half of the world is a big number. So it's the fifth most common cancer in the world still. And as a pediatrician, I'm also very intrigued because H. pylori is an infection of children, and it's a microbe that persists for the lifetime of the individual and causes disease later in life. So for all these reasons, it's a very fascinating bug. And when you look at biopsies, you see that people describe it living in the mucus because it cannot survive the acid of the lumen of the stomach. So it quickly finds its way to the protective mucus of the stomach where they are swimming, and also can attach to epithelial cells, where we're going to refine this model through these studies that I'm going to show you. The bacteria can actually inject virulence factors into the host, and also they sit very specifically on the epithelial junctions, as you can see in this confocal micrograph. Now, I wanted to show you that all models are useful in some way. So most people that have studied interactions with epithelial cells use this adenocarcinoma cell line, which is really not a good epithelium. It forms little clusters of cells that expresses some junctional proteins, but really it doesn't form polarized monolayers and doesn't behave like an epithelium, it behaves like a cancer. When we infected it with H. pylori and we're looking at its effect on junctions, we actually found that H. pylori could reorganize junctional proteins like ZO1 to come to be underneath the bacteria. And here you can see after 4 hours that the bacteria were changing shape. So a long time ago, we made a model of polarized epithelium using a dog kidney cell line, and we're able to show that H. pylori also bound to over the junctions and actually modified the junctions in these cells. And we were able to show that this was due to its major virulence factor that's associated with cancer. But trying to understand how this affected the epithelium and the bacteria, we started making movies of the bacteria sitting on top of the epithelium and on top of the junction. So just watch these two bacteria over time. And what you see is that they don't go and destroy the epithelial junctions or break open the epithelium, but you see that the epithelial cells continue to divide and the bacteria stay in the same junctions where they attached. And in the end, what we found is that they form colonies on the epithelium that we term microcolonies. They're clonal colonies right on top of the junctions. And over many experiments, we found that they are acquiring nutrients and modifying the epithelial polarity to be able to colonize with minimal nutrients in these monolayers. So H. pylori, basically, it's turning our cell surfaces into a place where they can live. But all of these experiments came from cell lines. Now we have the capacity using organoid technology to see if this happens also in human epithelium. For infectious diseases of this type, one of the main problems is that organoids are typically grown inside matrigel droplets, and so the lumen is inaccessible. If we want to stick the bacteria in there, we would have to microinject them or break them open, as you've seen. But we came up with a different method. We actually decided that if we remove the matrigel, perhaps we could alter the polarity, because we know that a lot of the signals to preserving polarity come from the basolateral part of the cells interacting with the matrix. So here's a movie of an organoid in matrigel. And what was really surprising is that when we removed them from the matrigel as intact organoids, the organoids actually flip themselves inside out as if they were a sock that you just took out of the dryer. So that is very useful, because now you have an organoid. This is the same organoid that you were just watching flip around. And we can stain it in the microscope and show that now the microvilli are outside and the junctions are also outside. So with this model, now many labs are using this, but I wanted to show you something that we didn't know we could do with these systems. So this is part of the beauty of using these new technologies, that we find that, for example, they can help us see cells from the side because they're spherical. We can set them in the microscope so we can see the monolayer as if we were looking at it sideways and get pretty good resolution to see the bacteria coming to the epithelium. And then we can again fix them and see that indeed those bacteria are coming in and in a few seconds attaching to the junctions. We could also then use other tricks. For example, we could mix green and red bacteria, infect the organoids, and you can watch them here attach in a matter of seconds, wash away the bacteria and look at them right after attachment. So we can see that they're randomly attaching to the epithelial junctions. And then if we let them grow for a period of a day, we see that they form clonal microcolonies that remain, different colors. And so we are making them work for a living because we don't give them a lot of nutrients in the outside media. So they have to really interact with the organoids. And here you see that if you look underneath these bacteria at Z01, that they're already modifying the junctions. So all of these findings that we had in now epithelial monolayers, we wanted to see if they were happening in vivo. Are they forming these microcolonies? And this one is started to get more interesting because when we made a mouse model, which is difficult because it's a human-specific infection, but we have a mouse model with a specific strain of H. pylori, and my student Josephine Lee used three-dimensional confocal microscopy to see where these bacteria were colonizing. And indeed, we expected them to be at the surface of the stomach. So this is a reconstruction of the surface of the stomach. And you can see that she found the bacteria sitting there attached in groups like these microcolonies. And when she looked very closely, they were on the junctions. However, again, the beauty of science is that surprises, because she then took thick sections of the stomach to look at the whole tissue. And here you can see the bacteria sitting on or swimming in the mucus in the surface, whereas you see the top of the tissue. But if you look deep into the glands, into each of those channels, there are groups of bacteria that are living pretty deep into the tissue. This space now tells us that there's something special, not just about the stomach, but about specific cells in each glandular unit. And to identify what that was, we just looked and we found out that this is the region where the progenitor cells live. So these cells are undergoing cell division, and you see here cells stained white for mitotic proteins that are phosphorylated during mitosis. So you can see that this means that we have now bacteria sitting right on top of our dividing cells, which is probably more dangerous than them interacting with the terminally differentiated cells at the top of the stomach on the surface. So to study this process further, we obviously wanted to know whether this happens in human, because it hadn't been described. And we went and did 3D reconstructions of gastric resections and found that here they are at the surface of the stomach, but also deep in the glands in regions where there's mitotic cells that we could also stain. So what we saw in the mouse and in the cell lines and in the organoids, we could also find in the human stomach. But actually looking at the human stomach gave us another idea, because at the bottom of the glands, there are also signals that look like bacteria, the way where the stem cells reside, not just the dividing precursor cells. So we went back to the mouse model that now we can use fluorescently labeled stem cells, mice with the fluorescent stem cells, and see whether the bacteria were there. And indeed, they are sitting right at the bottom of the gland as well in little colonies. So we started studying whether the bacteria were inflaming the stem cells. And I'll just show you one experiment, which is that we infected mice for a month, and the mice then had twice the number of stem cells, which is in itself a potential risk for developing cancer if you duplicate your stem cell population. We also found that these stem cells were expressing higher amounts of stem cell associated genes that are actually came from the intestine, because we didn't know really what the normal gastric stem cell expresses. But I'll show you that this was an important finding in the end. Finally, we can also use bacterial genetics, because we were able to find a mouse, a bacterial mutant that can colonize mice fairly well, similar to wild type, but it lives on the surface only and doesn't get into the glands. And this mutant, when we looked at pathology, these are experiments take a while, but after 6 months, you can see that the epithelium gets very hyperplastic, tall, with a lot of inflammation when you infect with the wild type. With the mutant, they did not. And also there was no stem cell activation. And so from these experiments, we conclude that perhaps where you live in the stomach is just as important as if you're there or not. So going back to the organoids, the organoids gave us ideas as to whether we could then find out whether the surface epithelial cells respond differently from the ones that are deep in the glands. And what we did is we took the organoids that had just been in matrigel, and at that stage they are mostly precursor-like organoids with a lot of dividing cells resembling the bottom of the glands. But if we differentiate them for 5 days, and if we do this in a staggered way so that our organoid cultures can be harvested at the same time and then infected at the same time, we could then compare these two types of cells from the same person. And you can see here that by single cell transcriptomins, the precursor type has a lot of immature cells, whereas the surface type looks like differentiated surface cells. My student Yulim Kim infected these two types of organoids from paired patients and found that the precursor cells responded very dramatically with a lot of inflammatory cytokines that had actually been described with the injection of this Kage protein that is associated with cancer. But the surface type organoids did not respond in the same way. In fact, we were surprised that there was mostly negative or downregulation of genes. So the last thing I want to show you is that these organoids can also be used for studying different stages of disease, as we have been discussing. And you've already been primed with the idea that we obviously can't do a 40-year experiment to see if we're going to make cancer, but we can get tissues from different stages of disease. And the question that we had is whether we could preserve the stem cells and the tissues, the disease state from different biopsies. And we were able to create we're creating a biobank of biopsies from different regions of the stomach, from different patients, and also from different stages towards cancer. And I'm going to tell you just one experiment using the stage of intestinal metaplasia, which is actually this last stage at which we can treat patients with antibiotics to reduce their risk of cancer. So here's what that looks like stained in the confocal microscope. You see a normal mucosa on the left, and then the mucosa that looks more white and milky is a gland that is a gastrointestinal metaplasia. So you basically have little intestinal glands living in your stomach. And we know that these glands are intestine because they have goblet cells that secrete goblet-specific factors, and at their base they have stem cells that are marked with intestinal stem cells. It turns out all of them, marker I showed you before, is not part of the gastric stem cell. And Ulim started looking for these intestinal stem cells in a model that allows us to culture the cells for a very long time instead of just the organelles that when we take them out and flip them only live for a few days. So this is an ALI system of monolayers in a transwell that are called mucosoids because they produce a lot of mucus that you have to remove every day. So now we have lots of milliliters of human stomach mucus in the freezer. The mucus looks like this from the monolayers. It's being secreted by individual cells. And however, if you look for markers of replication, the cells are still dividing. These particular pictures were taken from a mucosoid that was 2.5 months old. Now we have some that are more than 9 months old in the incubator. So they really are long-lived and self-renewing steady state cultures. We can also do single-cell transcriptomics on the mucosoids, and they have gastric precursors and all the different cell types that we find in the glands, including rare cells, including tough cells and enteroendocrine cells. But importantly, the ones that are coming from metaplasia also have intestinal-like stem cells and differentiation into some of the intestinal cell types. So if we just look at the marker for intestinal-like stem cells in normal mucosoids, there's very few that you can measure or see using transcriptomics and confocal microscopy. But in a mucosoid from gastrointestinal metaplasia, you see that there's very dramatic increase in these intestinal-like stem cells. And indeed, what I found really interesting is that in the adjacent tissue from the same patient, there are already these abnormal stem cells, even though this came from a part of the tissue that does not have intestinal metaplasia. And several people have alluded to the fact that there's epigenetic preservation of gene expression in these disease states. So we can preserve these features for a long time in culture. This is just looking at the olefin 4 gene by ATAC-seq. So let me show you one experiment and then finish, which is that we can make these GIM mucosoids differentiate. And this is a type of lineage tracing experiment because we can see that the cells that are expressing olefin 4 become the cells that express now a intestinal marker. And so those are truly precursor cells. And we can infect these mucosoids with H. pylori, and they will grow as microcolonies as before. They also can grow on top of the intestinal-like stem cells. And in this experiment, we infected GIM adjacent tissue for a period of 2 weeks. You see the very large microcolonies on the right. And if we look underneath those microcolonies, you see that there's more olefin 4 in the infected than the uninfected samples, which we also measured transcriptomically. So this suggested that perhaps even in what still looks normal, there are already olefin 4 positive intestinal-like stem cells, and we looked to see if we could find H. pylori down there in tissues. So we came back to the tissue. And if you look very deep into this gland here and zoom in, you can see that indeed there are these isolated olefin 4 positive cells with the bacteria growing on them. And here's another example. So just to finish, I just wanted to show you that these organotypic systems can help us find things that are quite difficult to see and predict, not just test just yes or no questions. So thank you, and I'll take questions later.
Great, thank you so much. So we move on now to the panel discussion. So we're going to open this up to all of the speakers of the first session. And I'm going to kick us off for the first question. So how can NAMs be used to complement research on diseases where the most important outcome are complex organism level phenotypes um, that can't be captured with isolated cells or tissues? Neurological readouts, psychiatric diseases where the readouts have to be behavior or cognitive or something along those lines. Are we getting there yet, or is this something that's doable? Thoughts?
I can give it a try, if that's okay, as I guess is like neural. So to be honest, I think no. I think like not at this point. Especially for circuit and behavioral readouts. I mean, as you've seen, we can build like relatively complex circuitry, but the circuitry in vivo is always much more complex. So for instance, think about like modeling epilepsy. Epilepsy is defined by seizures, is defined by an EEG. And just because, for instance, a neuron that is derived in a dish will be hyperexcitable, does not mean that that will result in seizures in an individual, because they're homeostatic events, of course, at the circuit level, because there are many other cell types. So we think that even for that, we need behavioral readouts. And of course, sometimes animal models are useful for that. Another useful preparation in this case, it's again through this transplantation methods, where you can actually, for instance, induce seizures in an in vivo context from patient cells. That's certainly one way of doing it, but we're certainly going to need much more complex behavioral readouts for these complex disorders. There's no doubt.
Thank you. Does anybody else want to take a stab, or should we move to another question? All right.
I think related to that, sort of maybe a more general question is, how do you determine inter-organ effects with the single cell type or organoids? I know there's been a few. The assembloids are somewhat like having organ communication, but there's lots of diseases that may start, like let's say in the gut, then move up into the brain. So how do we sort of tackle inter-organ effects?
Yeah, I think we're just learning to connect these different organ types. You heard Don Ingber talk about this human on the chip approach. Of course, very early, but I think we do have some interesting findings which demonstrate that differently engineered organ systems can communicate with each other. Sergio talked about neurons. There's a lot of paracrine activity between tissue types. So this is a promising field. It will never be the same as an in vivo model. But the beauty, of course, is that you can simplify these systems. You can very precisely define them. You can put cells in or take them out, and that allows a lot of precise investigations, I would say, which are not possible in vivo.
Yeah, thank you. We have a question in the chat for Dr. Pasca. Do the assembloids in your talk contain oligodendrocytes or other glia, and are the axonal projections myelinated?
Yeah, that's a very good question. So it depends a little bit on the brain region. If it is for the cortex, sadly, and that's probably one of the limitations that is worth mentioning, that because timing is actually quite well conserved, most of these experiments are incredibly long. Myelination in the human cortex is primarily a postnatal event. Oligodendrocytes are formed up to 2 to 3 years of age. And that will actually be recapitulated in vitro as well. It's very hard to actually grow some of these cultures. I mean, it's possible. It's not very practical to grow some of this culture for such a long period of time. For other brain regions, for instance, for the spinal cord, it's much easier to get oligodendrocytes. They appear much earlier. They're actually myelinated. We can see examples of myelination, but certainly will not be to the same extent in vivo. One way is certainly to use, again, a transplantation model where you can actually see myelination of some of the neurons in vivo over long periods of time. So I think that's one way. There are various preparations where you can actually make oligodendrocytes a little bit like faster. But again, the issue of myelination is always very difficult in vitro. I think all of those in the glia field will agree.
Yeah, I had a follow-up question to that. Sort of again, broadly speaking, how do we model aging? So most diseases of, neurodegenerative diseases, for example, there's the aging component. So how do we model that in a dish where most of the cells we're using are sort of have these developmental profiles?
Yeah, that's also a great question. It's certainly one that has been discussed in the field quite extensively. I think there's no doubt that the timing is conserved, and therefore, if you keep an organoid for 2 years, it would likely be at early stages postnatal, but not necessarily late third or fourth decade. There are ways of circumventing this at some extent. So, for instance, there are disorders that have early onset neurodegeneration. There are neurodevelopmental disorders that have a neurodegenerative components. Those are certainly much easier to, for instance, vanishing white matter disease, which appears like very early. And then there have been strategies for trying to circumvent this by direct reprogramming, which are still sort of like ongoing. And then there have been discussions that many of these neurodegenerative disorders actually have an early neurodevelopmental phenotype. The classic example has been Huntington disease, where we know that there are very early defects, and Parkinson's disease as well. Of course, the question remains, are those defects, relevant to the pathophysiology later? So if you focus on restoring, on understanding one of these early phenotypes, will those have predictive value later? But I think that remains an open question, and I think there should be a sustained effort for trying to skip time, advance them in maturation for studying these neurodegenerative disorders.
Okay, cool.
If I can expand on
Yeah,
that,
please.
yeah. So, for example, in a general maturation status in the hepatocyte field, at least, this is somewhat similar to Sergio's comments. Are, for example, looking at bulk RNA sequencing, looking at tissue maturation signatures. I think that is actually projected to early postnatal windows at the best. But what's interesting to us is actually we recently looked at the mesenchymal signature or stromal signatures, and in comparing those with the progressive, inflammatory conditions, and then comparing those without inflammatory status. Then we actually, getting the patient's RNA sequencing from the fibrosing, group versus, modest fibrosis groups or different age group. Our, in vitro fibrosis signatures in the mesenchymal population is actually projected over relatively sort of adult stage equivalent fibrosis signatures we can actually, observe. So I think depending on maybe cell components and looking some, pathogenic signatures, we somewhat study even adult onset type of disorders. It could be artifact, but I think, when we compare those with the existing, therapeutic options, we can actually reverse this, aging-related mesenchymal activation signatures. So I think depending on the question and in cell types of interest, I think there might be some opportunity to study even with the relatively sort of later onset disorders to tackle on even in an ideal cell state. So it's kind of like, new territory we actually started exploring, but I think it's quite fascinating for if you limit the particular cell types of interest, maybe we can do something even with existing immature looking states.
Okay, thank you. So another question we had for the group is, this is a therapy development session. We've heard a lot about using NAMs for understanding these biology and coming up with targets and even early screening or early efficacy or even, toxicology. I haven't heard any discussion of NAMs, at least today, about, are there things that we can't do with them, like, pharmacokinetics? I mean, is that ever going to be doable? Later stage, therapy development where you have the IND enabling work, are NAMs going to be there in the near future, or are we still going to need animals for that?
I think we still need animals for that.
Yeah.
But, I mean, many things can, of course, be simulated in the dish. I'm not a specialist in hepatocytes. I think Takiba needs to say something about that. But My understanding is that we've tried some of that. We have tried a little bit, but in our hands, these models never express the profiles of, for example, cytochrome systems that you would expect from the liver. But that doesn't mean that they are not there. They may just not be there with the right profile, with the right activity. And we may just have to accept that these models are a bit different. I mean, you mentioned the aging part. I think we also aging in a dish is different from aging in vivo. Yeah? We can see beautiful senescent-associated peptide profiles in iPSC models, but that doesn't mean that these tissues are aged. So I think we just have to may redefine or the term aging in the dish, and see what is similar to in vivo and whatnot.
Yeah, one thing I can add in terms of liver-related stuff is there are a couple of ongoing initiatives, like for example, ARPA-H, Catalyst program is essentially trying to create complement existing PK/PD models with the more predictive in vitro model system, where we actually contributed one of the project as well. But there is potential windows, for example, like pediatric populations. It's very difficult to predict the pharmacokinetic profile, just by allometric scaling, which is like very preparing methods of predicting PK. But because, PK-related transporters are not linearly, correlated with the adult, counterparts. So many, translational pharmacology scientists has a struggle in terms of like how much dosage you need to provide for, immunosuppressants and, many other drugs. So for these, relatively under-investigated population in the IND enabling part of the studies, maybe I think our, maybe immature looking organoid is actually more, suited for studying the clearance parameter, for example. So we just started collaborating with my colleagues at the Children's Hospital with the, extensively PK profiled patients populations with a differential response to particular same drugs, like, cyclosporine and tacrolimus and sirolimus. And then looking at, organoid counterparts, looking at how much drug is eliminated over time. And then maybe we can, do in vitro to in vivo extrapolation or IVE type of, mass-based analysis to predict the clearance parameters. It cannot really explain everything in terms of PK parameters, but I think at least some aspects of the vulnerable population, maybe we can complement with the in vitro type of model system. So that's kind of like our, current perspective on that.
Okay, cool.
And so we have a couple of questions in chat, but before we get to them, sort of just following up on that discussion, the converse is how do we, if we use animal models, how can we improve them for modeling diseases? Because that's, I think some of the impetus for these NAMs is that there are things that we can't model in animals, but can they be improved?
No takers on that one, huh?
I'll take it. I think we're constantly trying to improve. So I think just to give you an example, that effect of when we keep cells in a dish. I mean, that was like shocking to us a number of years ago, because we're keeping the neurons. They look pyramidal. We were patching them. They were like clearly larger than mouse neurons. I mean, we knew they were immature, but when we actually compared them to actually human neurons that came out of a biopsy of a patient, we were shocked that they were like so small, and that they should have been much more advanced. And so we spent the last few years actually trying to figure out what that could be. Because not everybody can do transplantation, and perhaps that aspect would be very useful. And we've done that through various ways. Like, for instance, we thought it was activity in the brain, and that we would never be able to mimic that, in a dish, because there's no way you can mimic all of that activity and those patterns, because nobody knows them even. And so we thought to exclude, to make sure that it's just activity and nothing else, we did transplantation in the kidney of a cortical organoid. And when you do a transplantation in the kidney, to our surprise, that the neurons were equally large, if not even larger in the kidney than they were in the brain. So then we thought it must be something secreted. And a very long story, in the end, we discovered that there was a very strong signature of hormones that was upregulated in the cells that are transplanted versus the one in vitro. So then we've done a screen of hormones that are not present in vitro, we thought. And the one that is the most striking is actually thyroid hormones. And so if you now come with a thyroid hormone and you add it, you can just see within a few weeks, the neurons are just like growing like 3 to 4 times larger. And we thought it can't be because there must be thyroid hormone. So we went on, B27 has thyroid hormone, theoretically in large quantities. But when you add it onto the human cells, it's de facto 0 because it does not activate any of the programs. So it probably is either taken by albumin or like there's not enough free. So you have to actually supplement it. So that's a very classic example. And they're essentially hyperthyroid neurons, most of the ones that we have in a dish. The way it was for cretinism, for like intellectual disability, where neurons are actually very tiny unless they have thyroid hormones in vitro. So I think there are a lot, we should not underestimate just how primitive most of the media that we're using in vitro are, and that we actually have to constantly improve. This is one example, but just imagine how many more there are that we actually have to do afterwards. So I think there has to be a systematic effort of trying to improve, of course, not of them, because nobody wants to make an in total preparation that would be useful, but for very specific preparation, having systems that are able to answer that question.
Yeah, that's so fascinating to us. Thyroid hormone is also our critical driver who drive postnatal maturation windows. And then, other independent group is also, probing the same, pointing the same fact for thyroid hormone control for, prenatal maturation, probably because as there is a prenatal surge of the thyroid hormones, maybe just after the birth, this, windows of endocrine driver is actually, putting the cells towards, postnatal stage from fetal states. So I think there are a number of hormone control is actually very much, missing, in the existing protocols that can be, leveraged for improved maturity protocols. Yeah, that's quite exciting to hear, actually. The same factors, systemic factor controlling the hepatocyte maturation together with the brain.
Yeah. But just imagine how many more there are that we have no idea.
Yeah, exactly. GH, so many other, exciting hormones we have never studied, right?
Same is true for muscle.
Okay, great.
Those thyroid hormones important.
Yeah. I'm sorry to cut you off. Is there more discussion on this, or should I move on? All right. So we have some questions in the chat that we've been kind of neglecting. So Dr. Imavera, you introduced lymphoids, and have you introduced lymphoids and or myeloid progenitors into your system?
Thanks. Yeah, we haven't worked with immune cells in the epithelial organoids, but there are models where you can culture whole biopsies and maintain the submucosal lymphoid tissue alive. But as a side of that, we are very interested. We're discovering that the epithelial stem cells themselves express a lot of immune receptors as well as cytokines. So some of the volcano plots I showed you of what the stem cells do when they respond to an infection is to produce cytokines to call in immune cells, but also they produce cytokine receptors. And so we're starting to explore that in these systems, especially in relation to these changes in differentiation that are driving the cells towards intestinal metaplasia, which is towards preneoplasia. And they're responding not just to the bacteria themselves, but to inflammatory signals in the chronic infection state. And so you can do that in a reductive way just by adding cytokines to these cultures. And I think that's another very nice way of using them.
Another question in the chat, is anyone integrating other technologies such as 3D printing in these kinds of studies? Or if you had a 3D printer, could you print any type of material, and how would you apply it to your study systems to make it more accessible?
Yeah, there's many groups that are doing, bioengineering projects of 3D printing the stromal compartment and then adding epithelial cells. For example, we collaborated with a group in Switzerland that first published a model of villi in the intestine, where they printed the villi with photolithography inside a matrix. So they cut out pieces of it with photolithography. And then they showed that the stem cells actually understand mechanical topography. So they will settle into the base of that model and they will behave like stem cells, while as the cells migrate up a villus, they differentiate. And we did the same thing for a stomach mimic of the glands. So definitely these structures are helpful. It really depends what you want to study, because they're not easy to do, and it depends on what the purpose is. But if that, you certainly can add that as a one of the variables.
Okay. All right, so we're getting a little short on time. We might have one or two more questions. So in the chat, what about the use of NAMs to replicate complex parasite life cycles that include development within 2 to 4 organs?
Well, I'm the infectious disease guy here, so I actually think the more interesting question right now is that are we able to reproduce parasites that are human specific? Because that's the thing about parasites; they're very species specific. That's why we don't get all the diseases that our dogs and cats get. But there have been really incredible findings. For example, Cryptosporidium is a parasite that's related to malaria and to toxoplasmosis, but it infects actually the gastrointestinal epithelium and can cause incredibly severe diarrhea, especially in immunocompromised children or adults as well. And they didn't have a model for it. We could never grow it. And so now with organoids, these parasites recognize that they're in a human epithelium and they do undergo their life cycle. So even started with just one component, I think it's a major thing that we can do. And this has also been done by a lot of different groups for viruses. Those are the other ones, like Sergio was mentioning, enteroviruses. They're specific to a specific host. So if you want to study polio, you have to study it in human cells. And so, but some of the viruses are not able to be grown in any cell line. And now we are able to do that with primary cells. So that's a very useful thing.
All right, so maybe an interesting last question to finish up, and Sergio sort of touched on this. What kind of regulatory hurdles are presented when using patient-derived organoids? What are the ethical implications and obstacles?
Yeah, well, I mean, there are certainly some of of course, there are like the regulatory implications of like, how good are the models? Do they have predictive value truly? Are we going to feel comfortable injecting into a patient a therapeutic that has been exclusively developed with these models? I think that is like the fundamental question. I would say that even if you have the biology in terms of safety, it still needs to be done in an in vivo and in animals, like at this point, and that's what we've done. Although again, there are ways in which you can circumvent that with N-of-1 studies. We thought it was actually very important to like do it properly. And there are ethical implications also beyond that. Some of them, like with some of these models, I mean, I can comment for the brain because we have a few that are quite often either because of misperception of what is really being done or because they're like, we're not there yet. But for instance, issues of, complex activity that is present in vitro, like pain, I mean, when we built the sensory assembloids, we were, accused of like building human pain in a dish, and that the assembloids are like screaming of pain in a dish, but nobody can hear them. Of course, any neuroscientist would know that that is not, really how it works. That even if you have the sensory pathway of pain, you don't have the emotional component of pain, the aversive component that involves the amygdala and other pathways. So I think there's a lot that is known to really, put some, clear frameworks around them. But there's still issues of consent, for instance, that are very important ethically. How are the cells consented? And especially when you put them in vivo, I think that is sort of like perhaps the most complicated. Like when you build larger sections of the nervous system of an animal to contain human cells, there are all kinds of ethical issues that are arising. We've been trying to address them. We've had a meeting at Asilomar in November, and then a paper that came as a group calling for experts, because it requires expertise both in stem cells and in neuroscience for this. And very often either neuroscientists have been either dismissive of this, and stem cell biologists have been trying to tackle it, but without the expertise that is needed. So we think that a group that has both will be very important for really identifying issues that are truly, need to be tackled. And so a body like this will likely emerge in the next few months with support from some of these organizations. So I think that is very much needed.
I think this has to be discussed very much in an organ-specific context also, because I mean, brain and other tissues are very different in how the cells interact with each other. If you have a biochemical interaction, you can, of course, work, I think, quite well across species. If you work in a system in which the cells need to be integrated directly into the syncytium of an organ, I think here it would be one really has to discuss what kind of animal model should be used. I strongly believe that in many models, allograft studies will have to be done also in animals, and not so much xenograft studies. Again, this is different in the brain, I think, because you have different ways of communication. But if you think about, a muscle cell talking to the neighboring muscle cell, this is typically done through direct gap junctions and connections. And if they are not there, you cannot really integrate these cells well. And what you see is probably more paracrine activity rather than a direct integration of cells. And this is also, of course, an ethical issue. So I can from the German perspective, I can say that our regulators did not like to see xenograft studies in IND enabling studies. They wanted to have a simulation of an allograft study in a large animal model. And I think this was a decent suggestion. Didn't make us easy for us, but I think it was a good suggestion.
All right, looks like we're time is up. So I just want to thank all of you for presenting your work and taking time. I think this is a important discussion, and I think as we move forward, this complementarity of NAMs and animal models needs to be emphasized.
Great, thank you, everyone.
Thank you.
Thanks a lot. We have a lunch break right now, so please be back at 1:15 Eastern Time. Thank you. Okay, so let me start the session, and meanwhile, just post it. Okay, so we are moving to session 2, which is titled Shared Resources to Validate, Promote NAMs, Research Frameworks, Tracking Activities. And I would like to remind participants you can post your questions, and also to presenters, if some questions remains unanswered, please go back if you have time and just answer them in the chat. Okay, so our co-chairs for this session are Christine Nadeau and Thomas Bell. So Christine is a program director in the Division of Cancer Biology at NCI. Her research portfolio includes notch signaling, breast cancer metastasis to bone, and systematic immunity. In addition, she helps facilitate 2 important NCI consortia, the NCI Metastasis Research Network and Oncology Models Forum. Thomas Bell is the Chief Scientific Officer at NDRI, National Disease Research Interchange, and Principal Investigator for multiple NIH-funded projects, including somatic mosaicism across human tissues, developmental genotype tissue expression, and human tissue organs for research resource, HTOR. So, cheers. The microphone from here.
Thank you, Oleg. I get the pleasure to host this session with Christina, so I'm going to start off first, then Christina will jump in. So again, the title of our session, everyone, is Shared Resources to Validate and Promote NAMs, Research Frameworks and Tracking Activities. Just quick high-level learning objectives. So we're looking at identifying infrastructure gaps, limiting NAMS adoption in the biomedical research community, understanding validation requirements across discovery and translational research, recognizing challenges in standardizing and scaling NAMS platforms, and then closing in on kind of understanding opportunities for NIH-supported resources to serve as hubs for validation and the coordination of advancing NAMS initiatives. So first speaker in this session is Dr. Michael Hellrath. Michael is a pediatric surgeon scientist at Cincinnati Children's Hospital Medical Center and also a professor in the Department of Surgery at the University of Cincinnati. He serves as director of the Center for Stem Cells and Organoids Medicine Custom, which I love acronyms like that, which is in his title, which works on advancing organoids for personalized medicine, disease modeling, drug discovery, and regenerative medicine. His research includes for this effort transplanted human intestinal organoids, immune competent gut cell models, and pluripotent stem cell organoids to repair damaged bowels. So I could turn it over to Michael for this session. Like the other one, we'll take all the questions at the end. So Michael, please take it away.
All right, thank you, Tom. Nice introduction. You can go to the next slide. Earlier this morning, if you're able to listen to Taka, he was giving some of an overview of custom. What I will tell you at Cincinnati Children's is that we're on our third decade of using iPSCs. Largely started with Jim Wells, who's on the bottom right. There's a group of us, four, me, Taka, Aaron Zorn, and Jim Wells, who really kind of spearheaded this process. And I would say that the process of iPSC development into organs really drove a lot of the papers and the funding that supported this work. And that by doing so and being part of consortiums, I really want to say the generosity, the academic generosity of sharing resources, technologies, training, the use of a core that Jim started was fundamental to starting the group. And that we largely in Cincinnati established protocols to make the whole GI tract and liver for Taka. And at the time in 2017, we were being asked to do other projects with pharma and others that I recognized and others was very different. And so we came up with the idea that Custom Accelerator needs to be a space that might be able to translate what we do in our research lab, which we call CAR. And finally, if we're going to put these cells into humans, which is my mission, it needs to have more of a dedicated approach. And so trying to come up with a strategy to do this, we formed what was Custom based on a lot of philanthropy and institutional support. Next slide. With that support, we really tripled the number of people at Cincinnati that use iPSCs from about 15 to now close to 40, depends on how you count them. And they now include investigators that do not just the GI tract, but as you see, many different organs. And they follow the same pathways. And what I will tell you is that their agnostic needs of using intestinal stem cells, iPS cells, the medias, the reagents, the systems that we built were largely supported by our previous experience. Next slide. What I highlight here is that most people, like we, develop their models with an end-stage disease as a proof of concept from a single cell line that demonstrates that the human pathology is replicated in the models that they do. Most of these models are in vitro. Some of our models are in vivo. And that's really supporting the academic research support. Next slide. What was key though to me was the gaps in trying to translate the things we do in the NIH type funded laboratory to translate out to the world is going to require a different approach. And so a decade ago, we knew we needed to build infrastructure across, which included not just training and mentorship, but the access to a core and the resources, as I previously mentioned. It was very key to me to provide gold standard benchmarks. As a surgeon, formalizing a consent process that we do for all patients, not just in this institution, but around the world, needed to establish the ability to share resources, to be able to publish, but also to be able to make companies and for-profit use of things so that the resources were expandable. we had to collect data around each patient. A line that is not described from the patient background is a potential problem. And so having standard demographics and conditions with all the lines was established. And the associated tissues that we can get from those patients were driving some of these examples that we feel need to be included when you're making models. And it's important to note, we talked earlier, that do we need animal models? Well, certainly animal models is one support. Humans are the best models, and having clinical data in humans, and the ability to take your cells and put them back into an animal so that the human tissues is a further way of demonstrating the in vitro culture systems that you do are maintaining the ability for at least our systems in the intestine can form a fully functional gut. I fully recognize that we need to have an iPSC library. To have a single line and call a human is not to understand human diseases, and that required a dedicated focus, which we continue to do today. I think it's important to recognize that things done in a lab work well, but if you need to do this at a scalable way, you need to standardize and validate it. And that is extremely important for an automated prototype that we've developed, because without being able to do that, you can't do the complexity of multiple lines in real time and do analysis with AI to support the complexity of human diseases that I feel need to be done. And finally, what is often misunderstood is that the infrastructure to support what is now a milestone-driven collaboration versus just a research-driven collaboration that needs real-time reporting out to pharma requires a lot of infrastructure and reporting and administrative support that doesn't traditionally live in typical NIH-funded laboratories. Next slide. So other cultural differences is that it's important to remember humans are different, and that animal models are the same strain used over and over again to get the same response. And therefore people use same animal models and same single cell strains, and that's been built into the way we've been trained. But we really need to look at human to human variabilities, and we need to be using more than one lines. To do so is challenging and expensive, and it needs support like I've described. People need to recognize that the media is probably far more modifying than the actual cells in your dish, and that many people have fallen back on conditioned media and the use of things like Matrigel, which is highly variable and not physiologic, and I would also argue represents an animal model in and of itself, because that's how the Matrigel is formed. Creating specific hydrogels is challenging, and being able to benchmark the reagents, as we've learned over time, is evolving as they become pure and their biology is becoming different, even though they seem to be the same, without notable readouts for the individual things that I'll touch on in a second. And finally, multidisciplinary teams all have different goals. And to be successful in this, you need to introduce very different people. And what I was mentioning earlier, the end product for us is learning how a disease is done. But a lot of the translational people developing are into the development of the tissue. And so anyone that wants to study an end product needs to have access to that product. They don't want to spend the time and energy to do the developmental process in the dish. And we need to develop a pipeline that gets those tissues to the right people to ask the right questions. And then I'll last say that, a basic science lab is a dedicated team. It's not a CRO. And to throw a CRO on top of a team is very challenging. And so the production of these lines really need to be in a reproducible, automated, scalable system. Next slide. So what is custom? Like I said, we formed this in 2017. It divides us into the academic research, which we're all familiar with. An accelerator was really understanding the ability to de-risk and to be able to do that milestone-driven culture. To do that, I went outside of Cincinnati, recruited Magdalena Cassender that was in a pharma company in Boston, and we largely built a team around learning how to do this optimization and protocol. Finally, as I mentioned, going to the FDA and getting preclinical data and to be able to do GMP production is also being done in that space. Next slide. So with that team, these are individuals that we recruited from 2019 and 20 to just basically do the work. And what I'll tell you, it's a multidisciplinary team with the mindset, as I said, which is very helpful. The team is fully integrated with our basic science labs, and so it's in constant communication, but they're also at the forefront of working with our outside pharma companies. Next slide. So, as I mentioned, the first example I'll give you is how we're trying to develop cellular product for placing into human. Next slide. Advance, please. So our cells, when we grow them in a dish, in intestine can translate into the animal. It can also be used as a cellular product delivered into the lumen of the bowel, which will heal transmurally the entire GI tract. So it provides not just epithelium, but muscle, vasculature, and nerves. But to do this and to put it into humans, we obviously have to convert this into GMP process. Next slide. So this is complicated. So to do GMP is to completely replace everything that's in research grade and do it in a xeno-free way. And this is completely done in a way of reporting out that is process developing to be able to present to the FDA. And what this did was allow us to look back at the stages of our methodology and look at individual readouts to show that we're actually patterning the cells that we want in the correct way we want so we can control this. Next slide. To be able to make tissue that's already patterned and frozen down is essential. It's essential for also research, but it's very essential for using a clinical product. This is very key to be able to biobanking and sharing resources. And you need to have a readout, namely a transplantation, to know that the tissue that you're actually freezing is behaving the same, even because many times it will grow in culture and look very similar, but it has effects of that processing that you have to control for. Next slide. Quality control, you have to, as I said, built in these quality metrics. And this has largely been very helpful now, not just to this, but to other projects. Next slide. Scaling is to be able to make it into a scalable way that's going to be able to be used clinically. Next. And then animal studies is obviously the off-tumor effects and stuff always will require an animal in our minds, as we discussed earlier today. Next slide. So what does standardization actually mean? And so to be able to have reliable, scalable, you really need to be able to characterize your cell source. You need to know where it's coming from, have all the differentiation trajectories fit. Next slide. So the engineering to be able to do that is to be able to remove the person-to-person variability and the fine examples of why things can be done in my lab and others has to do with the very high skills of the people. But it needs to be able to be done anywhere by anyone using the same systems. And this is going to enable AI and other feedbacks I think are going to be essential for the field. Next. And so that's for data standardization. So to be able to have outputs and reads in databases that are standardized and can cross over is a large part of what we do in this space. Next slide. So standardization is not simplification. It's the foundation that enables reproducibility, scalability, and real world impact. Next slide. So here's an example of where we had to take something from a handcrafted model and put it into a scalable infrastructure. And I'm going to give you an example of research that Taka presented. Next slide. So Taka showed you earlier today his model that can look at drug toxicity and cholestatic effects. This is published and it's extremely good at showing how the livers can look at patient-to-patient variability. But why was this not scalable? Well, there's many things that were limiting this model. It was dependent on matrigel. It was labor-intensive. It wasn't scalable. And it really needed an expert-driven analysis that took a lot of time. Next slide. Next slide, please. So to engineer the scaling, we had to develop an automated system that we use micro cavities that got rid of both the matrigel and the ability to visualize each organoid over time. Next one. Next slide. This is fully automated, where all of the liquid handling and the imaging was all done in an automated process. Next slide. And we were able to use AI to investigate all the dishes to be able to interpret the data. Next slide. So here it just demonstrates the removal of matrigel into these dishes gives us more uniform organoids that are accessible for data. Next slide. This just quantitates that the reduction of both inter and intra-line variability is reduced with this process. Next slide. So going back to the question of how do we study a population in a dish that may have an effect here in drug toxicity, certainly next slide. We take advantage of databases and iPSCs. I told you we've developed an IPS database, which is now over 750 consented donors, which represents really the heterogeneity of the US, but we have similar ones that reflect Asia and Africa, which is equally important, and all behave very differently. This required a very dedicated process of focus to develop, and we continue to do so. Next slide. So with that, we were able to complete this study that was supported by Danaher Molecular Devices, where 30 different groups, 10 in each, had different responses to the drug responses that Taka generally went over. And with this, we were able to demonstrate that we had the ability to predict specific risk of DILI that was predictable in the European population, but not in the Asians and Africa, to represent the fact that the risk score that was developed against European, and that you really have to have your population identified if you're going to do AI modeling. Next. So this just demonstrates a in vitro platform where without immune cells, you don't able to see the specifics. And so the addition of immune cells and the complexity is absolutely required. Next slide. And it just was published to just demonstrate the power of the potential of in vitro model systems. Next. So the custom ecosystem, I really think, involves discovery, and that's basic science. The translation is what we call in that space. It really is, I think, essential for using something that's going to get out, and then deployment. And obviously, regulatory and validation back to human models, human patients is absolutely, I think, important. If you make the ecosystem right, it should be seamless, and it needs shared resources to have the real world impact we feel. Next. So again, I just to highlight where we are today, we're supporting our basic science researchers. I did not talk about my own science. I love to, but I tried to be more general to meet what I felt was the needs for this talk. We can talk about the custom accelerator. It's a work in progress. It was really largely supported by the ability of me to raise philanthropy, institutional support. Many of the things we do are not really deemed that exciting to people sitting in the study section, but are absolutely essential and require a tremendous amount of work. And so I think it's benefited a lot of our investigators, and I think it's absolutely important to meet the needs that we're here talking about today. And then organoid medicine, I'll just highlight is that we take care of, I'm a surgeon, so I have many patients that I've actually changed the care that I do today just based on stuff that we've learned in the last decade from their own tissues. And so the more we understand the rare diseases that we take care of from organoid medicine, the more we're going to find discovery that's going to become more generalizable and translatable to larger, less complex, less isolated diseases and more broad diseases, which is ultimately the goal. Next slide. So obviously I won't have a chance to answer any questions today, but I look forward to hearing the other presentations, and we can talk when it's all done.
Great. Thank you so much, Dr. Hillmrath. All right, it's my pleasure to introduce our second speaker, Dr. Scott Lowe. Dr. Lowe is the Chair of Cancer Biology and Genetics Program at Memorial Sloan Kettering Cancer Center and an investigator with the Howard Hughes Medical Institute. Dr. Lowe began his independent research at Cold Spring Harbor Laboratory, where his group made significant contributions to understanding the p53 tumor suppressor, multi-step carcinogenesis, cellular senescence, and tumor cell drug resistance. At Memorial Sloan Kettering, his team integrates mouse models, genetics, and genomics to explore the interactions between tumor cells and their environment. And today he's going to be talking to us about NAMs for integrating cancer. Thanks, Scott.
All right, thanks. Can you hear me okay?
Yes.
All right. Yeah, so I'm going to talk a little bit about efforts here at Memorial Sloan Kettering Cancer Center to use these new approach methodologies to interrogate cancer. Just reminding all of you that cancer, of course, is a very complicated tissue pathology. It's highly heterogeneous. It involves disruption or alteration of a wide range of basic cellular processes. Some of these can be targetable, and of course, this is one reason we want to understand them. Importantly, it evolves progressively through loss of growth controls and cell state changes. It arises in a complex tissue microenvironment. And again, the heterogeneity within tumors can lead to resistance and recurrence. And so again, a challenge to model outside human patients. Now, the most widely used cancer model, sort of historically over the last, I don't know, 30 or so years, longer for cell lines are shown on this slide. The 2D cell lines derived from patients and genetically engineered mouse models. I've listed the pros and cons of both. I think we all appreciate them. Obviously, the 2D lines have been by far the most broadly used, most commonly used in industry, but often not considered incredibly predictive. Genetically engineered mouse models capture much more of the physiology, but of course, they're mouse and not human. And there's also some concerns about their predictivity. So that's led over, again, for the last 20 years or so to the development of additional and various mouse models we're calling new approach methodologies. Some I'll talk about are listed on this slide, ranging from patient-derived xenografts all the way to new genetically engineered mouse models. Now, here at Memorial Sloan Kettering Cancer Center, have, I think, a really outstanding facility that enables investigators to do preclinical or in basic discovery science studies in animal models. It mirrors very much what we have in the clinic for patients, with the ability to monitor tumors, to characterize them in great detail, to do pharmacokinetics on drugs, so on, to characterize genomics. And this allows, I think, unique capabilities that most labs wouldn't have on their own, gives access to a range of different systems, ensures consistently across experiments because of the trained personnel. Data is all collected in one place, so it's easy to integrate and store. Because investigators sometimes use the same models over and over, it also allows cross-comparison between studies. Now, this facility was initially developed as a setting to do human tumor xenografts in nude mice, probably in the late 1990s, but has evolved to be much more sophisticated. One of the major resources is developed, in fact, this was funded by a grant, a U54 grant from ORIP about 5 years ago, is the patient-derived xenograft system. This, of course, is when one isolates tumors from patients and grafts them into immune-deficient mice, allows them to take and then uses them for various studies. Importantly, here at MSK, all of these xenografts that we have are linked to the genomic data, the transcriptional data, the clinical records, and all of the histopathology. Of course, these have a number of strengths. They're human. They better maybe retain features of human cancers than the 2D lines. Certainly, it captures the heterogeneity. Now that we sequence all the tumors, we can know what that heterogeneity is, at least at the genetic level. But obviously, it's a human and mouse setting. It still uses mice. They can be quite expensive, and while we have many models, it is prohibitive to do a lot of them together. This is sort of a summary of the resource that was actually enabled by our U54 grant. We have 2,400 different patient-derived xenografts capturing a very wide range of tumors, including very rare cancers. And these can be used again for a variety of studies. I'll just show one example here where there was a co-clinical trial done with new NTRK inhibitors, this is now a few years ago, in a patient whose tumor responded, ultimately relapsed, and in the patient and in the PDXs that enriched for cells with a RAS mutation, which suggested at the time to combine with a MAP kinase inhibitor, trametinib, that led to an improved response, and that also worked in the patient. Now, we heard also more about this organoid technology. This, again, has really been something that is, paralleled, if not replaced, our PDX approach, where one can identify or characterize from human tumors patient-derived cells in vitro, culture them, store them, characterize them genomically, just like for the PDXs. I'm just going to mention one sort of platform that Karuna Ganesh developed, which I think is really exciting, and I think illustrates the power of this, where one can isolate, from example, normal organoids from the colon, from primary tumors, from lymph node metastases, and from distant metastases, study them together, all from the same patient. And again, this is a very efficient methodology. One can actually genetically manipulate these cells. But of course, organoid cultures in Matrigel by themselves are not perfectly recapitulating the environment. Now, one can do co-culture studies and so on. But again, these can be a flexible and powerful human-based approach. And Karuna's used this, for example, just comparing primary versus metastatic samples to actually show that the metastases sort of acquire progressive plasticity, acquire fetal states and other states you don't tend to see very much in the primary tumor, and has used these for preclinical studies as well. Now, another platform that we can integrate into our core is really generating models from human embryonic stem cells, or iPSCs. This sort of a more reductionist setting where one starts out with a normal tissue that can be derived by differentiating embryonic stem cells to a certain lineage and then engineer it in a certain way, for example, putting in controlled genetic alterations. Danwei Hongfu here at MSK has done this in sort of the pancreas, putting in pancreas cancer drivers, and use these to quite great effect. Again, these are really great for studying sort of developmental biology. It allows isogenic models to be produced, really great mechanistic studies. But of course, again, these were derived in vitro and lack the tissue component. Nevertheless, she's been able to generate, with successive engineering, essentially cell states that mirror the major driver gene combinations in human pancreas cancer, and use these to study various aspects of disease. We heard in the keynote about sort of organ-on-chip methodologies. We've been working together with Dan Hu and Heijiang Liu at UPenn to sort of bring these on board, really taking advantage of technologies they've done. Again, we've heard that involves mixing human cells and stroma on a chip where one can have a dynamic microenvironment and try to really mimic the physiology of tumors. And you can use these in different ways. One that we've done together, again, with Dan and Heiju Zhao, is to create tumor cells together with a sort of inflamed stroma and test CAR T cells. Control CD19 CAR T cells are one that we've developed that targets a cell surface protein that's high in a lot of cancers that have sort of a fibrotic environment. And one in these sort of organ-on-chip studies can see the T cell penetration and killing. And an interesting factor about these particular CAR T cells is they kill not just the tumor, but some of the inflamed stroma. And again, you can't see that here, but we can study that as well. Now, another area that, again, we're quite excited by here at MSK because of our access to human tissue is to actually use tumor-derived precision-cut tissue slices. Again, this preserves the native tissue architecture and involves embedding a thick slice of a tumor tissue in agarose, where, depending on the cancer type and conditions, these can be cultured anyways from days to about a month. And studied with different models. And of course, these retain the native architecture. They have the sort of natural immune composition, albeit outside of an organism. But limitations are at least some, for example, liver really have a short culture duration. Obviously, you have to have access to the OR and be able to work quickly. So this isn't something anybody can just do, but nevertheless can be a powerful approach. I'll skip this slide for the purposes of time. We also have been interested in doing new mouse models. Again, mouse models, despite the fact they're mice, can capture the full sort of tissue context of cancer from early initiation to progression to metastasis to therapy response. But of course, the standard genetically engineered mouse models are very slow. It requires a lot of genome engineering, crossing mice where there's a lot of weight. And so there's been a number of approaches that have been used to try to engineer immune competent mouse models that are faster and involve less waste and can capture greater complexity of the genetic features that occur in the human disease. We call some mosaic. This might say, for example, take an organoid model, a mouse tumor-derived organoid model, and graft it in an orthotopic site. Another approach we've used is actually not to cross different alleles, but to engineer them all in one embryonic stem cell and make the mice directly from those. So all the mice are exactly what you need. And another one is to use somatic engineering, where one can engineer in tissues the types of mutations that drive a cancer. And these can be quite powerful, although these do require some substantial expertise. Here's just an example of a study actually from a paper we recently published where we could engineer ES cells that have this complex genotype, so this equivalent of 4 different alleles. We make mice from these, so there's no crossing. And this particular strain can turn p53 on and off in the pancreas just by adding doxycycline. So it creates a lot of flexibility for temporal and spatial perturbations. And this is just an example here. We know in pancreas cancer in human and mice, inflammation can expand cells that are sort of pre-neoplastic, but they don't continue to progress. A cell state that we know is important is these stained by HMGA2. And if we suppress p53 here by doxycycline, you see that state expands. So again, we're really looking at this sort of early neoplasia and how p53 can constrain it using this sort of approach. And I should point out the tissue microenvironment not shown on this slide dramatically changes around these p53 suppressed cells. Now, another approach that we really like is something we call electroporation-based mouse models. This is where one actually engineers tumors using constructs like CRISPR or a cDNA transposon vector that is electroporated directly in the tissue. This involves usually a surgery that exposes the organ and then electroporation. And that's a lot of work. But what's powerful about it is that one can mix and match the types of genetic elements that one can introduce and can also alter the host recipient hosts that is affected. And these can be probably at least 10 times faster than the typical genetically engineered mouse model and enable a lot of different genotypes to be produced. And again, here's just the illustration of one of the cool things one can do with this sort of approach is, if one wants to understand, say, for example, immune surveillance, one can electroporate genetic elements. This happened to be MYC and something, CRISPR that disrupted APC in either a wild type or immune deficient animal. And not surprisingly, in the absence of the immune system, the tumors come up faster. But the real striking result, this is a gastric cancer model, if one looks at the liver, there's massively more metastases, suggesting again the importance of immune surveillance in this context. And so again, one can take advantage of these models and the array of them to sort of match a question to the model. And again, at places like MSK with the anti-tumor assessment core, it allows us to sort of combine our expertise into one place where people can, with specific questions, can address this specifically. I just want to end and say, of course, and we heard a little bit about this before, there's also a lot of interest in using human as the model. Cancer, of course, is a very rich field for data. And here, and at many institutes, cancer centers across the country, one can get from patients genetics, chromatin, gene expression, cell state, tissue organ, and patient data all together in one place. And of course, this is a very fruitful area to apply things like artificial intelligence. And all of these data are starting to also get incorporating data from these perturbation models when a patient has a study that's been on their tissue. This is all integrated into the CBio portal many of you may have seen, which allows this data to be put in one place people can access, but also apply AI. And I don't have time to talk about this paper, but a study that was published about a year and a half ago really was the first example here of how one could actually take this information to use it to think about better ways to monitor patients and predict their behavior. And so I'll just finish by saying, of course, we spend a lot of time discussing here today and in general about the best system to use is one better than another. And I've given you some examples of different types of mouse modeling, or I'm sorry, cancer models. But I just want to remind everybody, saying from George Bach, sorry, some of the typos here, is that all models are wrong, but some are useful. And so I think it's always really important to think about what the question is, what we want to know the answer to, and use the appropriate model for that. So with that, I'll stop and let the next presenter go ahead.
Thank you very much, Dr. Lowe. Excellent talk. I get the pleasure to introduce the third speaker in this session, Dr. Brenda Ali. She's a professor of biomedical engineering and pediatrics at the University of Minnesota. Her interdisciplinary, and if I say it, interdisciplinary research lab focuses on an exciting pathway of taking integrating stem cells, engineering, cytometry, 3D printing to study how extracellular matrix proteins can influence stem cell behavior in the cardiovascular systems. Our teams developed extracellular matrix proteins, or biolinks, for 3D printing human cardiac muscle tissues for studies. And let me turn it over to Brenda, please.
Great. Thank you so much, Thomas. This is wonderful to be with this group today and as part of this workshop, because I've been thinking about the topic of my title for several years now. And it's great to be able to bring it here and to be able to hopefully discuss a bit in the panel afterward. So I want to start, before getting to that idea of coordinated infrastructure, to just a little bit about my research, because it helps you see my lens and also why we've kind of progressed down the road as we have here at the University of Minnesota. So my lab is very interested in how the extracellular matrix impacts on stem cell behavior and importantly, how it drives the differentiation of particular cell types when presented in particular combinations and concentrations. We showed that almost 20 years ago now, and what we've taken from that are many different avenues of study, one of which is to take an engineering approach to try to optimize a formulation of ECM proteins that, when provided to a stem cell, will drive the specification of particular cell types. And we've developed 3 different formulations now. And the one that I'll share with you today, we've utilized to drive the specification of cardiomyocytes. And it just so happened that when we developed that formulation, 3D bioprinting was just becoming hot. And I tried to stay away from hot topics. However, it just happened that our formulation fit nicely into a bioink format. So we could start to print these ECM proteins, and we decided to print them in the context of a structure or a geometry that mimicked the chamber of the heart. And others were starting to do this as well, but they were printing the differentiated cell types. And we're finding some success, but had to use large numbers of cells, and especially in the context of a cardiomyocyte, it doesn't move well and it doesn't proliferate after it's specified. And so it's really challenging to obtain contiguous muscle function reliably from print to print. And so what we did differently was use that formulation I talked about. So instead of printing the differentiated cell, we printed the stem cell and allowed them to expand and then differentiate in the construct. And in that way, we were able to achieve contiguous muscle function. And that's shown here. You can see this macro scale video on the left is showing the spontaneous activity. Hopefully it'll run, of what we call an HChamp, human chambered muscle pump. And as that's loading, I can show you the histology on the top. We have largely cardiomyocytes as indicated by the cardiac troponin I staining with a few smooth muscle cells, endothelial cells, and later we showed also cardiac fibroblasts. Not sure why it won't play, but I'll give it another second. You can check out this paper as well as the videos. It's already published. It's in Circ Research. And just to show you too that the thickness of the construct is about 500 microns. That might be inhibit me from advancing too. Hang on one second. Go to the next one, and I hope my next videos will work. Let's see. Okay, so I just want to, oh, this is maybe not the best slide to go to, 3 more videos. I'll just try one. This is just optical mapping, just to better demonstrate the contiguous electromechanical activity. I'm glad to see this one working. Again, the big advanced is contiguous muscle function. And so we also are able to measure pressure volume dynamics using our conductance catheter rather in the laboratory of Dwayne Townsend. And this way we can get one of the first clinical measures of cardiac function in a dish. And you can see on the top both the pressure and volume traces, and you see this pressure volume loop, which doesn't look like the clinical case because we don't have valves, but you can see actually a volume inside the loop, and it changes with the addition of drugs like isoproterenol. We can also perfuse it in a bioreactor that we just published a couple of years ago. And importantly, with this bioreactor, it's not just perfusion. We also place mechanical valves outside of the bioreactor. And by timing them and coordinating them with the spontaneous beat of the H-champ, we can actually mimic cardiac waveforms while also measuring pressure volume dynamics. We've collaborated with Victor Brokas's group to not only physically measure some of the flow profiles and flow associated metrics, but also to be able to model them. This is important for studying various disease states that progress with altered fluid dynamics. And it's also been important and actually motivated. Much of this work was motivated by our collaboration with Medtronic. They were very interested in a system where they could, in addition to measuring clinical metrics, also have a structure that was large enough to apply some of their medical devices, including pacemakers. And here I'm showing a study we published using ablation catheters. So for this community that we've been talking a lot about NAMs and in vitro model systems, and usually the default is we're studying health or disease or we're studying new drugs. Well, I just want to put out there that in addition to drugs, medical device testing will really benefit from this platform because they can make all kinds of different changes and tweaks to their device, testing them in really large sample sizes before going into a preclinical animal model. So this isn't the only engineered cardiac tissue. I wasn't able to catch it, but I think Wolfram Zimmerman must have talked about his engineered cardiac tissues as well. There are many different varieties, and as the previous speaker noted, the model system that's most appropriate depends on your question. And so in the cardiac space, we have everything from spheroids on the left-hand side, progressing into patches, and then perhaps the most commonly used engineered cardiac tissue is the engineered heart tissue, which is just a strip of tissue, typically suspended between stationary posts to impose passive resistance. And we also utilize this model system in our lab. But what we were finding, and what a lot of the speakers today have been talking about, is that providing these model systems to other laboratories can be quite challenging. And so about 6 years ago now, we started to partner with Mayo here in Minnesota to be able to provide the model systems we were developing in both of our campuses to our campus and also to the other campus. And we were helped by support from our state in the form of the Minnesota Partnership Grant. And what we really wanted to do, and Mayo was very focused on patient-derived organoids and engineered model systems. And here in University of Minnesota, we were more focused on iPS-derived systems. We really envisioned the ability to expand these model systems and distribute them at our universities, much like so many of you have done. And we're now able to do that. Where we have run into challenges is with preservation. And we've heard a little bit about it, but I think it's a much bigger issue than people are discussing at this point because there are a lot of other things to consider, like quality control on their fresh tissues. But we really wanted to look at preservation. And it seems like for some, especially organoid types, there's been some success. I think we're still developing those quality control metrics because what might appear to be functioning after either cryopreservation or cold storage may not be exactly the same as what the fresh tissue was doing. And so we've been thinking a lot about that because cardiac tissue has been especially challenging to preserve, especially when you get larger than the cell or organoid level. And that's also reflected in the fact that donor hearts have a very short time span in which they can be used. And I think it's because of these attributes of cardiac tissue. Basically, their intracellular structure, especially the sarcomere, is very susceptible to and is damaged easily by ice formation. Also, the cells rely on direct electromechanical coupling and also cell ECM engagement. And when that's disrupted, you get a bunch of cells that aren't no longer functioning. That means the tissue doesn't function. You may have islands of functioning cardiomyocytes, but the entire tissue won't function together. Also, the cardiomyocyte has very high density of mitochondria, meaning high metabolic vulnerability. And cryopreservation-induced calcium overload is especially problematic for the cardiomyocyte that relies on that tight interplay of calcium efflux and influx. So we've been thinking about that a lot and how we can get past it. And I'm excited to share some new data with you, a breakthrough we recently had to recover macroscale electromechanical function in engineered heart tissue. And we did this using vitrification and a novel cryoprotective agent that addresses specifically those cardiac tissue susceptibilities I was just talking about. And then we also used a cryo mesh to hold the tissue and also to facilitate heat transfer to reduce thermal strain. And that comes out of the laboratory of John Bischoff, who I'll talk about again in just a minute, just to show you the videos here of a fresh construct. It's off its posts right now, just so you can see its macroscale beating function, and then the exciting part, which was the cryopreserved sample coming back after preservation. And so this is the beginning of me thinking about coordinated national infrastructure. So we really benefited in that preservation study from interaction with an NSF engineering research center called ATP Bio. And this center is led by John Bischoff and Mehmet Toner. And the whole goal is to stop biologic time and to be able to preserve everything from organs down to organoids and small model systems. And so, again, we benefited tremendously from that. So we started thinking a little more broadly. Can we get past our individual local organoid or engineered tissue cores and think about a need for a manufacturing, preservation, importantly, and distribution center? And so together with some really great collaborators, we put out that concept last year. And we've been together continuing to think about that and how we might, how that may or may not be beneficial for the field. What would the structure look like? And so we started to think about what the center criteria would be, not much different than what has been said already here today. Inconsistent starting material, big challenge from taking lab-grown tissues into full research impact. Limited ability to generate large batches, even in-house and even in our organoid core, we sometimes are limited in that way. Inconsistent evaluative metrics. I think the whole field is really driving toward that. And then limited ability to share and store, which is what we focused on most recently. And so together with a bunch of collaborators across the country, we've been thinking about and sort of envisioning, and this is just in the visioning stage, something we're calling MORE, or manufacturing organoids for researchers everywhere. where the idea, and it's an idea, is that organoids or model systems that are mature, or even those that are already in a company and are being sold and developed, but still need some process standardization, would come to this facility for scale-up production, quality control, preservation, storage, and perhaps distribution, or a subset of those things. But that we would have the national infrastructure to do that, to be sure that there's a central location that we're all utilizing or at least touching base with in some way, shape, or form. Whether it's one physical space or a compilation of different types of infrastructure in different spaces, that's probably the more likely version or rendition of this that may come to fruition. Because I think this is an incredibly dynamic time, as we all know, with lots of resources being invested, new ones being considered, and it's exciting to be a part of. You've heard of I think it was mentioned in the very beginning, among other things that the NIH is supporting, the Standardized Organoid Modeling Center, of course, the Research Resources Directory that ORIP has had for a long time, and then other bodies like the Standards Coordinating Body, which has been trying to help govern SOPs and quality control of engineered tissues and regenerative medicine for some years now, as well as companies like ATCC that were started long ago to help us do the same thing back then for cells and cell culture, and maybe utilizing a lot of the lessons that they've learned to help us build out something similar. And I know that Carolina, the next speaker, is going to expand on that. And I think the word that I'm hoping to just leave you all with today is coordinated. I think that there's a lot happening, a lot of exciting new investments happening. And I think as a field, we want to stay coordinated. We want to learn from the lessons of custom. You just heard earlier in this session, and other institutions that have really been building out a lot of these features. How do we come together to share what we know and continue to build that out even further so that we can have reliable product? We actually get to the space where we are using this regularly for IND applications, and that people start to develop lots of confidence in these model systems excuse me, going forward. So with that, I just want to thank those in my lab who've contributed to both the work and some of the ideas going forward, and then all of the collaborators that I showed on that perspectives piece last year. So with that, I will turn it over to the next speaker. Thank you very much.
Great. Thank you so much, Dr. Obel. Okay, so I'm happy to introduce our fourth and final speaker for this session, Dr. Carolina Lucchesi. Dr. Lucchesi is a principal scientist and head of the Microphysiological Systems program at ATCC. And at ATCC, Dr. Lucchesi leads efforts to enable the adoption of advanced 3D human-relevant models by developing standardized, quality-controlled biological resources, harmonized workflows, and fit-for-purpose qualification frameworks. Her work supports shared research infrastructure by facilitating validation, benchmarking, and consistent implementation of microphysiological systems across the biomedical research community. And I will hand it over to you, Dr. Lucchesi.
Thank you. Good afternoon, everyone. Can you hear me well?
Yes.
Today, I'll be talking through how standardized biological materials combined with quality frameworks and scalable validation are essential for bringing NAMs from promising research tools to reliable and decision-grade platforms. So first, a quick overview of my organization. ATCC is a nonprofit institution with over 100 years of experience in biological material stewardship. Our mission is to advance public health, scientific innovation, and enable national biodefense for emergency responses. At the moment, we count with over 700 employees, including scientists with deep expertise in cell biology, organoids, MPS, cryopreservation, sequencing, bioinformatics, and quality systems. We have been operating 24/7 with secure access control facilities to ensure uninterrupted handling and compliance. And this institutional backbone that supports everything I will show you next. So we heard earlier today all of the barriers limiting the 3D model adoption, such as inconsistent starting materials, variable in culture methods, weak cross-lab reproducibility, and immature qualification frameworks. The common thread is the lack of defined performance criteria or consensus guideline for qualifying organoids as NAMs. And without comparable inputs, QC metrics and data practice, 3D models remain hard to interpret across labs and tissue types. So these practical barriers are standing between promising 3D models and the real-world adoption. And the challenge is extended to a microphysiological system, which faces additional layers. MPS platforms vary enormously in design, such as static, fluidic, scaffold-based design, making cross-platform comparison extremely difficult. But here's the critical point. Regardless of the technology, high quality and well-characterized biological inputs are essential. So the cells going to the system need to be authenticated, consistent, and reproducible within the same donor or a lot. Beyond biological inputs, we need harmonized protocols for seeding, culture, and characterization endpoints. We need a consensus for qualification criteria to determine when a model is a fit for purpose, having a level of validation and quality robust enough to successfully meet the demands in a specific context so that the results from one lab can be meaningful compared to another. And while the organoid technology is advancing quickly, with models across many tissues, the field remains fragmented across institutions. So organoids are made in small batches with variable starting materials, inconsistent QC across researchers, and limited distribution, which makes it harder to build cumulative evidence. And this fragmentation, it is a scientific problem, not just a logistic one. So if every lab is working with a different version of the same model, the field cannot build collective knowledge, and comparisons to animal or clinical data become unreliable. My colleagues presented very clearly the need to address reproducibility. So first, authenticated provenance such as RMB and donor consent, standardized quality control baseline, such as identity, sterility, mycoplasma, viability, and karyotyping. Third, molecular and functional characterization against the tissue of orange. Fourth, lot level consistency through defined passage window and cryopreservation. And last, align metadata that travels with the model. So these shared resources organizations are uniquely positioned to maintain these standards at scale, serving as a neutral, quality assured hubs for the research community. And building on those quality foundations, we need to structure framework for determining when a model is truly fit for purpose for a given application. Number one, the biological fidelity asks whether the model resembles the target tissue. Number two, the functional validation asks whether it predicts relevant outcomes across operators, sites, and lots. Number three, the translational utility asks whether it can support decisions include regulatory relevant endpoints. And the benchmark is only credible when materials are standardized and QC controlled. Now, let's take a moment for the conceptual point that shapes this workshop. Why it's important to integrate NAMs with animal models than rely on each other alone. They might answer different questions. NAMs add a human-specific mechanism, genetics, and patient context, while animals add the whole organism, PK, immune interactions, and longitudinal endpoints, or when human and clinical data is not available. The most useful signal is integration. When the data converge, it raises confidence, while the discordance is informative and drive points to hypotheses such as species difference, exposure, or technology maturity difference. So standardized materials make that comparison credible. And a centralized infrastructure is practical enabler for everything I've described. So developers optimize the models and deposit the models into a research organization with infrastructure to provide authentication, SOPs, manufacturing, preservation, release criteria, and wide distribution, ensuring research is everywhere with the same characterizing starting materials. That is the bridge from a promising local model to a global, accessible, consistent, trustworthy NAM resources. And here is a simplified version of a production workflow, starting with acquiring the model from developers, characterized, develop SOPs and release criteria, establish cryopreservation and traceability, then bank and distribute it, and provide downstream end user support. The point it is end-to-end control. Each lot carries a documented history of the materials, reagent, equipment, handling, and the performance. And very importantly, it facilitates the adoption by making it possible to deploy models developed by multiple entities with standard settings, open protocols, and make it accessible worldwide. Now I wanted to share a successful consortium that brought stakeholders' strengths together, the Human Cancer Models Initiative. In 2016, NIH launched the HCMI to generate large collection of patient-derived cancer models. So institutions across the world received resources to develop the models, and ETCC contributed with the infrastructure in place to deploy the collection, making it visible and providing end-user training and support. The NCI also managed the HCMI searchable catalog, which is a tool to facilitate the model selection and links it to the NCI genomic data common page, which provides the patient clinical data and molecular characterization as an open source. So it does take a village, and a consortium setting can bring strengths together for the development, characterization, data access, manufacturing, distribution, and operating as a one ecosystem. Now, let's take a look at the function evidence. In this case, the context was access a model sensitivity to a drug that was targeting a specific mutation variant, the KRAS G12C. So using colonic models with different KRAS variants or the absence of this mutation, the sensitivity appeared where the biology predicts it, the G12C mutation. So this mutation, let me see if I can get it, my pointer, apologies here. So this mutation appears where the biology predicted here on this mutation, and this specific response is kind of a functional evidence that a standardized organoids can provide when it can become clinically in molecular context. And this slide reinforces the biology relevance at the transcriptomic level and sets up the cross-model comparison question directly. So the organoids cluster away from the cell lines and preserve difference between the primary versus metastatic disease state versus the cell lines. So before using a NAM for a benchmark, we need first to show that its biology represents the disease that is being modeled. And to connect NAM outputs with the in vivo evidence, we need to reduce the noise before comparing the results by using authenticated lot control inputs, define concordance metrics in advance, benchmark against reference data sets rather than single studies, use patient-derived models with linked clinical and molecular data when possible, and treat discordance as information, not failure. Overall, an infrastructure enable all five by standardized material, QC, and the metadata. So the future is a portfolio. It is not a one model type. Technologies such as spheroids, co-culture, organoids, iPSC-derived tissue organ-on-chip systems each fit different questions. And our goal is to make contributions in quality control inputs and harmonize protocols across that portfolio so model complexity is matched to the purpose. As an example, we perform omics characterization for hepatocytes to provide donor level variation. So the genetic background, CP and transport expression, proteomics and transcriptomic and functional activity all affected the interpretation. So this characterization enable a lot to be used as a reference because users know what the biology that they are comparing to. In addition, using harmonized protocols, we perform basic model characterization, which can facilitate complementary data across platforms. The choice of a spheroid or fluidic or scaffold culture should follow the endpoint being tested. So overall, a shared infrastructure can advance NAMs through some actions, such as validation hubs, reference standards, harmonized metadata, cross-model studies with shared inputs, scalable access and training, and cross-sector collaboration. Our mission is to support the community with stewardship, quality system, and distribution capacity, so validated NAMs can be used consistently and confidently. I would like to acknowledge the fantastic MPS team and all of the people supporting these efforts, and thank you for the organizers for bringing these stakeholders' strengths together for such an important discussion. Thank you.
All right. So again, thank you, everyone. To help with the discussion, we just put a couple of questions here to get things started. So we put 8 questions out. So everybody jump in first. Christine, or you want to do? I'll do the first one.
Sure.
Okay.
Yeah.
All right. So I would put the first one to Brenda on the left-hand top left here. What are the most critical infrastructure and resource gaps currently limiting the broader adoption and validation of NAMs in the biomedical research community? And others can jump in as well. Brenda, I love that you point the ATP bio stuff. I saw your talks there. I love John's ideas of getting things out. So please share your thoughts on that.
Sure. Yeah, maybe I'll kind of pull out the things maybe we haven't talked about as much, in addition to kind of reiterating. I love the idea of centralization, as I talked about in my talk, and I loved Carolina's second to last slide about what could be provided in a neutral, centralized location, and I think that's important. The gap that I think is still there, I think we're going to get at scaling, I think we're going to get at QC, and all those things technically are very feasible right now. The thing that has not been technically conquered, in my view, and what I kind of tried to bring out in my presentation, is the preservation. I think some systems don't need it. They're just a little bit perhaps easier in that the cells don't rely as much on contact with each other or the matrix. But I think to get to the point where we can actually stockpile, store, and then disseminate NAMs, that's what I think really needs the most investment or attention right now. And then the second thing I'll say is just in terms of the interface between human and animal models, I think the NAMs provide a really nice clear bridge. I mean, you can do with the numbers, which I think is also linked to the preservation. With the numbers, you can really start to do, design of experiments, really start tweaking therapeutics before they get to the animal models, which I think in most cases are still very relevant and needed. There are, however, some animal models that just don't recapitulate, or where we've seen that the clinical outcomes just are the opposite of what's happening in the animal. And in those cases, I think we can really benefit from the collected animal data we already have, utilize AI, and really try to get at what are the key features missing in that animal model that we really need to recapitulate in the NAM. So 2 kind of very separate ideas, but that's where I see the big gaps.
Super helpful. Thank you, Brenda. Are there any other speakers? Michael, Scott, Caroline, want to address that question as well? Or you did a great job. Thank you, Brenda.
If not, we can move on to our second discussion question. This one we wanted to direct to Michael. What level of standardization is needed for biological materials, including provenance, quality control, and documentation, to improve rigor, reproducibility, and cross-study comparability?
I think that's an important point. The standardization, to me, if it's human, is the patient it comes from. So understanding the biology and, specifically people who do or don't respond to a therapy, have a specific, drug toxicity, whatever that's been documented, is very helpful then when you try to model it back in the dish. And so, you have to be able to generate tissues that represent those patients. And if it's an iPSC model, that means scaling iPSCs in real time to be able to do the appropriate studies against that. The readout also sometimes, and we talked about, the issues with Brenda's and the cryopreservation, where many people's models in a dish they say are fine when they thaw them. When we put them back in the animal models, they've been dramatically changed. And lines and tissues that are used all the time, whether they're CD31 cells and you say you have a vasculature from a UVEC, they're tissue specific and they don't behave similarly when you put them back. And so I think many models, just like animal models, are going to prove to us to be useful in some ways, and are going to also get us down a pathway of mistakes. And so the more you can validate your assay, the better it's going to be. So you need animal models. You also need to be able to transplant your NAMs into a positive control, which I think is an animal model. And you need to go back to the human systems. And I think all three of those should be considered gold standard.
Brenda, Carolina, feel free to jump in as well.
I completely agree with Michael. I think, that we cannot exclude one versus the other. We need to do the integration to be able to compare.
And I think I'll just add a little plug for that. I haven't had interaction with them, and I'm kind of curious what the other panel members think, but where are those standards ultimately amassed? This is the standards coordinating body that's been set up working. What's missing from that? It seems like everyone right now is developing their own standards, publishing it in some cases, but maybe not in others. And I want to know, I think that, I would like to start to interact with the standards coordinating body, but I'm curious to hear others' thoughts.
Yeah, but I would say it's very specific to the question that you're answering. So some things are easier to standardize than others. And I think, as a clinician who understands that, I'm operating on a baby today, if we're studying that baby in the lab, if it doesn't rhyme, it doesn't rhyme. And I feel like, you need to bring multidisciplinary groups together. And so the translation of what you're doing is very different when you're trying to study something and prove a mechanism for a publication versus developing a one-off. And human-to-human variation is very difficult to write a grant against, because it's very confusing when you have different results from two different lines to know, is that biologically relevant or not, but without a positive control. And it frustrates the PhD, very focused person who's looking at that difference is thinking it's artifact. And then they have Matrigel and other things that are completely making their system not reproducible, and they don't understand, is it the line, the cell, or other. And I think, you can do that as a standardization, but I think some of it is just training people to start seeing the questions differently than what many of us have been trained to do. And it takes support of providing things to allow that new generation to think differently. And it's not going to happen in a year or two. And it's not going to be recognized in study sections because those are the people that understand it. It's going to take us a decade to put this into place, and there's going to be a little bit of wins along the way. And resources that support people to use more than one line and stuff is, at least that's been my mantra for the last decade, is it happens much slower than I wanted to, but it is happening.
Yeah, I want to bring out two of the things you just said. One is more funding to do multiple lines, to truly evaluate multiple lines is not sexy, but it's very expensive. And then the second thing you brought out is that there's sort of at least two categories. One is sort of the trying to mimic the person you took the cells from. That's sort of one group of or type of studies. And the other is sort of developing a cohort of lines that represent, and there are all kinds of variations in the population, but let's just say, male and female, whatever. That piece, I think, can start to be standardized. The piece where you're trying to match to a person and a personal response is going to be challenging and more of a in your own research lab kind of expedition.
I think one point that you mentioned, Brenda, and also Michael mentioned, is to bring those things together, right? Because there is a lot of standardization being working in different places, and it is really important that we bring them all together because, the majority should be overlapping overall in terms of the criteria, at least for that. So I think, centralize that and bringing all of the community together, it is a key here to speed it up the standardization and guidelines, at least.
Excellent discussion. I'm going to move us on to the third question. I'm going to bring Scott up to bat. I'm a Phillies fan, Scott. So I'm going to put the top question, third number here in the third spot here. So how can biological materials be developed, characterized, and stewarded to better support both NAMs and animal models as reliable, fit-for-purpose research tools? Please, Scott.
Well, I think in the human models, I mean, in the context of cancer, there's sort of a straightforward process. If you work with where you here, what we do is we monitor the OR records. We look for the type of cancers that are coming out. We can often even know the genetics of what that cancer might be because the test would already have been done, and have a pipeline in which the material can either go into a mouse or into an organoid culture or into a that I think is straightforward. I don't have enough experience with other pathologies, but I mean, when I've wanted to get normal, it's always been been very difficult. I think in terms of just related to the quality control of all of this, I think it is a really big challenge. We were just talking about this, the variation that is in biology, ranging from, particularly once you start to incorporate human systems, is enormous. And I think we don't have a good sense for, how many of a model we use or how many we don't. Again, I can speak for something that in cancer has been really powerful for these systems, which is where we know the genetic alteration that's a driver, and one can have cancers that either have that driver or don't, and use a drug that targets that driver, and then see that there's a pattern of response in one genotype and not the other. And that, I think, is satisfying. It's a great, illustrator of success. But I think as you move beyond those really defined questions, the noise becomes, much more challenging. And so I think it is going to be hard to create, reproducible human-based systems, across, institutes or whatever. I think, SOPs is, I think, the best we can do is having standard procedures for how, we go ahead, isolate material and use it is going to be the most important, and maybe some guidelines for the N that one needs, to develop conclusions.
Thank you, Scott, very much. Any of the other speakers, Michael, Brenda, Carol, want to jump on this?
I guess I would only add the observation that we've learned by making GMP versus research grade is that the reagents over the last decade have changed, and that they're becoming pure, and you get a different readout without a way of looking for the biology of an FGF or a WNT or whatever you want to look at how it's processing. So it's evolving, and there isn't easy readouts to those. And when we talked about thyroid hormone and stuff earlier this morning, the things that are influenced in serum and whatnot that is different across that we don't really understand plays a huge biology in the way our readouts are. And to develop toolboxes to look at that isn't sexy. Who's paying for you to look at that? Because no reviewer cares about that. But it makes a huge biological difference when you try to freeze down and do something. The progeny that is the reproductive progeny are lost, but everything else is there. You think you're fine and you're not. Where do those standards come in play, and who's going to pay for them? And I've always said I thought the world is going to want these readouts, and what the readouts first are drug toxicities and stuff to tell drug companies to stop, the word no. And so it's not exciting to them to develop something that puts them at a stop, don't invest more money, versus yes, go ahead and develop this drug because you're going to have a new therapy. And so you need the world to have a yes to move this forward. And I thought the research world was going to move it forward, but I realize now it's the other end of clinical applications that is going to be the driver of this unless we change the way things are funded.
Tom, we have a question in the chat. Should I go forward with that?
Please, yeah.
All right, so it has to do with describing precisely what is meant by validation of a NAM. What are minimal characteristics, for example, functional, anatomic organization, sex, age, etc., to demonstrate, measure, and observe? Or what are the characteristics that are required to validate a NAM?
I mean, we've sort of started to talk about this and continue to. I mean, I think for every model system that's been developed, people have also developed in tandem, the basic functional characteristics that NAM has, and they've defined it on their own and or worked with standards coordinating body. But as Michael was just saying, it's like, I think that going forward, that it's going to be very device or drug category specific. Like this NAM can be used for this, and it will have these characteristics. And then, for example, the FDA will say, yes, that will count in your IND application, and it will be I think it will be very specific, become very specific, like this thing, and it's got to look exactly like this. But for general studies and the way we use it in research, those have been and are being defined. That's just the way I see it. I see it evolving in sort of two different realms. One is like very clinical and applied, and the other one is very research.
Yeah, I completely I agree with Olga, with Brenda. I think this has everything with the last question here, that the challenge for the validation is really dependent on the context of use. An NAM that is used for discovery might only need to show biological relevance or reproducibility, utility for prioritization of the mechanism or candidates. But for translational or regulatory use, the bar is going to be very high with more defined performance criteria, acceptance threshold, cross-site reproducibility, and documentations, and all of the limitations, right? And there is the variability across the platforms as well. So I think the question here for validation is really going to depend, what is your question? What you are trying to validate for what? Right? And it is where you should start.
Any other thoughts on that question?
Maybe just one more thought, in that I do think there's good news, and maybe that I just spent a sabbatical analyzing omics data and trying to harness some of the data sets that we already have, knowing that also prices are going down just in terms of our ability to sequence individual cells. And I think that plus AI is going to make it really powerful for us to be able to quality control our tissues. We've been setting up maturation indices and metrics that we can map the cells of our engineered tissues onto to know, exactly what, or ish, post-week conception our tissues are from a maturation standpoint. Anyway, I think there's a lot of exciting things coming up where we can do a pretty good job, a thorough job of quality control in the future, at least at the, transcript and protein level, how the functional unit, that's sort of something we're going to continue to address, but I'll just say that's the good news. And then just another plug for, and we're going to need more money for that. So those kinds of unsexy studies.
Thank you, Brenda, for going with the good news there. I always like hearing the challenges and the good news on the horizon as well.
So we have one more question, or sorry, did I interrupt someone?
I was just going to add, I think one of the learnings from having an automated system in the liver is that what was very dependent on very highly skilled individuals became something anyone can do. And the ability to take the product that normally takes 20, 28 days and freeze it down with a readout that's very addressable and AI driven demonstrates that you improve your outcomes and your reliability is high. So the ability to automate something and give you a readout is I think, going to be one of the ways to validate outcomes. Although, unfortunately, I don't think people can all afford an automated system. It does demonstrate that it's not user dependent. It's standardized and reproducible across a network that anyone should be able to do. And I think that I think will eventually be the gold standard.
Just to build on that, I think, it is very relevant what we are talking here about the standardization, the automation. Brenda just mentioned, the data generated from omics, proteomics, but it's very important that we have in mind how we are going to be sharing this information across our sites, because that is going to be really impactful in terms of a validation of demands or how a model can be used for Y versus Y and X and Z, the selection of a model, it is not easy to make.
All right, well, that brings us to 2:45, which is the end of this session. Obviously, to speak for Tom as well, thank you very much for speaking and sharing your thoughts during the discussion. Really appreciate it.
Thank you so much to everyone. We have a break. Please be back at 2:55. Thank you.
Thank you.
Welcome back. We are moving to our third session with the title Building Comparative Databases and Tools to Support Benchmarking and Model Selection, Promoting Predictivity and Reproducibility through Interoperable Data Standards and Platforms. We have two co-chairs, Christine Heppel and Elias Tarantal. So Christine is a program officer in the Office of Special Initiatives in NCATS, where she serves as a subject matter expert in NAMS. She is the NCATS coordinator for the NIH Common Fund Complement Animal Research and Experimentation Program and contributes to several other Common Fund efforts. Elias is a professor and vice chair for research in the Department of Pediatrics, with a joint appointment in the Department of Cell Biology and Human Anatomy, School of Medicine, and unit leader in multimodal imaging core lead at the University of California, Davis. She directs the Translational Human Pluripotent Stem Cell Research Facility. Her research program focuses on gene therapy, somatic cell genome editing, and stem cell transplantation, perinatal onset of inherited and acquired diseases, and innovative applications of translational liver imaging. Please take the microphone.
Great, thank you. So the learning objectives for session 3 today will be to understand how comparative databases can improve model selection and translational relevance, to understand the importance of interoperable data standards for integrating diverse model systems, to identify key features of platforms that support reproducible and transparent research, to identify the role of fair data principles in NAM advancement and recognize how shared data resources can accelerate discovery and reduce redundant research. And finally, to recognize that sustained investment in data infrastructure and curation is as important as an investment in experimental model development. So for session 3 today, we will have 4 speakers. We will hold questions until all speakers have had a chance to present their work, and then we'll have a panel discussion as with the previous sessions. So our first speaker for session 3, and I apologize, hopefully I'm getting your name right, is Dr. Murat Kirkid. He's at Javelin Biotech. Murat is a bioengineer and co-founder and CEO of Javelin Biotech, which is focused on new approach methodologies, integrating microphysiological systems or organ chips, tissue chips, with computational modeling to improve preclinical drug testing, predictive accuracy. So with that, I will let you go ahead with the presentation. Thank you for being here.
Thank you for the invitation. For my screen, hope you guys can see the full screen. Slides are advancing.
Yes, looks good.
Perfect. So again, thank you for the invitation. Happy to be here. Today I'm going to talk about our developer perspective, how to improve the reproducibility and predictability of the organ and chip technologies, and also how we're assessing the biological function, how we can compare certain application and context of users from in vitro to in vivo. Just to start with the motivation while we are doing that, and a specific way for our platform, why we are designing in a certain way. So when we look at the journey of a drug, right, when a new compound seen the human biology, it's very limited at the earlier drug discovery stages. It increases during the clinical stage, and once after the approval, the real human diversity and the drug comes to market. And we see along the way many challenges, clinical failures and post-market withdrawals. So we envision to bring in a continuum to drug discovery from the late optimization and the preclinical stage, and really improving the mechanistic understanding of the drugs, avoiding the interspecies variability. So with that, our focus was the generating a technology initially to overcome the challenges commonly being discussed in the field. I don't have much time to go one by one on those details, but here I would like to explain on this video what type of improvement has been done. So first of all, removing the PDMS and going to more robust thermoplastic improves a lot of the manufacturing challenges. And with that, we were able to build a physiologically relevant technology where we have the liver technology being perfused throughout the system. Incorporating the ease of use principles and the onboard pumping enables the robust operations, and also bringing the oxygenation to the system, extending the culture period over 30 days. So bringing really physiological, continuing physiological functions. In the same design, we were able to add the drug addition and sample collection in the same design to reduce reproducibility, handling reproducibility. And with that, we can run long-term studies, understand the long-term effects of the drugs. And the same platform enables the liver connecting with the second organ, asking different applications that will be important from drug discovery perspective. And a unique way we are approaching this is we are not a high throughput system. We are developing a high content system. From a single tissue, from a single chip, we generate over thousands of data points in a month-long study. And with that, we enabled to bring in the physiological relevance. We were able to bring that without using any membrane or scaffolding type of technologies. With that, the flow is similar to physiological rates and the oxygenation profiles, and the drugs are interacting with the MPS, with the tissue, vascular first, and going to space of tissue and reaching to hepatocytes. With that, we can generate a really high content information, collecting samples from the media, understanding the simultaneously how drugs are metabolized, disappeared, new metabolite formation, multiple clinical toxicity biomarkers that's been evaluated also in the clinic. So it will be a direct comparison of those biomarkers, and also diverse set of clinical biomarkers related to PD and PK effects. We support that image-based analysis as well, so you will be getting more mechanistic understanding of the drug, the changes on the morphology, changes on the physiology, both biomarker level and the drug level. And at the end of the experiment, omics analysis also getting a tissue level understanding, cellular level understanding. And we apply this to variety of applications, advanced modalities, vector characterization, drug delivery systems, and the small molecule testing. So I will show brief examples of how we are validating, qualifying applications for these. Let's go through the biology. The most important step is the evaluation of the biology. And here we are showing our structure, cytostructure of the liver physiology, where we can get the hepatocyte cells, stellate cells in a gel-embedded format with the L-sex in the system. We can maintain that over 3 weeks in the system, giving us a period that understanding the chronic effects of the drugs. And we evaluate the system with known parameters, liver-specific parameters. Here is the albumin or CYP activity. And also we're looking this technology for more advanced application. We characterize for the receptor expression that's relevant to drug delivery systems like the LMPs, or the liver-targeted drug systems like GalNAc-associated oligonucleotide-based therapeutics. I will give some snapshots of the evaluation process, how we're doing that. And this is from our FDA iStent application. So when we get the donor, characterization is really important, and maintain looking at the functionality. So we look at the multi-parametric characterization, looking at the morphology, sustaining that over 28 days. We can look at the liver-specific functions, the albumin output or urea output from the system, how it's been maintained throughout the system. We can look at more specific metabolic activities, CYP activity over time, and looking at the different CYP isoforms, how they are maintained in the system. And also, we add this, we repeat that with multiple donors. We don't just pick a single donor, because we work with the primary cells. The donors will change over time, and we are qualifying the donors according to our procedures, and look at that, where will be the reliable cell sources with that. Another area is why we need human models, what type of improvement we're bringing to the system. So this is a bioinformatics workflow we recently published. In this case, we look at 200 liver biopsies and look at the interdonor variability. And here, that's what we're plugging here, the interdonor variability out of 200 donors. And we can compare that with the mouse, rat, and monkey liver transcriptomic samples and create a pairwise calculation. In this case, the transcriptomic similarity is around 50% compared to human. We can expand that to the in vitro models. Here's the primary human cell models as the highest in vitro model and comparable to the animal models. And later on, when we apply our system, a perfused liver MPS, we're able to get within the human variability with our donor, and also we're able to maintain that. But these are additional points that we are improving the functionality of the tissues and also human relevancy. We're expanding this with the disease models right now, identifying our media formulations and looking at the disease pathway similarities between human liver and liver tissue. Several applications we have published with this to understand the in vitro, in vivo correlation of certain applications. In this particular one, we look at the hepatic clearance of the small molecule drugs. And here is a very traditional plot showing the in vitro, in vivo correlation. Here's our predicted values and the clinical observed values. So for most of the cases, we are on the unity line, showing the perfect predictions. And we compare this with the animal rat models. Actually, we were much better predictions than the animal rat models. So we're providing a really unique platform, predictive platform from to getting translational parameters for PK. Later on, we expand this work to a multi-organ system connecting to the liver system with kidney and muscle, and this is a recently published work. In this case, we were looking for more complex processes using the in vitro technologies to predict clinical outcomes. In this case, we focus on the IV drugs to understand three critical PK parameters: hepatic clearance, renal clearance, and volume of distribution. And in this particular example, we test about 15 different small molecule drugs, and we assess the in vitro, in vivo correlation, all these parameters. Later on, using these parameters, we can use the traditional PBPK models to correlate that to the clinical findings. So in this particular case, we were showing the concentration time profiles of the drugs, how the plasma concentration will be, and also we predict the exposure of the mixed clearance drugs. So these are the few examples that in the pharmacokinetics work, how we can evaluate a system and compare directly with the clinical findings, because it's more quantitative. Either AUC or the half-life can be compared with that. But when we move to the toxicology type of applications, this is not that quantitative. It's more mechanistic based quantification. And in 2 slides, I want to highlight our application on this one. This particular case, we use mipomersen. It's an antisense oligonucleotide-based drug, approved but withdrawn from the market due to the liver toxicity. And during the clinical trial, preclinical development, because of the human specificity, the surrogate compound is being used with the animal models and no animal toxicity being observed. And that's one of the reasons. When the compound go to the clinic, early indications of the toxicity was there, but later on the compound goes to a larger population, the steatosis become the reason this drug being withdrawn from the market. So that was our case study, whether we can understand this with our complex liver model. After a 3-week exposure of this ASO drug, we're able to show the dose-dependent steatosis in our system. We can complement that looking at the liver function. So in this particular case, we look at the albumin reduction. We can see both doses in our system, the albumin being reduced, indicating the liver toxicity. We compare this with the spirit models. Even we go to the higher doses, we were not able to see these differences. And also collecting the data and high content data, we get insights from that, the mechanism of action of this toxicity. So here, after the second dose addition of the drug, low dose of the ASO drug, we can see the increase on inflammation and leading also ALT increase, as we see in the clinical trials. So this was the examples about the oligonucleotide-based therapeutics. Another example is the human-specific, organ-specific drug delivery systems. In this particular case, the challenge was to look in multiple AAV vectors to identify which one will be better transduction efficiency on the liver and less toxicity. And currently, this workflow is heavily dependent on the NHP studies, and we are working towards improving this existing workflow. Particular example, in a blinded study, we look at the 2 different AAVs, 3 different AAVs. I'm showing you a few of the results. You can see that better transduction of the system in day 7. We can remove from the AAV. We can see the persistence of the AAV transduction efficiency in the system. And also, when we look at that clinical findings, we can rank order these AAVs for their efficiency, liver efficiency, and compare that with the clinical findings. And going into the details a little bit on that, is the rodent models was not able to get the right rank ordering. And also NHP models, one of the species are derived from the NHP models, was not reactive to that. So using the human model, we are able to rank order these compounds from better than rodent models and the NHP models. So these are the few examples of how we are comparing the the quality predictability of application with human findings and some cases on the animal findings. And another important part for us is being a commercial organization and enabling these technologies to our customers. We have a lot of rigor to give the technologies in a robust way that our customers can use that. And we have many different procedures being incorporated here, from the manufacturing QC to MPS qualification of the primary human cells, that we look at and use this in the improved SOPs. We train our customers, and also for their applications, we evaluate the systems very carefully. So here I want to demonstrate one study, inter-company study that we performed with our system. 3 different companies being trained by us, first-time MPS users, and they run a blinded study, 2 different compounds. And here the results that our CV was very low in company-to-company predictions was really well, and you can see the chip-to-chip variability from the error bars. So overall, these are the rigors that we've been taking to developing a technology, evaluating MPS's applications, and then ensuring that for our customers. That I'll stop my presentation. I believe questions will be afterwards. Thank you.
Great. Thank you so much for the great talk. All right, we're going to move to our next speaker, which is Dr. Melissa Hendel. Dr. Hendel is a leader in precision health and translational informatics at the University of North Carolina at Chapel Hill, where she serves as director, and Sarah Graham Keenan, distinguished professor in genetics. Dr. Hendel's research emphasizes rare disease diagnostics, genotype-phenotype integration, knowledge graphs, and data standards. And with that, I will pass it over for your presentation.
Okay, let's see. I'll show. Are you all able to see my screen?
Yes, looks great.
Excellent. I'll just get that away. Terrific. Well, thanks to Dr. Happel and Dr. Tarantal for including me in this session. I'm really delighted to be here today. These slides can be found at that link for anyone who's interested in any of the links that I'm presenting. And I just wanted to mention that I was asked to speak specifically about our work in the Monarch Initiative program, which is an ORIP-funded program that develops a number of different data standards and semantic tools for data harmonization. And some of the examples that I have really have used similar technologies for mechanism discovery for rare disease diagnostics, as well as for drug repurposing. And here we're most recently developing some resources that we think may be useful for the NAM community. Let's see. So just to start, our biology central dogma is, typically that genes plus our environment over time results in subsequent phenotypic features or traits. But as we know in mechanistic biology and in our new approach methodologies, we're really modeling a much deeper, richer collection of associations. And it's the standards really need to be up to the task of inventorying and comparing many of these different kinds of relationships in order for us to understand what is the regulatory context and decision-making that we might be able to best leverage these new approach methodologies. So one of the problems is that in the NAM field, the information about NAMs are very scattered, and we don't really have holistic standards that we can use to harmonize the data in the way that we have historically for other mechanistic discovery types of projects. And so we've been chipping away at some of this. It's still early days, but we hope that the work that we've been doing is useful to think about and could be advanced in the context of our work together. So for example, we have lots of different kinds of NAMs shown there on the left. There are different assays, different protocols done by different labs. There are different terminologies, and the sources and data and the format of those data are vary enormously, more so than almost any other disease modeling context. And the technologies at the same time to realize, and we've just seen some wonderful examples from the Javelin platform. are really just advancing more rapidly than the standards to help inventory and compute over those in the ways that we've historically done for those other use cases. So we don't really have a comprehensive view of what NAMs are available. We don't know which aspects of diseases are actually being modeled and what those endpoints are. And we don't have a good visibility into what the unmet needs are for which NAMs should be developed and how will they complement and be synergistic with existing or complementary disease models, whether those be whole organism or NAMs. And therefore, we have fragmented data landscape and inconsistent methods that limit our confidence, comparability, and progress towards using NAMs for regulatory advancements. So we have uncertainty, inconsistent data and methods reduce our confidence in the NAM evidence. We have knowledge gaps. Missing information prevents understanding of each individual NAM's capabilities and its limitations. And we have regulatory barriers that limit our ability to use NAMs for regulation and decision-making. So ontology, schema, and knowledge graphs. Why we need a standard schema for NAMs. And I'm going to just a prelude, discuss later some efforts that we have to develop a NAM schema that kind of helps bring that much of the NAM data together in a way that we can compute on and help generate evidence. So first of all, we need to represent the phenotypic features and physiological characteristics that allow us to align and compare across models, across space, and across time. So spatiotemporal disease modeling. We need to inventory the biological processes that are being modeled. We need to make sure that the data is fair and AI ready so we can help our agents do more work for us. And then to understand which NAMs best recapitulate specific disease attributes and what evidence is actually lacking so that we invest in the NAMs that we most need. So this is just an example of the types of technologies that we've been able to leverage historically within the Monarch Initiative program. Here we've characterized phenotypic features across different species. Here shown are human on the right and mouse on the left. And by using semantic, good old-fashioned AI, this predated the LLM revolution, we were actually able to reveal mechanistic underpinnings of diseases for which we did not know the molecular mechanisms. And so this is just a good analogous thing that we might consider for how we might take a similar approach, but now in the age of LLMs, to reveal underlying mechanisms that the diseases that are being modeled, so that we can model those in the NAM and develop the evidence we need for regulatory decision making. So wanted to highlight a few relevant standards that are included in the NAMO work, which I'll come to in a minute. The first is the Uberon ontology. The Uberon anatomy ontology also includes the cell ontology, and it's a long-standing community effort to integrate numerous anatomy and cell ontologies, and it's used in a very large number of programs for standardizing anatomical content in programs such as ENCODE or HubMap or the New Whole Person Initiative, and it underpins all of the anatomy within the gene ontology. So Uberon is particularly relevant for spatiotemporal validation considerations. So for NAMs, we need to be able to model the biology in a spatiotemporal way that exists within a cell, so subcellular morphology and parts, process kinetics, the cell type level, cellular morphology, omic signatures of individual cells, states of differentiation, cellular neighborhoods, intercellular morphology and connectivity, and cell signaling, molecular localization, tissue organization, structural integrity and function, and we saw some great examples of that in the Javelin platform. Temporal changes, and then at the organ level, signaling, electrophysiology, and all the way up to the whole organism. So all spatiotemporal levels of NAM characterization should be relatable to human physiology, and all of these things are candidates for standardization. And importantly, if we structure the content well, are really great subjects for AI methods development. So the monarch knowledge graph is a graph that it contains Uberon in the cell ontology, but it contains many other things relating diseases and genes and phenotypic features. So just very simply, there's a lot more content in the monarch knowledge graph. We use it again for diagnostics as well as for drug repurposing. Here we're leveraging it for our characterization of NAMs, but it really combines information about gene function, gene dysfunction, and all of that anatomical classification that I mentioned earlier. And it's also the basis for an AI effort that we've been undertaking called DISMEC, which I'll talk about next, which is essentially an AI-generated knowledge base that sort of has been created on top of that monarch graph and all the ontologies and data sources that monarch integrates, with a structured evidence representation so that everything is fully provenanced. And it contains a collection of computable mechanistic hypotheses in the form of causal graphs of pathophysiology for now over 1,000 diseases, and essentially has evidence packets for mechanisms. And we think this is a really interesting way of thinking about how we model NAMs and how we might collect evidence for the degree of corroboration for the diseases that are being modeled in an individual NAM. NAMO is a component of DISMEC, and in that sense, what it does is it's a schema that underpins a component of DISMEC, which is used for lots of different things. But the part of DISMEC that relates to NAMs, we call NAMO, or the new approach methodologies, ontologies. And it's a schema that characterizes and classifies different aspects of different types of NAMs. So we have here just an example showing very simple taxonomy on the left for some of the popular NAM model types. Here, an organoid is broken out to having specific characteristics that can be collected in a specified manner. And then there's an example representation of a specific Alzheimer's disease brain organoid there on the right. And what you can see is it's almost like a standardized mechanism for pre-registering characteristics of your NAM akin to pre-registering a clinical trial for clinicaltrials.gov. And the schema integrates all of the existing standards that we've been able to identify, and we would certainly welcome more from the community to try to make it easier for users to automatically create the templates they need for this type of reporting that can go alongside other data sharing activities around their NAMs and allow the AI agents downstream to actually leverage that content to the best of our ability for examining degree of corroboration and evidence evaluation and the like. So here's a NAM ontology in action. This is modeling that a gut organoid. So you can see terms from the Uberon ontology for the small intestine. There's a NAMO term for a human gut organoid. It has specific characteristics that associate that organoid with a specific function, intestinal barrier function, which has been implicated in a specific disease here from the Mondo ontology, inflammatory bowel disease. And it connects using AI agents to experimental data and NAM omics data. And it also includes information about the cell types by which it was derived, with specific characteristics about its culture and its physiological measurements. And so this is just a small snippet to kind of give you a sense of how the data modeling can support standardization across different labs and across different NAM systems. So this is using DISMEC now. So back to our sort of AI constructed causal graph library. And this is just showing an example of a snippet of that whole causal graph here for CFTR dysfunction. And here what we're trying to understand is how do we model diseases as mechanistic causal graphs? And as I mentioned, DISMEC is used for a variety of different purposes, but here we can use NAMs to demonstrate where we have orthogonal or overlapping recapitulation of disease physiological features at different levels of anatomical granularity. And that collectively, all of the disease modeling together can demonstrate the evidence that is needed for translational implications or regulatory approval. and help us automatically derive standards and algorithmic metrics of concordance. And so that's one of the things we've been working on is how do we sort of algorithmically compute what is the degree of concordance across NAMs. And so here's an example. This is an example brought to me by my colleague Adra Bhatt at UNC. And this is a human two-dimensional monolayer where she's studying chemotherapy-induced diarrhea. And so you can see the model there in the middle. The drug is applied apically, and so that mimics luminal exposure. She is looking at barrier defects on the left that are measured with tear, reporter leak, microscopy. And then on the right, she's looking at epithelial injury measured with cell viability, apoptosis assays, and microscopy. All of the different individual factors that are going into creation of this NAM are documented. The cells here are derived from intestinal tissue from human healthy donors. And you can go and look at the DISMEC entry for chemotherapy-induced diarrhea to kind of look at what that causal modeling looks like there at the bottom. And so I just wanted to highlight what we think kind of what a standard for documenting or a recipe for documenting NAMs for potential future regulatory context or decision making. And so the first kind of requirement is defining the context of use. What kinds of decisions are being made? Are we developing a therapy? Are we trying to argue for a phase 1 trial? Et cetera, et cetera. And so here she is defining the context of use as a chemotherapy-induced diarrhea. It causes treatment failure, impedes drug development, is difficult to predict. It's only evident in human trials, it's variable, and it's impossible to predict so far. So trying to understand, how do we leverage this NAM in the context of chemotherapies that might be going to trial. So then we specify the biological context. What is the spatiotemporal representation? What part of human biology are we modeling? What is the perturbation? Specifically here, it's chemotherapies that are metabolized by the gut microbiota. And we choose the specific endpoints. Here, barrier permeability measures such as tear and reporter leak, epithelial injury such as cytotoxicity, viability, junction protein expression. And we plan for comparators. What do we know about the known incidence of diarrhea for a given chemotherapy? We can look at reports such as post-market reports or the CIDR database and those kinds of things. And I have a recipe for this at the end for those that are interested in the extra slides that sort of suggests towards a standard for how we actually document the kinds of things that are needed for NAM to be part of the ecosystem of comparisons and evidence generation. So I wanted to mention that, we really believe that combining NAMs helps us form a complete picture, that batteries of NAMs and batteries of disease models beat single models. And we even saw it in the prior talk on the Javelin platform in the comparisons with whole organisms, with human-derived samples, and with, physiological modeling, that together we can use NAMs to our greatest effect. So we can just for example, use an organoid to discover what changes, use a chip to test how it behaves under real forces and flows, and use physiological models to ask what human exposure makes that happen. Similarly, we should be looking for the same signal across layers. So for example, goblet surge to barrier drop to predicted airway doses. This is decision grade evidence, but for each, we need to state the question that it's actually answering. What is the endpoint and what is the threshold that moves us towards a decision? And so I think, again, it's all about standardizing and structuring the data so that we can combine data coming from different models together and looking for those same signals represented in different ways across those different models. And then finally, just use layers for what they're good at. Organoids for cell state or lineage fidelity, or organ on a chip for function under physics, or in silico models for translate such chip or organoid results into human dosing. And then finally, again, cross-validating signals across model before in vivo escalation or phase 1 trials. So why are reporting and standards worth the investment? This interoperability really matters. We need comparability, the ability to do meta-analyses. We need our data that we're generating from these NAMs to be much more AI ready than we're at right now. There are no nomenclature committees or model organism databases that are going to do this for us. It really has to be investigator provided. And we also just, for rigor and reproducibility purposes, need to be able to present just minimal metadata in that sort of pre-registration context so that we're evaluating what we think we're evaluating for the reasons we think we're evaluating it. And that leads to provenance, provenance, provenance. How did you make the model? How did you evaluate it? What was the replication? What are the limitations? For example, if there's no immune compartment or no cyclic stretch. AI can help. Instruction-tuned LLM extracts from papers and protocols into that NAMO LinkML schema that I mentioned earlier can allow us to create comparable AI-ready NAM cards. So we propose the idea of a structured interoperable NAM card, which would then allow us to regularly iterate and generate that evidence-based concordance information and inform policy. And so together, all of these resources that I've mentioned, Uberon and the NAMO schema, the Monarch DISMEC and knowledge graph, and alongside AI can really enable evidence-based model selection, where we match the biology and the throughput and cost. We can reduce animal use and come up with a validation framework that is really what we've been calling a structured concordance result or metric, which can be aligned to the FDA Modernization Act 2.0 and the EPA NAM work plan. So I wanted to briefly mention that we have held several workshops together with the Triangle CERC and the FDA. And I just wanted to provide those links there where we examine in much more greater detail surrogate endpoints using NAMs for rare diseases and just other characteristics and examples that I didn't have time to go into today. Wanted to thank the great team as well as the, in particular, Office of Director ORIP program that has funded the Monarch Initiative. Thanks very much.
Thank you for the wonderful presentation. We are going to move right along to our next speaker for this session. Our next speaker is Dr. Jason Spence from the University of Michigan Medical School. Dr. Spence is the H. Marvin Pollard Professor of Gastrointestinal Sciences at the University of Michigan Medical Center, with appointments in internal medicine, cell and developmental biology, and biomedical engineering. His lab pioneered methods to generate 3D human lung and intestinal organoids from pluripotent stem cells and expanded these approaches to liver, esophagus, and stomach tissues. And his talk today is entitled From Atlas to Benchmark, the infrastructure required to compare organoids and NAMs to human tissue. That I will hand it over.
Great. Thanks very much for the invitation to come and present and for that introduction. So hopefully my talk will complement Melissa's, which I thought was fantastic, but from a completely different perspective. As somebody who is really involved in creating new NAMs models. And just before I get going, I wanted to give a plug to the University of Michigan Human Organoid Core that I direct. So we have a core that has access to about 300 different human organoid lines across the primarily GI tract. We have media experimental workflows, and we're starting a new program to provide access to matched patient tissues to the organoids. And so feel free to reach out if you're in need of organoids. Okay. Disclosure as well that I have developed IPS-based human intestinal organoid model and spun a company out with a couple of co-founders from my lab. The work I'm going to talk about today is unrelated to the company, however. Okay, so sort of the premise of my talk for today, as somebody who I said is involved in really creating new organoid models or NAMs models, is that I sort of wanted to go through the workflow that we use to develop these new tools. And once they're developed, how do we describe the cellular complexity of these models? And then how do we demonstrate that they can recapitulate some aspects of human biology? So what is our approach to the validation and benchmarking? And where do we find challenges and difficulties? Okay, so the work that I'm going to talk about today focuses on lung organoids. And we heard from Dr. Ingber this morning about this minimal alveolar unit in the distal lung where you have stem cells or alveolar, which are known as alveolar type 2 cells or AT2 cells, which produce surfactants, but they can also give rise to the alveolar type 1 cells, which are these long, thin cells that sit just juxtaposed to capillaries where gas exchange takes place. And so over the course of several years, my lab has developed methods, both from iPS cells and from primary human tissue, to isolate or differentiate progenitor cells that we can then subsequently differentiate into alveolar type 2 cells, which then can be used to give rise to alveolar type 1 cells. And so what I'm showing you are sections and some molecular markers for each of these major cell types, differentiating as organoid within the organoids in the tissue culture dish, with the caveat that I'm not going to talk about it all today, that all of these models are grown in Matrigel, and we've heard some challenges and limitations with both the models, the Matrigel, and the media that they're grown in. Okay, so the steps that we take then to benchmark these tissues is really what I want to talk about today. And so for this particular project, we actually obtained, because we know that these are, as you've heard, these are more immature cell types, and we actually think these are more akin to pediatric alveolar cells. And so what we wanted to do is really benchmark this against the best reference atlas. And so we created a pediatric reference atlas. So we needed to obtain tissue with the appropriate clinical context. We then created the reference atlas. In this case, we used a couple of different modalities: single-cell RNA sequencing, multi-omics sequencing, spatial transcriptomics. From that, we developed a queryable benchmark that we can use as a comparator to score NAMs against that human reference. And then for us, what we really hope we can do with these databases, especially from the primary tissue, is make novel observations that we can test, that we can use to develop hypotheses that we can test in organoid models. And we hope that we can then learn something from the organoids that applies back to the patient. Okay, so this study is available as a preprint on bioRxiv, so you see the reference up here. But essentially what we did was obtained tissue from donor lungs, 23 different donors, 0 to 2 years of age, histologically normal lung. We had limited data from the EMR. We performed a couple of different modalities of sequencing, as I mentioned. And then what we wanted to do is build this reference atlas to be able to compare data from diseased lungs, which I'm not going to talk about, but then also from patient-derived organoids with and without perturbations. Of course, one of the first challenges is getting access to patient tissue. And this required us to connect with a collaborator of mine, Gail Deutsch at Seattle Children's Hospital. And Gail really had a nice workflow or pipeline of collecting tissue from autopsy or surgical residual material that was properly consented. She then did expert pathological review to identify it as normal. She was able to then also provide us with limited data from the electronic medical records, and then was able to send it to our research team. So this is what the data looks like. What we've done here is taken all of the data and subsetted it to just look at the epithelium. This is the single nucleus RNA sequencing. And then I've subsetted these specialized cells, epithelial cells of the alveolus further, so that we're just looking at the alveolar type 2 cells and the alveolar type 1 cells. And what I'm showing you here are the computational clustering that we identified, and we're calling these initially alveolar type 2-1 and type 2-2. So really two different flavors of the alveolar type 2 cells. And so through additional analysis that I don't have time to get into, we've essentially defined two different alveolar cell states within this tissue. What we're calling them are FMO5 positive, and there are large cohorts of genes that distinguish these two cell states, but we've identified a couple of markers that we use as sort of a reference or an anchor to label these. And so we call them an FMO5 positive state and a CFTR positive state. And these have genes associated with lipid metabolism, whereas the CFTR are more, have genes associated with inflammation, proliferation, things of that nature. Okay, so one of the first challenges that we ran into, and I think rightly was brought up by the reviewers of our manuscript, was what are these states and why haven't they been defined before? At this point, there are dozens and dozens of single-cell sequencing studies of the human lung, and I'm just showing you maybe one of the first or the first paper using single-cell sequencing to describe the normal human healthy lung. And in this paper, they described also two states of alveolar type 2 cells that they defined as AT2 and AT2S. And then a very recent paper within the last month or so that's been come out, where they've defined three different states of alveolar cells, AT2, AT2S, and AT2I. And it's really unclear how any of these cell states link to one another across these papers. And so I think this is, one of the challenges that we're faced with in terms of describing a tissue or cells in human tissue, but then ultimately linking that to the models that we're using in the dish. And this is confounded a little bit by the ontologies. In terms of alveolar type 2 cells, there's really only one in the cell ontology database. And so, I think it's important to think about that the cell type, which is the alveolar type 2 cell, is not equivalent to every cell state within that type. And so there's additional heterogeneity that we need to think about, and that new cell states may be identified based on the disease or the perturbation as we look at more and more contexts. Okay, another thing that we found that was really confounding our initial analysis was that the depth of data for these comparisons really matters. And so what I'm showing you here is some data that just came out of Maria Basil's lab at UPenn, where they also did sort of an atlas of the pediatric lung, and they had collected data and analyzed it and collected tissue and generated data using two different single cell sequencing chemistries. And so they used the 10x platform, and they did some sequencing using the version 2 chemistry and the version 3 chemistry. And so these are essentially just, the version 2 is an older technology, the version 3 is a newer technology and captures more mRNAs and is therefore more robust. And so what we really wanted to do, and this was again prompted by the reviewer critiques, why aren't these signatures, why haven't they been defined before? What we really wanted to do was then ask whether or not we could find the signatures from our data in other people's data. And this was an interesting use case because what I'm showing you here is that we've developed signature scores for the CFTR positive cells and the FMO5 positive cells, and we can plot them based on the rank of each individual cell. So the cells up here in the top left are highly scoring for the CFTR signature, and the ones in the bottom right are highly scoring for the FMO signature. And so what we found is that really we could find in this independent data set, we could find this axis of AT2 cells only in the version 3 data. In the older version 2 data, essentially that didn't that axis or this transcriptional signatures and these cell states were unidentifiable. And that's not because they weren't there. It's because we know that the feature depth or the number of genes in the version 2 data set was just too low to find the genes that are contained in our signature score. Okay, so we've used a similar scoring approach then to start comparing our signature with several of these other data sets. And so, it's, I guess, comforting to know that we can find these cell states in other people's data. And again, back to the data from Maura Solar's lab that was recently published and that I mentioned a few slides ago, what we were really able to show is that what he calls AT2 cells matches up very nicely with what we call FMO5 cells, and both the AT2I and AT2S match up quite nicely with what we're calling the CFTR positive cells. And so, I think in thinking about constructing these comparative databases and back to Melissa's talk about making all of this sort of machine readable and comparable, it would be really nice to think about how we do this across all of these diverse data sets. Also keeping in mind that there are these caveats that I'm talking about, like the depth of the data and the sort of age of the chemistries. Okay, so now that we've sort of convinced ourselves that we can identify these two different transcriptional states in the native human lung, the question is, can we recapitulate these states in our alveolar type 2 organoids? And so essentially what we did is a big screen of cytokines and growth factors and assessed genes that were enriched for these two different signatures. What I'm showing you here is just a limited number of genes that we assessed by qPCR that gave us an initial indication that IL-1 beta and TNF alpha are able to drive genes associated with the CFTR state, whereas dexamethasone a steroid was able to drive this FMO5 positive state. And this is really one of the coolest pieces of data, I think. We were able to go back to some of the limited metadata that we had from our patients and ask whether or not any of those patient lungs were treated with dexamethasone prior to going into surgery. And if yes, was there an abundance of these FMO5 positive cells? And what we found is that patients treated with dexamethasone prior to surgery had a much higher abundance of these FMO5 positive cells compared to those that weren't treated prior to surgery. And so this really gave us an indication that what we're seeing in the tissue culture dish might be reflected in the patient population and the treatments that they're receiving prior to surgery. Okay, so then what we did is went one step further and we asked, rather than just looking at a few genes by qPCR, what if we just do our same treatment groups and we do different doses and different timing, and then we do bulk sequencing and use that signature to score each of the cells from our in vivo data set. And what we can see is that with our organoids that were treated with IL-1 beta or TNF alpha, that's really driving this transcriptional signature towards these CFTR positive AT2 cells within the single cell reference data set. Okay, so just to wrap up, the approach really that we take is to generate a new NAM and then create data from that NAMS data set. We have, in this case, built a reference atlas, but I think this works, of course, with existing atlases, as long as the data that you're really looking for, I think, is present in that atlas. You can use the comparison with the reference data to annotate your NAMS data set. And then, of course, you can do perturbation experiments, which you can then compare back to the original NAMS data set or the reference atlas. You can do the same thing with disease data from in vivo lungs or presumably from genetic perturbation models in your organoids, although I didn't have time to talk about that today. And so with that, I'll just summarize by saying that this is kind of going over some of the infrastructure requirements for benchmarking NAMs and organoids, that you need your patient tissue and clinical context. This really depends on available data or individual collaborator relationships, ideally healthy and disease states with some limited amount of drug history will be available for your comparisons. From there, building a human reference atlas or using existing atlases, processed with annotated data that's available for comparing across data sets. This is actually one thing I think we could talk about as a community. And back to Melissa's talk, in what format should that data be, and how can we then really most easily compare one study to another? Data quality matters. Ontologies need to keep up with the evolving landscape and should be shareable and searchable. And then really, I think having maybe some standardized approach to benchmarking would also be useful as a sort of guideline for the community. So whether that's using signature gene sets and cell scoring, like I just talked about, or whether there's other methods that people really like and that give robust results. But I think some guidance about how we might want to benchmark in the case of sequencing data, as I was showing, and then ultimately, I think there's the final benchmark of showing function, which I didn't talk really at all about today. Okay, so with that, I'll stop, and thanks very much for your time.
Thank you so much for your talk. That was wonderful. And with that, I'm going to move along to our fourth and final speaker in this session. So our last speaker is Bridget Hill from General Dynamics Information Technology. So Bridget Hill is a data scientist at General Dynamics Information Technology. She's a contractor supporting the new NIH Office of Research Innovation, Validation, and Assessment, or ORIVA. And she spent the last 3 years supporting the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM. And she focuses on new approach methodologies as potential complements or alternatives to traditional in vivo methods. Her talk today is going to be building confidence through curated data, and open access interoperable computational tools to support NAMS-based chemical assessment. Thank you.
Hello, everybody. Can you hear me okay and see my slides?
Yes, they look great.
Awesome. So thank you so much for the opportunity to be here today. It's been so exciting to learn about all of these ongoing projects, and I am very excited to be here today to talk about data curation and exploring data through open access computational tools. So I am first going to provide a very broad overview of a publicly available data resource, the National Institute of Health's Integrated Chemical Environment, or NICE, as I'll refer to it for the duration of the presentation. I'm then going to talk about general data curation workflows and provide a specific example workflow for a in vitro high throughput screening data set. And then I'm going to talk about ways in which these data can be explored through open access computational tools with a focus on two tools that can generate human relevant predictions. So if you aren't familiar with NICE, this was first developed by the National Toxicology Program's Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, to support the development, the evaluation, and application of NAMs. It houses a diverse set of toxicological data, chemical information, and exposure predictions, in addition to a suite of computational tools that you can explore this information. And so taken all together, this can help identify data gaps and identify areas where a NAM can complement a traditional in vivo test method, for example. And so on the right here are some highlights of the releases of last year, things related to updates to chemical information, data sets, and actually allowing for users to upload their own information for select tools. And please be on the lookout for the next release targeted for this summer, which will include updates to reference and non-reference chemical lists, which can help explore these data, updates to cancer visualizations, and more parameters for the two computational methods that I'm going to talk about later on. And so here is a snapshot of the toxicologically relevant information in NICE. It is organized into toxicity endpoints of regulatory interest listed here on the left of this table. And the X just indicates if that specific data type is available for that endpoint. And as you can see, for the majority of these toxicity endpoints, there are at least two different data types available for exploration and download. In terms of chemical space, so there is a wide variety of chemical information available, such as physiochemical properties, chemical structures. Something that's new is chemical taxonomies, in addition to exposure predictions and also chemical use and functional use properties. So now I'm going to talk about just sort of general inclusion, curation, and processing of the data that are available in NICE. And this is really guided by the Interagency Coordinating Committee on the Validation of Alternative Methods in which NICETM supports. And data are sort of highlighted based on their usefulness to develop and evaluate NAMs. And so the first step is to retrieve data. So data are included from a variety of different sources, some of which are shown on this slide and can be anything from an official test guideline to a peer-reviewed publication. The next step is a subject matter expert review. So reviewing the actual data and the information provided in addition to the methods and how these data were obtained. And so there is an emphasis on including non-proprietary test methods, high quality experiments, using recognizable chemical identifiers, and in the instance of in silico methods, inclusion of an applicability domain assessment. The next step is these data go through a minor processing pipeline, and this is to align the information with data fair principles as much as possible, so ensuring things are findable, accessible, interoperable, and reusable. And some of the areas that this pipeline addresses is to identify and remove data duplicates and harmonize information as much as possible across the data set. So this could be conversion of units to standardization of chemical identifiers. And then the last step is to upload the information to NICE. So this QR code will send you to the main datasets landing page there, where it will provide more information on this process in addition to a table, a snapshot of which is shown on the left here, that will provide the specific dataset, once again, sort of organized by toxicity endpoint, a brief description, ways in which you can explore that information through NICE, and ways that you can download it. There's also a menu on the left side of the screen that will have different pages that explore these data sets in more depth. So it will include any additional curation, the exact data sources, and the endpoints. I next wanted to take a few minutes just to provide an example of a data set that has been curated and made available on NICE. And so I'm going to talk about a curated high throughput screening in vitro data set that is first retrieved through the EPA's ToxCast program, which also includes Tox21 information. And so this data is reviewed and it goes through this pipeline that's shown here. And this is really meant to be a complementary workflow to further bolster the confidence in the outputs and the biological relevance. So if necessary, there are various areas that are flagged related to the chemical quality, the concentration response modeling, and identify areas where the chemical could potentially interact with the assay technology. And lastly, the assays are annotated. So these incorporate ontology terms gathered from the Open Biological and Biomedical Foundry, or OBO Foundry, and this is really to further structure this information and increase the interoperability. The main aim with this step is to take the really useful detailed mechanistic information that's provided and relate it to a biological process or a mechanistic target in a toxicity pathway or a mode of action. And so this process is really subject matter driven as well to ensure that the most appropriate terms are selected for the assay. And below here, you can see the knowledge organizational structure. So we always include the assay endpoint names. We then annotate it using an ontology term to that mode of action. We categorize it to a toxicity endpoint that is utilized throughout NICE. And then lastly, annotate using an ontology term to a mechanistic target. And so this figure is just to show the distribution of the number of assays that are annotated and categorized into those toxicity endpoints. And as you can see, there's a pretty wide coverage among them. The two areas that have the most assays annotated are related to acute lethality and developmental and reproductive toxicity. So I'm now going to switch gears a little bit and talk about the computational tools that are available in NICE. Unfortunately, I don't have the time to cover everything, but just want to mention that there are tools available for data exploration, chemical similarity, biological responses, and understanding potential exposure scenarios. But the two tools that I'm going to focus on today, again, are those that can generate human relevant predictions. So the first tool that I'm going to talk about is the physiologically based pharmacokinetic tool, which already was talked about earlier in this session. But just as a quick recap, these use mathematical models to predict tissue-specific chemical concentrations. They're calculated using absorption, distribution, metabolism, and excretion parameters, which can ultimately help answer some of these questions shown, such as how much of the chemical makes it into the body and how do the body's enzymes help clear this chemical. Traditionally, these are require extensive knowledge on human body, the biological systems, in addition to extensive coding knowledge. And so the good news is that there are models that are publicly available to help with that, and want to highlight those that are produced and made publicly available through EPA's high throughput toxicokinetic packages in which NICE utilizes. And so these models can really help to relate external doses to internal concentrations, help prioritize chemicals for screening approaches, and at which concentrations to test for. And so here's an example of the input page for the PBPK tool. It essentially kind of takes these publicly available models that the EPA provides and adds a user interface to them. So you have the option to go in there and select some various options, customize some features to create these user-friendly models without needing that coding expertise. So some of the inputs that you can choose from are the species. So currently there is human or rat, but the next release will feature about a handful more, including rabbit and mouse. The source of the ADME data, so whether it is experimentally measured, it's an in silico prediction, or a combination of the two. Exposure scenario information, which is really related to the type of PBPK model. So right now there are 3 models, an oral or IV model, a inhalational, and a maternal and fetal model. And a new feature of this tool is that you can upload your own physiochemical data to be used within these models. So here are some examples of the visualizations that will show for the results. In addition to these, there is also a table of information that you can explore, filter, and download. This top left figure shows the chemical concentrations over time per tissue compartment, and the figure on the bottom right shows the distribution of the maximum chemical concentration per compartment. Both have the option to take your cursor and hover over various areas of the plots to find out more information. And you also have the ability to filter by compartment. So for example, if you were only interested in viewing the gut and the kidney, you could filter out all of the other compartments. And all of the figures in NICE have the ability to zoom in and out using your cursor. So before I move on to the next tool, I just wanted to highlight 2 features that all NICE tools have. One is a filtering option, and shown here is just an example of the different filters that you will see within the PBPK tool. And another feature is to send the results to other NICE tools. So this enables the seamless population of information from one tool to another. And so essentially you can use both of these to further refine your results, find additional information, and create workflows. So for example, you could start in the PBPK tool. You could further refine your results to only focus on chemicals that have a high accumulation in tissue, and you can find data sets that were tested using that chemical list through sending your results to the search tool, or you could send it to the IVIVE tool to generate additional human relevant predictions. So the IVIVE, or in vitro to in vivo extrapolation, was also covered earlier in the session, but again, just a quick recap. This utilizes in vitro assay information to make human relevant plasma estimates. They incorporate toxicokinetics, so often involve PBPK models, and consider how the mechanistic in vitro information that can be garnered from those assays can be related to the human body. So you can take a concentration response curve that was generated using a cell-based system, select a specific metric within this curve, and use these mathematical models to ask or answer the question of how does what is the predicted equivalent dose in a human to cause the same effect. And so here is an example of the input page of the IVIVE tool. As you can see, it's very similar to that of the PBPK and includes pretty much the same customizable inputs. It also utilizes publicly available models through EPA's HTTK package, in addition to a few in-house models. There are a variety of options for in vitro data selection, including taking from those annotations of the curated high throughput screening data set mentioned earlier, and also uploading your own in vitro data. In addition to that, you also have the option to upload your own physiochemical property information to use in the model, and also data to overlay in the results. So this is a one of the results that you'll come across. It is visualizing the estimated human equivalent doses, the distribution per chemical. And once again, you could use your cursor to hover over these specific data points to get that estimated dose. in addition to the specific in vitro assay that was used to make that estimation. There's also some options to overlay information. So if you were interested in comparing these predicted values to in vivo measured data, there are a variety of options to do that within NICE. You also have an option to upload your own in vivo data for overlay. And what is shown here is just an example of overlaying some of the exposure predictions within NICE so you can understand how these predicted doses that cause an effect within the human in vivo system, for example, can compare to potential exposure scenarios. And so just in summary, I hope you have a better idea of what NICE is, the types of information that are available, and how you can explore this information through the tools, including the interactive visualizations, utilizing computational models without coding, and enabling workflow development through the filtering and the send-to features that all of the tools have. So as I mentioned earlier, there's a lot about NICE that unfortunately I don't have the time to cover. So I just wanted to put a few resources here to help you explore it on your own. One thing that I want to note is there are trainings available both in person and virtual. So if this is something of interest to you, please do not hesitate to contact the email listed here with your interest and request. Lastly, just wanted to quickly share some other publicly available data resources with a sort of a specific highlight on the first two mentioned here. So the first is the EPA's CompTox Chemicals dashboard, in which you can find the really detailed information on the high throughput screening data that was mentioned, in addition to where you can find the high throughput toxicokinetic packages that are utilized in the two tools that I talked about today. And last but certainly not least, I just want to thank the NICE collaborators, in addition to the amazing group of people that are listed here that I have the opportunity to work with every day. And just want to thank you so much again for the opportunity and for your attention. And I'm happy to take any questions.
Thank you so much for that presentation. And thank you to all our speakers for this session. We will now move into our panel discussion. So with that, I'm going to hand it over to my session co-chair, Alice Tarantal, who will moderate this discussion.
Thank you, Christine. And thank you to all the speakers. The presentations were really excellent and tremendously helpful. I wanted to start, I know there's a question in the chat, but I wanted to start first. Perhaps Melissa and Bridget could address these, and then the other speakers chime in. Could you begin to speak about how to harmonize nomenclature? And, I'm thinking about, as I hear all these tremendous presentations today and the sheer volume of different methodologies that people are using, some of which were mentioned in this session as well. For folks that are starting out, how do they begin? Where do they start? How do they even begin to tackle some of the questions if they really want to get into the NAMS field? So I think perhaps we can just start with the harmonization of nomenclature.
Sure, I can go first. I think just in general, in the development of standardizing nomenclature, which is something that's near and dear to my heart, anatomy diseases in particular, it's very challenging, and it's different communities use different vocabulary to mean different things. They can have different labels for the same thing and the same label for different things, and so coming up with so that's one point, is that I like to say that harmonizing data is just as much about harmonizing the people attached to the data as it is the data. Governance and decision-making and community inclusion, documenting policies and processes, as you start to mature, is really important, so that everyone feels that they are having their needs met, that they have a say, that their contributions matter. We also, I said it in my talk, but provenance, understanding where concepts and definitions for those concepts are coming from, so that we can always go back to that. There's a lot of terminologies that are used, especially clinically, that don't come with any context of use or definitions. And we wonder why our downstream electronic health record data, for example, is so messy. And so that's a really, really important thing, is to we need to understand the context of use and the actual definitions when we're applying those standards to the data. Otherwise, it's just a label, and labels can mean a lot of different things to different people. The other thing is just spending some time and actually, and resources to understand the requirements. So there's a tendency in some standards communities to over-model or over-engineer standards. And it's important to find that right balance of what is useful and feasible versus, letting, slots in a schema fill the need. And I think that's some of the things that Jason and I are going to likely end up talking about here in a minute. Because we can't standardize everything. It's not possible, and it's not useful. And so making sure that we understand, if we had really good standards and they were harmonized, what would we do with them? Where does it get us? And then, of course, I'll just close my comment by saying that AI is not going to fix this problem. It's something that works very well hand in hand with a lot of the AI technologies, but it's not going to make making our data, truly understandable about the context isn't something that AI can do for us. AI does not generate data or understand, why you did an experiment the way you did. That's in your head, right? So we have to find ways of documenting things that we can give to our AI agents so that it can really use that information in really meaningful ways.
So Bridget or Jason, do you want to chime in?
I'll just say I completely agree. You hear standardization and harmonization. It's only two words, but it's extremely complex. I think an area that would be useful, and I already know some initiatives that are doing this, is to get together researchers and experts in a field and come together and think about ways that you can use ontology terms for assessments, for example. And so I think that that's really exciting to see.
Jason, did you have something you wanted to add to that?
No, other than to say that, I really agree about the point that Melissa's making about provenance. And then the other thing is that, at the end of the day, each cell type has to have a function. And so, while my talk was really based a lot around transcriptional profiles, at the end of the day, these cells need to make surfactant and secrete surfactant and differentiate into an AT1 cell. And so, I think being able to align things at the end of the day based on function is one way that we think about this, this problem of, annotating a cell type.
Okay, thank you. Murat, did you want to add anything to that?
Sure. If I jump in there, a little bit based on our experience, right? The data is associated with the study design. And I think that becomes an important aspect, how to link the data and the study design, because at the study design, there's a lot of variability between the NAMs. There are a lot of NAMs towards more acute responses. There are the chronic responses. And the more standardized way of assessing the study design, whatever NAM is in that process, and looking at the outcomes, the data complexity, finding that link, that will, I believe, improve the process. We're not just looking at a standard protocol, trying to get the standard protocol, but the studies, while it's been running, that the diversity of the studies that the pharma or the researchers are asking, how can that dynamically connect that the complexity, chronicity of the diseases or the responses in that data sets? I think it will be really important expanding to more high content data, more diverse set of data, not just repeating the one task over and over, but expanding the knowledge with the data and understanding the studies.
Yeah, thank you. So there's a comment in the chat for Dr. Spence. Do you really think without the vascularization establishment, we can compare, I assume they mean organoids, to human tissues? And if yes, how?
So the question is about without vascularization. So, I mean, yes, I do think we can compare an organoid to human tissue. I mean, but there's a million caveats there, right? So that have been mentioned by a lot of other speakers, including the media and the Matrigel. I think the cell types that are there and not there are also really important things to consider, right? So I forget, one of our speakers in the last session maybe sort of said that it comes down to the question that you're asking and whether the system that you're using can address the question that you're asking. So I think, an alveolar type 2 cell can be used to ask some questions, but it can't. Yeah, you're right. You're not going to ask whether, an alveolar unit can exchange gas without, having all of the appropriate cell types there. So I think it really just comes down to what's the question that you're asking. I don't know. Would any of the other speakers or panelists like to chime in on that?
Yeah, I was going to mention too, again, I think that's to my comment about, which aspect of the, human physiology or disease biology is actually being modeled. We're not going to model all aspects of a human in any model, right? And so if we can understand, what makes the modeling of that component valid in that context, and how can we combine that information with other disease modeling, whether that's in a whole organism, in a different NAM, even AI models. I think that's what I was trying to get at, is use each NAM for what it's good for. Start to push the limit, of course. We want to innovate in our NAMs, but let's not expect them to, pretend to be a whole human.
Okay, very good. Any other comments on that? Okay, because there's another comment in the chat. It says, I was under the impression that ARIVA was supposed to focus on biomedical research, complementing NICE-ATM's focus on toxicology. Are similar efforts or tools being developed for biomedical research, more specifically basic research, disease modeling, and translational?
Thank you so much for your question. Yes, so for those that aren't aware, there's a new NIH office focusing on research, innovation, validation, and application, and it will focus more on biomedical research and human relevance. It's just getting started. There is definitely efforts beginning to incorporate more basic research, disease modeling. I don't have specific examples, but it's definitely an area that we're working on, and be on the lookout for updates in the future.
Thank you. Any further questions on that? Any questions in the chat? Okay, so perhaps we can go back to talking a little bit more about comparative databases and improving model selection and translational relevance. I'd like to just sort of open that up for some further comments based on the 4 presentations we heard. So whoever would like to go first.
All right, I'll jump in.
Thank you, Melissa.
But I think there's a lot to discuss here, so hopefully it'll be an active discussion. I think, when we talk about sort of reproducibility and, with these really complex models and the complex data that's generated, and Bridget presented some really, great tools and resources for, kind of analyzing and comparing those data. I think we need to think about, this is not specific to disease modeling or NAMs, it's just biology in general. We all could be doing a better job of understanding the evidence that we're generating in any given experiment. So, do we have the same degree of concordance when we run the same experiments in our own lab? What about when we run complementary experiments that corroborate that evidence in our own lab? What if we run an experiment in the same experiment in 2 different labs? That's different than running corroborating experiments in 2 different labs, right? And so, these kinds of approaches can together kind of create a more comprehensive picture of how corroborative is the evidence that we're generating, if we're going to be using that information to be making policy decisions or designing trials. And so I think, again, that's one point. And the other point is there is standardization as the tip of the iceberg. Like, we've all heard the story about the animal husbandry person who was wearing perfume, and it influenced the outcome of the experiments. Like, we're not going to standardize every single thing. It's just not possible. Biology doesn't work that way. But we have to be able to understand, what does it mean to actually corroborate evidence more than reproduce an experiment? And that's where I think we kind of have not quite reached the computational strategy or even a culture of thinking about things in terms of degree of corroboration rather than degree of reproducibility.
Any other comments on that one, speakers? Okay, I think we just have a few minutes left. If not, was somebody getting ready to say something? Go ahead.
No, just want to add to that. I think the most important, based on our experience, is the defining on the context of use and application and working backwards what's necessary for that particular decision to be made. It can be sometimes as basic as the inclusion of the positive negative controls for that supporting data, or ultimately we want to have a decision without any positive control and whatever the readout is. But to get there, it will take time, and it will be stages of when the technologies are evolving over time, or our knowledge with those technologies are evolving, not just the technological evolvement. There's more compounds being tested. In that case, working from backwards, defining what will be necessary to have a reliable decision, and what's the impact of that decision? Is that the decision is selecting a compound? Is that the decision pushing forward an IND, or designing the first in human decision? That will be different levels of needs for that.
You bring up an important point too, I think, that we haven't really spoken much about today, in that, that oftentimes when we're building disease models from human context, human tissues, we don't have all the clinical context of those human beings most of the time. We really separate the work we do in the wet lab from the sort of clinical characterization, and it goes well beyond just what's in the electronic health record as well, right? So, I think we're going to have to find ways to build more HIPAA compliant environments that can allow us to capture that, and then, create de-identified versions of some of that information if we're really going to understand, because every human is an N of one. And if we're building models that we want to generalize across, across different disease contexts, across different types of experiments, then we have to really take into account that heterogeneity of the human population that we're actually using to derive the NAMs.
That's an excellent point. And also, it sort of gets back to the point about next steps. So the point was also raised in a previous session about using a variety of different cell lines versus one. So obviously we're going to see a variety, which you would expect and you would want, because you're going to see the same in the human population. So I guess the question is, if I could just throw it out there in the last 2 minutes here, is what are the best possible next steps to moving towards these kinds of databases, comparable ways of looking at questions? We've gone through this in many other subject areas over time and with the NIH. So what might be next steps to really try to pull some of this together? So Bridget, I'm going to turn to you. How about you might make a comment on that?
I think, I hate to keep saying harmonization and standardization, but I think that's really a next step in providing as much information as possible on these different cell technologies and NAMs to increase the confidence and the understanding of the underlying biology of these technologies and identifying areas in which they can be combined.
I think another comment I might make too is just that, a lot of post-generation harmonization is much harder than if you work together when experiments are being designed and data is being generated. And so if there's a way to include, those of us, several of whom are on this call, in those, experiments, in those, funded programs, further upstream in the process, and this is true for all standards and data integration activities, instead of as an afterthought after the fact, we have to go and try to harmonize these data. It's just a lot easier if it's built in at the outset. And the people who are really experts at doing the disease modeling aren't usually the experts in data harmonization, data standards, the kinds of AI compute that we want to do downstream across different resources, and so how could we better, combine our different communities together more at the outset?
I think that's an excellent point. Exactly that, because I think sometimes listening to talks with experts like the four of you and going through some of this can be a little overwhelming for folks who are not really understanding a lot of the databases and how you approach those questions. So thank you all very much. Great discussion. Appreciate your talks. And I see Oleg is probably ready to call our session.
Thank you very much. So we have our last break. Let's be back at 4:35. Okay, it's 4:35. We can start our last session. I'm sure it will be as spectacular as the ones which were before. We have two co-chairs, Malgorzata Achesinskaya and Yonjin Su. So Malgorzata is a program leader in the NIH Office of Strategic Coordination, where she leads the complementary program to accelerate the development, standardization, validation, and use of human-based NAMs. She recently launched the NIH-FNIH public-private partnership effort to establish the complement area validation and quantification network for NAMs adoption and implementation. Yonjin Su is a program officer with the NIH Office of Research Infrastructure Programs, where she primarily manages U-Series cooperative agreement grants that support research infrastructure, model systems, and multidisciplinary resources to advance human disease research. Please take it from there.
Thank you very much, Oleg, and I'd like to welcome everyone to this last afternoon session of the meeting, and the last session being multidisciplinary collaborations, training, and future direction. So we're very happy to have a great panel lined up for you. And without further ado, I wanted to introduce our first speaker, who will be Dr. John Wixwell, who is going to be talking about organs on chips and organoids. What can you ask of your microphysiological systems? How far can you extrapolate beyond what they tell you, and what's needed? And Dr. Wixwell is the university's distinguished professor of biomedical engineering, molecular physiology, and biophysics, and physics at Vanderbilt University, where he also holds an AB Learned Professorship of Living State Physics. He founded the Vanderbilt Institute for Integrative Biosystems Research and Education, or VIBRA, in 2001 to advance interdisciplinary research at the interface of biophysics, bioengineering, and medicine. And Dr. Wixwell's research focuses on microfluidics technologies, organ on chips, stem cell systems, and intelligent well plates for biological analysis. So I'd like to hand it over to Dr. Wixwell. Welcome.
Hello. Thank you very much for the opportunity to speak today. I have been quite impressed by the work that's being presented and want to contribute a few thoughts, some of which are largely historical. Okay. Can you see my, whoops, I haven't shared my screen yet. Just a second. Can you see my screen now?
Yes, we can see your screen. Thank you.
Okay, great. So I think the fundamental question is, why is biology so complex? These give you some numbers as to just the magnitude of the problem we're trying to solve that has guided me over the past 25 years. We need new experimental and computational approaches to get the data for better models, and those models include NAMs. Okay, this introduction should be quite knowledgeable to everybody here, but I just wanted to bring it up to get it in context. Disease is in fact a multi-scale problem going from molecules to animals and interconnections both ways. I view homeostasis as an orbit in a high dimension phase space, which means that disease is a perturbation in that orbit. So classically, biology has been driven by reductionist biology, going down to genomics. The high throughput screening system has allowed us to get to this, as has gene sequencing. Now, Laszlo Barabasi in his network analysis argued that in fact, he had a slightly different shape, and he just draws it as a triangle. But integrative physiology is at the top. You have dynamic, functional, and cellular measurements in the middle. And that is, I think, extraordinarily important for microphysiological systems. And the conventional multiomics is at the bottom. So I came to the conclusion that a few years ago that you need a hermeneutic circle. Hermeneutics is the study of knowledge or meaning. And in hermeneutic circle, you can't understand the whole unless you understand the parts, and you can't understand the parts unless you understand the whole. That applies. That applies to language. You can't understand a language until you understand the vocabulary, and you can't understand the vocabulary without the context. You can't understand history until you understand both the historical events and their context. And so in animals, you can't understand the animal until you understand the parts, and you can't understand the parts until you understand the whole. Now, this is a quote that I got in writing in a paper with Dave Watson and Rosemary Hunsaker. Biology is conducted on cells that have 2D biology on plastic is conducted on cells that have cancer or inbred diabetic. Potatoes on a stiff couch without exercise, enjoy neither gender nor sex, live almost entirely in the dark, gorge themselves on sugar once a day, maybe slowly suffocating in an increasingly acidic environment, live in their own excrement, never bury their dead, may take a complete or only partial bath every day or two, and talk only to cells of like mind. So this has, I think, been a rationale against 2D biology on plastic. Now, this is another one of a network analysis from Laszlo Barabasi, which is basically a gene map of disease. And it shows all the different genes and their groupings and diseases. So there are very few diseases with a single gene. Cystic fibrosis, as Francis Collins once showed, is a single gene disease. But the majority of diseases as we know them and have to study them are multi-gene, and that contributes to the complexity of everything we're doing. Okay, I'm primarily been interested in hardware, and this morning I asked Claude, the LLM, to help me analyze some of my earlier writings and also the market. And it comes up with this grouping at my request of companies that are hardware specific and more supportive companies. Much of this is coming from the 3 R's, not entirely, but much coming from the 3 R's page. Now, what's interesting is if you look at all of the people in the organ chip business, there are only 4 that, to my knowledge, that are currently advertising platforms. Vividyne, I think, has the capability, because of Dan Hu and his colleagues spinning out from Penn, the ability to sell complicated hardware, but I have not yet seen any of it. Emulate has its initial offerings, and the subsequent CN Bio has the physiomimic system, Memitos with the rocker plates, which are limited but very parallel, and Tissue specializes in coupled organs. Hesperos has a platform that couples organs, but I don't believe that it's for sale. It's basically for contract research. I think this is something that the community will have to address, not only for organs on chips, but organoids and assembloids and connectoids. So I asked Claude this morning to build me a table of the points that I had raised in the articles, the review articles I'd written for the first 5 years I was in the organ chip business. Scaling, some of these have been addressed, some are not. So the challenge is to find how to scale your organs if you're doing multiple organs, set functional scaling criteria, get the right dimension for your model. Don Ingber has gotten a tremendous amount of very beautiful work out of 2-dimensional organ chip models with barriers, so 2+ dimensions. And the key point also made today is you want your models to be simple enough, but not too simple. And the trick in diseases is to figure out what is too simple a model. So in the fluidics and microenvironment, there has not been all that much work been done. I think the universal serum blood surrogate has been addressed by a number of groups. Don's original point was that endothelial barriers will make that mitigate that problem. I agree totally. I also believe that the vascularization of tissues at realistic surface to volume ratios is going to be critical, and that's why I think that the vascularized organoids are definitely extraordinarily important. There are a few groups working on that, one of which that I hold in high regard is Brian O'Grady and Ethan Lippman's work at Vanderbilt. So the trick is, can you reconstruct a 3D context with perfusion and interstitial flow versus 2D monocultures? I've been focusing on low dead volume pumps valves that are cheap and parallelizable. There's been mention today about sensing and analytics, very little mention about control. And so the question is how do you do analytical chemistry on smaller and smaller volumes? I'm a strong believer in ion mobility mass spec, which addresses salt removal issues for regular mass spec. I've also concerned about the need to recapitulate pharmacokinetics and pharmacodynamics. There was a couple of mentions of that today. And this is predicated, the ability to do this at volume, at scale, means you need to have low volume metabolic soluble vector sensing and automated microscopy. Tremendous amount of work done on automated microscopy, not that much on the sensing. Now, what has not been mentioned at much length is the control of these systems, and it could be as simple as pH control. We'll talk more about that in a minute. How do you diagnose your MPS, organ health versus disease? And as I will show, how do you compensate for missing organs? Now, critical in this whole business are the models, and they're going to have to be multi-scale models. I'm a believer in, but not yet a practitioner of, machine learning model inference. You have to have noise-tolerant pharmacokinetic estimation. Important if you have a model is how do you get the parameters for it, and how do you validate the parameters you've chosen. There's been a lot of very good discussion on reliable cell sourcing and human iPSC differentiation and characterization, some of which is becoming highly automated. There's also been discussion on integrating immunology to get the cost low enough that you can do high content screening, which means large enough volumes of cells. And another point that was made today that is extremely important is what about the low probability adverse affect issue with drugs and also rare diseases. You have to figure out how do you get enough models to get statistically viable results. We've talked about, or there's been discussion about standardization, qualification, regulatory acceptance, and defining what your model must do, because the model is not perfect. You have to design what you want. So in terms of technologies, I've been working on the microfluidic hardware, which is basically pumps and valves and microformulators, low-cost disposable chips, and also biosafety level 3 utility, which we've done several experiments, and as have others in BSL-3. Sensing and analytics, there's not that much been done. A tremendous amount on automated microscopy and image analysis, but not that much on sensing and analytics. I'm a believer in ion mobility mass spectrometry and other multiomic techniques. The modeling in terms of building biological models, self-assembled perfusible microvascular networks, machine learning, automated model inference, the media, these are all modeling issues. And then the translation is how do you validate your models? Where do you get reference compounds to test your models to show that they're in fact working? We'll talk a minute more about demonstrated negative predictive value, which is extremely important. And how do you do PK/PD and PD/PK? And what constitutes a disease model and how easy is it to use? So these are all the things that are needed. I've worked on a few of them. Others have worked on others. I think the community in general has to work on all of them. Now, going to my specific work, this is a key patent. It's Mike Schuler's patent from 2002. It is, in fact, the one that introduced organ chips, and it's the concept upon which Hesperos is built. What's probably not widely known is that my group did the original engineering presentation for Don Ingber's first DARPA proposal, which led to 2 patents, one Wixow et al. And another Wixow et al. Describing that work. This work has not been implemented either by Emulate or only minimally by Vanderbilt. What came of it, though, was the realization we could build, back up, we could build what we called a missing organ microformulator. I don't know if it's not in these drawings, but it was basically a gadget that would deliver to your cells whatever was supposed to have been delivered by the organ that you could not put in. So I presented this slide in 2015, earlier slide in 2015, Society of Toxicology, and Matt Wagner, then of AstraZeneca, came to me and said, "Can your missing organ microformulator add pharmacokinetics to a 96 well plate?" In oncological drug development, he was extremely concerned about pharmacokinetics. So we set up a collaboration with CN Bioinnovations. We provided them with pumps. They built their own valves. And we did a paper on pharmacokinetics of cancer drugs that was published in PLOS Biology in 2022, where this is the in vivo PK profile. This is the conventional high throughput screening profile. And this is what we were able to replicate in our microformulator. And there are studies, which I won't go into, that basically showed that, yes, we can get pharmacokinetic profiles. And they were extremely thrilled that for these particular drugs, they could get the ordering of tumor volume as a function of different drug with the appropriate pharmacokinetics. And we even found in one case that a drug worked beautifully for a mouse pharmacokinetic profile, but not for a human pharmacokinetic profile. For reasons that I don't understand, there don't seem to be that many people interested in oncological drug pharmacokinetics at this point. Now, that work then went on to build better pumps and valves by our group, David Schaefer and Ron Reiser being the key people on that. And Ross King asked me to build a chemostat for yeast cell culture. And the idea was we could build a system that had a microformulator, reservoirs, pumps, chemostats, and sample withdrawal systems. And this is a picture of a system that's currently running at Chalmers University of Technology in Sweden. We're in the process of expanding it to a 48 well. This is a 48 well plate. We're using 12 of them in this version. We now understand how to build this thing as a 48 channel chemostat that would allow you to run 48 perfused experiments. Now, the microformulator can run in both directions, which means you can actually use it to withdraw media from one chemostat or organ chip and deliver it to another. So this is a cartoon of how you could have multiple organs talk to each other. This has not been implemented. Now, on the analytical chemistry side of things, Barry Pallotta, the program manager at DARPA that ran the organ chip thing, contacted me about doing mass spectrometry multiomics. And eventually a program was created and we were funded for mechanism of action. And the challenge was to identify the mechanism of action of an unknown drug in 30 days or less. We were handed 50 micromolar cisplatin, applied it to A549 cells. We had all sorts of different approaches, proteomics, metabolomics, phosphoproteomics, RNA-seq. And the first 30-day workflow on cisplatin found 250,000 unique features. We then did a canonical map that basically said, these are all the things that we are finding changed in the multiome of A549 cells with exposure to cisplatin. We came up with a data-driven network, which was everything, and the expanded canonical network. So this is everything we saw in A549 cells upon exposure to cisplatin. And that allowed us then to look at a resistance-driven network and then postulate an expansion of the cisplatin mechanism of action resistance. DARPA would not fund us to validate that mechanism. So it's a purported expansion of the cisplatin mechanism of action network. But it showed that time-resolved omniomics had great potential. We then applied it to bendamustine. We got up to 780,000 features with 12 time point 7 platforms and came up with a time course and Pino has movies of how that signaling varied over time and came up with a postulated mechanism of action for bendamustine. And then we looked at methotrexate and showed that we had a fair number of features. We basically had 1.2 million statistically significant features in our multi-omic spectrum. Now, as a quick aside, if you are building a mathematical model of a bioreactor, you ask, does it acidify? It turns out that has been historically viewed as a very hard problem. If anybody's interested in a digital twin that can calculate acidification rate through metabolic flux analysis, My graduate student's PhD dissertation was a demonstration of a technique to calculate the pH of Brent yeast culture media from first principles. Now, I asked Claude, as we wrap this talk up, for two things. What are the kinds of things you can ask, and what can you do to extrapolate? So I don't want to take the time right now to read this because we're almost out of time. I want to point out that Melissa Handel's talk, this is a slide I grabbed from her talk. I think it's totally relevant. I was thrilled by many of her slides because she was doing things that I had hoped someone would do for a long time. And key things: batteries beat single molecules, use layers for what they're good for, and cross-validate signals. So this morning, the paper by Watson was basically effectively my exit from the organ chip business. I've done a little bit, but nothing as significant as I had done before. And Dave Watson was the first author. He was at the time at Eli Lilly. And Rosemary Hunsiger, who at the time was at NIH and was one of the originators of the organ chip program. She basically gave 4 conclusions. You have to earn negative predictive value because the systems are great if you want to show that a drug is toxic because you're showing it a toxin. The challenge is, what do you do to show that there is no toxicity? Now, the difficulty shows up is that if you put your battery of tests together and if you don't do enough of them, you're going to end up getting false negatives, and they will end up convincing you that a drug is safe where in fact it may not be. So there is a great risk on small numbers of tests, on limited model systems, that you will have false negatives that will deceive you. You also have a problem of making coupled systems reliable just because of the odds of any chip failing. And you have to win regulatory trust because as Watson basically stated, the first MPS safe but animal toxic discrepancy will be hard to resolve and be long remembered. So my key observation is that the work today is truly impressive. Microphysiological systems will have an ever greater value for testing drug efficacy and mechanism of action. Interconnected vascularization of organoids and assembloids will replace organs on chips. Regarding drug toxicity, how do you address false negatives? Do you need every organ? Artificial intelligence will prove ever more important for digesting large multi-scale, multi-messenger data sets. And AI will play an increasing role in experimental design, but it needs automated sensors and actuators. And there's a need to develop hardware to increase experimental throughput. So my one suggestion to NIH is get more money to develop hardware. And it's unrealistic to dismiss animal and human experiments. Thank you very much.
Thank you so much, Dr. Wixbo, for the very comprehensive presentation and summary. We will hold questions until the discussion portion at the end of the session, so thank you again. And now we'll move on to our next speaker. Our next speaker will be Dr. Joseph Wu, who will talk about stem cells, genomics, and AI for drug discovery. And Dr. Wu comes to us as the director of the Stanford Cardiovascular Institute and professor of medicine and radiology at Stanford University. He trained at Yale and UCLA, and he specializes in adult congenital heart disease and cardiovascular imaging. His research focuses on stem cells, organoids, genomics, AI, precision medicine, and drug discovery, including developing clinical trial and dish models. Over to you, Dr. Wu.
Thank you so much, Dr. Ochenska, for the invitation. So I'm going to talk about session 4, multidisciplinary collaboration. I changed the title a little bit because I want to highlight this biobank that we could use to collaborate with other investigators. This is my disclosure slide here. And I think, as you know, the drug discovery is both time-consuming and very expensive. For example, typically it'll cost you about 1 to 2 billion, and then 10 to 15 years to bring a new drug from R&D through clinical trials to market approval. Now, what some of you may not know is that many of the FDA-approved drugs in the market today may not work as well as you think. And shown here, for example, the top 10 highest sales of drugs in the US. Blue means that the drug works, and red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps one person. For clinicians, we call this NNT, or number needed to treat, to show one positive result. And for most cardiology drugs, the NNT will be around 20 to 30 to 40 for drugs such as beta blocker, ACE inhibitor, and aspirin, and so forth. So overall, this is pretty poor response rate and definitely not quote unquote precision medicine, right? And so this is the reason why the editorial here is titled Time for One Person Clinical Trial. Now, this slide highlights, in my opinion, why I think is one of the major problems for the high failure rate in drug discovery and the imprecision medicine I just mentioned, and that is a lack of genetic diversity and pharmacogenomic differences with traditional models such as using immortalized cell line and genetic inbred mouse models. For example, because these are immortalized and inbred, the deviations that you see here within each group, drug X versus vehicle, this deviation, when you stop and think about it, this deviation is actually due to the technician's pipetting skill for cell culture work. Whereas this deviation here is actually due to the technician's tail vein injection skill for animal work. On the other hand, the differences that we see in the population of, say, 1,000 diverse iPS cell lines is actually more due to the underlying genetic variability that exists among all of us. And this pharmacogenomic difference is ultimately what you're trying to understand before you go into clinical studies that is otherwise impossible to capture or recapitulate using traditional immortalized cell line or inbred mouse models. Now, in my opinion, I think there are 4 key platforms that anyone who's interested in drug discovery should be aware of. And these are human-induced pluripotent stem cells, next-generation sequencing and single-cell multiomics, CRISPR gene editing, and an AI computational design. However, while individually powerful, they can become transformative when you integrate them synergistically, especially in the context of drug development. And the key difference here is that the emphasis on human-centric biology for drug development, as highlighted in this recent review article. Because again, session 4 is on multidisciplinary collaboration and sharing resources, I do want to highlight this iPSL biobank that we have generated. And so over the past 15 years, and more recently in collaboration with Greenstone Biosciences, it's a startup company that I co-founded, we have made iPS cells for more than 2,500 patients with different medical history, ethnicity, and sex. This is the world's largest biobank of human iPS cell lines at one single site. These lines are linked to medical information. We perform multi-omic sequencing on them to help us understand how human genetic variations impacts drug response phenotypes. We work with the FDA and drug safety testing. We also share these cells broadly with the scientific community. And to date, we sent out more than 4,000 bios to more than 450 investigators. This slide shows the demographic breakdown in terms of age, gender, as well as ancestry distribution. As you can see here, we have very broad distribution in terms of our ancestry. And then this slide here shows the disease breakdown as listed here. And again, as I mentioned, we sent out more than 4,000 bios and more than 450 investigators. And if you're interested in any one of these cell lines, we'll be happy to share with you. Now, this is our typical workflow for patient recruitment. We recruit from diverse population of patients in our clinics. We then generate, in this context here, 2D micropattern cardiomyocytes, 3D human cardiac organoids, and also 3D human engineered heart tissues. And this is a recent paper that we published in which we screened for 35 different combinations and identify a differentiation cocktail that gives rise to vascularized cardiac organoids as well as vascularized liver organoids. This is important because, as you may know, for most drugs, the first pass metabolism is the liver. And now you have a system using these vascularized liver organoids to metabolize drugs, and then you can then test the drug metabolites on vascularized cardiac organoids, as well as any other types of organoids out there. And I think this model would help further accelerate the drug development process. And again, in the context of session 4 on multidisciplinary collaboration and training, I also want to highlight that we published many of these protocols for characterizing iPSL derivatives and also iPSL organoids, such as contraction, calcium, imaging, action potential, multi-electrode arrays, and many other assays. We will be happy to share these protocols with anyone who's interested. Next, I want to show you some examples of human iPS cell lines that we generated from various cardiovascular diseases and their associated genetic variants. For example, for dilated cardiomyopathy, we have recruited patients with actin, lamin, myosin heavy chain 7, troponin T, and titin mutations. In the case of troponin T, about 15 years ago, we will refer this 3-generation family with familial dilated cardiomyopathy. The family carries a troponin T mutation with origin in the tryptophan switch. And here, to make a long story short, we're able to show that the disease, the cardiomyocyte, had altered regulation of the calcium ion, decreased contractility, and abnormal distribution of the sarcomere alpha-actinin. Importantly, if you treat it with the drug metoprolol, which is a beta blocker, it can be used to improve the function of these DCM iPSC cardiomyocytes, thereby recapitulating results from multiple large beta blocker clinical trials. For hypertrophic cardiomyopathy, we have recruited patients with myosin binding C, myosin heavy chain 7, ryanodine receptor, cardiac troponin I protein, and tropomyosin. In the case of myosin heavy chain 7, about 15 years ago, we will refer this large family with familial hypertrophic cardiomyopathy. And then through a whole series of experiments, we're able to show that this myosin heavy chain 7 with alternating the histidine switch, codon 663, causes increased intracellular calcium, which leads to calcium handling defect, as well as cell arrhythmia. It also causes activation of the MFAT signaling pathway, which leads to hypertrophic response. Importantly, if you treat it with a calcium channel blocker, such as verapamil, or a beta blocker, such as propranolol, you can use it to reverse both the cellular hypertrophy as well as the cellular arrhythmia. And as you can see in these red boxes here, back then, this is about 13, 14 years ago, we're starting to use these cells as a platform to screen and to study drugs. And then for left ventricular non-compaction, we have recruited patients with calsequestrin, nebulin, phospholamban, T-box, transcription factor 20, troponin T. In the case of TBX20, we have also identified the pathophysiology mechanism for this genetic variant. And so about 12 years ago, we will refer this large family from Stanford, as well as a smaller family from Japan. The Stanford family has a mutation in the transactivating domain, is shown in red, and the Japanese family has a mutation in the T-box domain, as shown in green. And through a whole series of experiments, we're able to confirm that the mutation can cause abnormal activation of TGF-beta signaling pathway, thereby recapitulating a key aspect of the LVNC physiology. And this is actually the explanted heart for the boy that we saw at Stanford. For long QT syndrome, we have recruited patients with KCNH2, KCNQ1, and SCN5A. In the case of KCNH2, this is a mutation in the hERG potassium channel and commonly referred to as long QT2. About 10 years ago, we will refer this patient with the cardiac channelopathy variant. The clinical context is that this patient became worried because his EKG shows prolonged QT, and genetic testing was found to have a VUS, a variant of unknown significance in KCNH2, with threonine to isoleucine switch at codon 663. However, his brother refused genetic testing, and hence we were not able to confirm the disease without additional family testing. This case was brought to our attention. Here we showed that by introducing the variant in healthy line by CRISPR, we created the disease phenotype, whereas correcting the variant by CRISPR led to normalization of the disease phenotype. And based on these steps, we're able to tell the patient that his variant is actually more likely pathogenic. Here's another example involving lamin dilated cardiomyopathy, and this is quite common as well. This is a 4-generation family cohort with lamin AC frameshift mutation that leads to early termination. As you can see here by this family pedigree, many of the affected individuals had cardiomyopathy, atrial fibrillation, atrial ventricular block, and importantly, 8 of them died from sudden cardiac death. And so this is a horrible toll in the family. Here we demonstrated that one of the mechanisms is due to abnormal activation of the PDGF signaling pathway in the cardiomyocytes, which then leads to abnormal cell arrhythmia as well as impaired contractility as shown in this cartoon. We also showed that if you block the PDGF pathway with the PDGF inhibitor, quinolonib, at very low dosage, it can be used to rescue the abnormal phenotype of these mutant cardiomyocytes. However, quinolonib is not yet an FDA-approved drug, and so we cannot give this drug to these Lammin patients. Now, at the same time, we also noticed that many of these Lammin patients also develop early onset hypertension at age 30s and 40s. So we were quite puzzled by this clinical finding because typically you would expect the blood pressure to be low in patients with dilated cardiomyopathy. So here we decided to differentiate this large family cohort's iPS cells into endothelial cells and compare the difference between healthy and diseased patients. Again, to make a long story short, we identified KRF2 as a transcription factor that is responsible for the endothelial dysfunction. We then used FDA-approved drug library screen this time, and we found that the lovastatin can be used to activate KRF2 and to rescue the endothelial dysfunction. In contrast to quinolonib, lovastatin is actually an FDA-approved drug, and we give it often to patients with high cholesterol. Here, we're not using it to treat the cholesterol, but we're using it as a repurpose to improve the vascular dysfunction. And therefore, the title here is Clinical Trial in a Dish, using iPS cells to show lovastatin can improve endothelial dysfunction. So that was just one example of how we use genomics, stem cells, organoids, and AI for cardiovascular drug and repurposing. And if you're interested in learning more about NAMs and drug discovery, here's another recent review article that highlight the major therapeutic candidates that have been identified in other organs, such as brain, heart, vessel, eye, lung, muscle, as well as the models that have been used to develop and accelerate the pace of been to bedside translation. And to help the NAMS field, here are many other examples of cardiac and non-cardiac diseases, as well as the associated genetic variants that we're happy to share. In addition, we also have the world's largest biobank of rare orphan diseases, generated for more than 600 patients. You're welcome to reach out to Greenstone if you're interested in any one of these lines. And so in summary, I hope I have convinced you why it's important to focus on human-centric biology by using well-phenotyped and clinically annotated patient pedigrees. And for drug discovery, I would say that it doesn't really matter whether you're using predictive or generative AI to find the optimal candidate, which could be drugs, peptides, or ASOs, or RNA therapeutics, and so forth. These testings could be followed by your in vitro and in vivo studies. But afterwards, a clinical trial in a dish model can then be used to help you further improve the safety and efficacy of your lead compound prior to going to clinical phase 1, phase 2 trials. So this concludes my talk. I want to acknowledge my postdocs, graduate students, instructors, my collaborators, and also my former trainees, as well as funding support for NIH, CERN, AHA, Chen Zuckerberg, FDA, and also partnership with Greenstone Biosciences. And if you're interested in any one of the cell lines that I just mentioned, you're welcome to reach out to Greenstone. They provide these lines free of cost to nonprofit organizations and academic institutions. The goal here is really to encourage more people to go into NAMs by using these human-centric and disease-specific biological samples. Thank you very much, and I'll stop sharing screen.
Thank you very much, Dr. Wu, for this comprehensive presentation and all of the resources that you have illustrated there. We'll hold questions until the discussion session for today, but I'll turn it over to my colleague and co-chair, Young-Jun, to introduce the next two speakers. Thank you.
Yeah, good afternoon. Yeah, so it's my pleasure to introduce our next speaker, Dr. Ashu Agrawal. Dr. Agrawal is a professor from University of Miami, and he's a leader in organ-on-chip and microphysiological system research. So his lab develops human-relevant models that integrated microfluids, stem cell-derived tissues, and biosensor technologies to advance drug discovery, toxicology, and disease models. So the title for his talk today is Chips on Organoid. Okay, the floor is yours.
Thank you. Thank you so much for the invitation to present. It's been a real honor to be in the same session. We learned a lot. I was at the Wyss Institute during the early years of DARPA grant writing and then actually executing on the grant. And to see Dr. Wixo and learn from him his microformulator designs and the debates we used to have around on-chip versus off-chip pumps. So it's nice to see that. So I won't cover what organs and chips are or why they're important. Much of the field was kickstarted, at least funding-wise, by Don's elegant paper in Science, and then the commercial entity Emulate. And today, as we've heard all day long, FDA is endorsing several different categories of NAM. But I want to focus on some of the external factors which are also driving NAM adoption. People have alluded to this. I just wanted to highlight that I was a part of a consensus study that National Academies put out a couple of years ago, where we really looked at the shortage of non-human primates as almost a national emergency, a security emergency, should there be a next pandemic. There were no NHPs coming to America from China, for example, which was the biggest suppliers, and that also kickstarted a lot of search for alternatives. And then the other external factor driving NAM adoption is all of the commercial startup space that's being populated now by academic labs, which, as we've heard, either have big systems with multi-organ systems or very specific singular tissue chips with specific contexts of use, and they're knocking on the doors of FDA now for validation for a specific context of use. So we were able to write, or got invited to write all of these thoughts in a very sober Nature Communications perspective on how, when, with the initial Science paper and all of the funding through DARPA and other funding agencies and the startups, and then the FDA Modernization Act, the expectation is just exponentially increasing, which is quite typical for new technologies. But what's also typical, this is taught to business school students as the Gartner hype cycle, is for more often than not, it always goes through a trough of disillusionment before it finds a steady state of applications. And this paper, we were asked to lay the groundwork on how to avoid this trough of disillusionment and what would it take for the basic tool developers, basic biologists, and the industry and regulatory bodies, through conversations like today's workshop, to remove those practical roadblocks through stakeholder engagement. So when I set up my shop at the University of Miami after having trained at the Wyss, was to really find a niche that the classic PDMS-based chips do not serve. And that niche is PDMS-free systems, which are for large volumes that often organoids require. So organoids can be up to 500 microns, 1,000 microns, or even 2,000 microns large. And when you have clearances like this, you cannot really rely on PDMS-based photolithography structures for such large clearance devices. So you can make them out of plastics. These can be bio-inert plastics like acrylic or polystyrene in my case. But when you start having cavities, which are about, let's say 1,500 to 2,000 microns, you lose the advantage of laminar flow. When the roof is so high up, the Reynolds number dictates that there would be a turbulent flow. So the secret sauce of the chips we've designed in our labs have been the physical sort of constraints and geometric features that we build into the fluid path, such as obstacles and chamfer cuts and narrow exits. And we solve for the computational flow dynamics within those constraints to still enforce not really a laminar flow, but at least a plug flow and a predictable flow. And we couple those timelines to the biological timescales. For example, in this case, a Michaelis-Menten relationship of insulin release by a pancreatic beta cell in response to glucose stimulation. So the combination of the biological timescale and the computational flow dynamics of the chip will then yield an optimal design of a large volume microfluidic where the clearance is enough for holding large organoids. So we've applied this design philosophy and this class of chips. As I said, we know very, very, very clearly from first principle fluidics how the physics looks like, and we measure that physics with particle imaging velocimetry, and we've applied that as the first case scenario towards pancreatic islets on a chip. So all of the work in my lab has been, with deep collaborations, and this was a collaborative MPI project with Dr. Sherry Stabler, who's at University of Florida. And she and I were able to put pancreatic islets sourced from rodents and from humans inside these novel fluidic architectures and challenge them after having them under low glucose with high glucose, and ask on the y-axis how the insulin response looks. So, people who understand insulin dynamics will tell you that they want to see a first response, which is very quick, but then a slow and sustained plateau response as well under high glucose stimulation. And then only under low glucose do you want the insulin to go back to zero. So we saw that very nicely with the rodents, but even from islet source from human pancreas, we were able to see that response. But what it also revealed with high sensitivity ELISA was that the response would go away within a week. So the shelf life of these pancreatic islets was only a week. These islets were being sourced for clinical islet transplantation for type 1 diabetics. So the challenge became, how can you extend the stable shelf life, the functional shelf life of these pancreatic islets? So for the next gen devices, we supplemented through the floor of the device oxygen. So we would create oxygen permeable floors and then supplement oxygen from the bottom. And through that second gen devices, again, we would measure the oxygen through liquid light guides. So these are optic fiber cables with oxygen spot sensors embedded in the device. And then we could measure we could control the actual oxygen concentration needed for pancreatic islets under high glucose. And very clearly through both live dead stain and with functional assessment, you could see that it extended the shelf life. The other thing we were able to do, and these were efforts funded through the Human Islet Research Network of NIDDK, and that consortium brought together a lot of basic beta cell biologists along with tool developers like us. So we were able to share our chips, and our chips actually live in many different academic units. One of them is the head of Diabetes Institute at Vanderbilt University Medical Center. Al Powers and Marcela Brisova were able to take islets and dissociate them, optogenetically transfect alpha, beta, and delta cells with different ion channels. And under a stimulation light of different wavelengths, they would excite only the alpha cells, beta cells, or delta cells. And having those islets, these pseudo islets, live in our chips and throw the light wavelength corresponding to alpha, beta, or delta stimulation, they could collect the corresponding hormones. And we were able to publish these, protocols and these results in a collaborative manner. Taking that story further, we are now making available an entirely new bioresource. People have been introduced to living slices for cancer, but also, for pancreatic slices. So these are human pancreatic slices that contain both the exocrine and the endocrine part of the tissue. And this work is in collaboration with Juan Bindala Dominguez, where the challenge became that the enzymes secreted by the exocrine tissue chew up the endocrine tissue really quickly. So if you really wanted to study the exocrine endocrine crosstalk, you needed a fluidic device in which your human pancreatic slice would live, but also get continuously perfused and maintain the right oxygen. So we again went back to first principles and designed a robot, a fluid handling robot that could take care of all of the fluidic needs while the chips lived permanently in a microscope. So you could do imaging of very rare cellular events over and over again over a long period of time. And I'll tell you what those cellular events were that we were going after. So again, we solved the physics of the number one killer of microfluidics being air bubbles. And because these are PDMS-free chips, the gas doesn't have a way of degassing out of the housing itself. These are solid plastic chips. So we understood through Henry's law and Ohm's law the dependence of the solubility of oxygen with both pressure and the temperature. And then with all of that in place, we could now get slices to live in our chips and image in our chips, and then also do glucose stimulation in our chips. So that was the first sort of set of results we published 2 years ago now. And in the latest paper we published in Nature Communication, we could now start watching these very rare events. So the rare events we were going after were transdifferentiation of exocrine tissue towards insulin-producing beta cells. So it turns out pancreas have a limited ability of popping up new beta cells far away from the endocrine tissue. So we were going after, these rare beta cell regeneration happening in the exocrine part of the tissue, indicated by that green fluorescence in response to THR-123 treatment. And that would have been so difficult to see in, rodent slices under that treatment. So we are enabling, not just the observation of these rare events, but also applying these chips towards, flowing in immune cells and watching the immune cells home towards a pancreatic beta cell cluster, which is stem cell derived. So here in red is a stem cell derived beta cell cluster. And in immune cells, these find they're primed against the antibody. So there's antigen specificity, and then they home towards the beta cell cluster, invade it, and then destroy it. So you watch that islet immune interaction happening in real time inside these chips, and that opens the doors for the search of the right immunosuppressants that these patients should be on. That work is in collaboration with Todd Driscoll and Clay Matthews at UF. So I'll switch gears and just say, because we learned how to flow in immune cells in a viable fashion, that opened the door to recreating a pancreatic cancer tumor microenvironment in the chip and see how MDSCs switch cancer-associated fibroblasts towards a different phenotype. That work is ongoing and not published, or in a liver chip and flowing in Hep C virus and see how that Hep C turns the liver to a fibrotic state, a cirrhotic state, and then a cancer state. I'll just give in the last couple of minutes one nice example that we love from the lab. The idea is that these chips have to hold these organoids for such a long time that the chips cannot fail. And one of those issues comes across in retinal organoid generation. You saw the brain organoid work and how long that takes. It's a subset of brain organoids that, these optic cups pop out, but then these optic cups, to create a good retina, have to get exposed to an oxygen gradient stably over many weeks of differentiation. So this was an NCI-funded R01 with Dan Pelaez at Boston Palmer Eye Institute at the University of Miami, where he asked us if he could create chips in which he could plop down these retinal organoids and expose them to a stable oxygen gradient to sort of coax the cells into the architecture, the beautiful architecture that the retina has. And so we designed open well chips. We didn't want air bubbles or anything else killing the system. And on the floor of these chips, we would have, so that's the surface where the organoids would sit, but on the floor, we would have an oxygen permeable membrane again, through which we would remove all the oxygen. So on the bottom, we'd have oxygen leaching solution flowing. And on the top, we would have these retinal organoids live for many, many weeks. Again, through simulations and through actual measurements, we verified that there was a steep gradient that these retinal organoids were getting exposed to. And then, we just put them in culture and actually could see early evidence within a few weeks of that cellular microarchitecture appearing in response to oxygen gradients. And we published those early results in a paper recently, where we could quantify the retinal ganglion cell expression responding to oxygen gradients. And in ongoing work, we are actually observing the same timelines of different cells appearing in the retinal organoid as they do in the human retina, because of the presence of the right oxygen gradients. And the grant is actually now to actually introduce the RB1 mutation, either sporadically or in a hereditary fashion, to create RB1 mutated retinal organoids. So just like Dr. Wixo mentioned, there's a lot of adoption barriers. If the chip is simple enough, but the architecture of the fluid handling looks like this, the cell biologists will run away and will never use your chip. So we invested a fair amount of effort in creating fluid handling robots, so that if your chip can be operated using a graphical user interface outside, it takes care of the entire fluid operation, and it's the size of an incubator shell. So you could fit it into your incubator. The partnerships through hardware manufacturers have been crucial in spinning out the technologies to different research centers across the US and in Israel through BioVitro. And that's also my couple of disclosures. The startup company that I didn't talk about was the Circulogics, and then BioVitro as well. These are my acknowledgements, and I will hand it off to the next presenter for the last talk of the session. Thank you.
Okay, thank you, Dr. Agva. We will have save the questions for the panel discussion. So now it's my pleasure to introduce our last but not least speaker, Dr. David Tufson. David is a professor and also director of the Cancer Center from Cold Spring Harbor Laboratory. Dr. Tufson is an internationally recognized leader in pancreatic cancer research. His pioneer work in patient-derived organoid and translational cancer models has advanced precision medicine approaches, and improve understanding of tumor biology, treatment, as well as therapeutic development. So with that, I'm headed to you, Dr. David.
All right. Thank you so much, Dr. Su, and thank you, Oleg, for the invitation. I may be the misfit toy in this last session. I'm not going to teach you about necessarily a new platform, although in the very end, I'll show you some co-cultures we're using. So I'm going to talk mostly about how we've been invested in organoids as a disease model for pancreas cancer by actually going from the tissue from the patients. So they're not reprogrammed cells like we heard about elegantly from Dr. Wu. But we've actually learned quite a bit, and I'll try to share that with you. This is my disclosure slide, which for this talk is not pertinent. The real question that we've had for a long time studying pancreas cancer is if we could stratify our patients, would it matter? Meaning, could we take the patient and put them on the right treatment paradigm? Sometimes that's referred to as precision or personalized medicine. Most of those approaches are based upon static nucleic acid information or biochemical stains on tumors. Almost none of it is based on dynamic assessments, which is what the model systems that we're talking about here allow us to do. We got into this space from a interaction I had with Hans Cleavers when my lab was still located in Cambridge, England. So this would have been in the late 2008, 2009 time period. Talking to him, he was having some success in modeling the intestine of a mouse and really extending that into the intestine of a human. My interest was in pancreas biology, like we heard about from the last speaker a little bit. We had generated mouse model systems of pancreas cancer, which were based upon inserting the genes that are mutated in human cancer into a mouse. Classic genetically engineered mice that possibly others have spoken about today. And we had pretty good models of that. And what we wanted to do is test the organoid system in the mouse, but also human patients. And so again, this slide is mostly a cartoon talking about how we can take normal mouse pancreas and normal human pancreas and generate organoid cultures using the classic ENR culture conditions with semi-solid media. Off-the-shelf product is Matrigel, but lots of other things could be used. So we worked with Hans for a couple of years and optimized his methods and had our first description of this, comparing the two species and comparing an organoid to the patient samples from which we derived them. And we saw a pretty strong correlation. And so we really pushed forward ahead to ask, could this be a useful test? And at the time, Ned Sharpless was the NCI director, and I was teasing him that this might be the bacteriology test for cancer, and which is, the simple-minded way I thought of it. And so this is just a 6-well dish with a human holding the dish and the drops of Natrigel on the bottom. And each drop you have your organoids embedded in. On a cartoon form, it looks like this. And again, just light microscopy on the right. We were able to collect enough samples from enough patients early on to show that there might be a correlation between the response of organoids to cytotoxic drugs and the outcomes of patients who are treated with the similar cytotoxic drugs. So it was a very large collection that we made over some time period, and we were able to cross-reference it to data from Canada where they had done laser capture on human pancreas tumors. And so again, these are the drug curves over here, just the simple-minded IC50s that we did ATP-based assays, the classic method used by biopharma and academia for decades. And we sort of tertiled it into less responsive in red, very responsive in blue, and intermediate in white. My postdoc who drove this was French, and so he used the French flag in a way. And so we could separate the organoids into these bins and then ask, do those represent a signature that we could find? Because you could actually get transcriptional signatures from the organoids, apply it to human data. And this in the middle is our first foray into that, looking at patients that were treated with cytotoxic drugs, showing that there was, for the patients non-responding, maybe the tumor's growing in 8 weeks, they had the red signatures, whereas the tumors, the patients who were responding had more of the blue signature. So not a perfect correlation in this transcriptional signature, but promising. And that led us to ask, if we did it on scale in a randomized trial, might the organoid be predictive? So that simple idea took actually all the king's horses in 2 countries. And so we posed it right after the paper. We got it going, and the pandemic arrived, and it made our lives also adventurous. That we all recall those days. We had 4 sites in the States: Hopkins, Memorial, Dana-Farber, and Northwell Health, which is a health system near Cold Spring Harbor on Long Island. One major one in Canada, University of Toronto. Jen Knox at Canada and Liz Jaffe at Hopkins were the co-PIs, and my site at Cold Spring Harbor, we basically were the receivers of all the tissues to generate the organoids from the patients. And so the clinical trial was to compare 2 types of chemo, and that's shown here. This is combination chemotherapy, FOLFIRINOX on the left and gemcitabine plus nab-paclitaxel on the right. And it was a classic pancreas cancer trial where patients with advanced metastatic disease, where usually the median survival of patients is somewhere measured in months. And if they receive chemotherapy, they will maybe get another few months of life extension. So we compared the 2 chemotherapies randomly, and we received samples to make organoids in all of them. And this is just a table of what the clinical parameters of the patients are to ensure that these were randomized. And it was fairly reasonable for that. We compared the 2 chemos that I described. One of the chemos turned out to be a little better than the other. This is the blue chemo, gemcitabine mixed with nab-paclitaxel, compared to FOLFIRINOX on the whole patient population, looking at overall survival. That was somewhat surprising, because historically there was this concept that this regimen on the left was, quote, stronger. And this is kind of a misnomer of oncologists, that the stronger chemotherapy means the patients live longer. A lot of times the, quote, stronger term comes from the city, actually. And I was really pleased to hear that toxicity was a parameter that many of you talked about in your lectures before me. So this was the clinical outcome, and there was something called transcriptional subtypes, which is true of all cancers. Cancer is a hard disease because it's not just one disease. Even though histologically and even genetically, we can say it's a particular type of cancer, there are transcriptional subtypes in every cancer, and then each patient has multiple types within itself. And so it's a moving target, different from, again, I would say, any other disease that we're treating. And so when people talk about the valley of death, they're not actually being fair to the biology that we're trying to go after. And so, these were the transcriptional subtypes. We knew historically basal patients did worse than classical. But again, the whole point of the trial was to collect information from the patients that would allow us to test were the organoids predictive. And so most of the samples we got were from liver metastases from patients, occasionally from the primary and occasionally other sites. And the biopsies went for organoid development, PDO development, and sequencing of the whole genome or the transcriptome, looking for particular biomarkers of subtypes like GATA6 and doing a deep immune profiling on tissue slices using a multiparametric IHC and immunofluorescence. We did also collect blood to look for exploratory biomarkers of cancer presence. And so this was the report card. About 3/4 came from the liver in terms of those biopsy sites. And this is how we did. And we've been at it for a while. We've probably made 500, I'd say, pancreas organoids by now in the last 14 years. We're still batting 50%. And so 50% of the time we could generate an organoid, and 50% we can't. So we fail half the time, which really, bothers me and makes it hard for me to see, a commercial approach until we can improve upon that. But I'm going to, bear with me, and I'll try to explain what we've learned. In those that failed, that included samples that in the end, we couldn't identify a cancer genome. And so we call these pseudonormals, which is this blue bar under the failure. But we also had samples that never generated organoids in the first place. Either didn't grow at all, which is in green, or it started to expand and then stopped. Occasionally, when you look under the microscope, there were no cells, so it was, a failure, but you didn't have a sample coming in. We've actually studied the failures pretty deeply, and the failures are interesting in that their genetics is not the same as those that worked. And I'll get to that in a second. We actually are fairly stringent on testing the samples right away to make sure it has canonical mutations of pancreas cancer. About 95% have a KRAS mutation, and we just developed a pretty straightforward assay to look for the mutant genome using digital droplet PCR. And our positives are shown on the bottom, on top, our negatives are shown on the bottom, and then we have these samples that have less than 45% of allele frequency of having a variant, but greater than 10 to 20%, and oftentimes these are malignant cultures actually. Here's the interesting finding: if we can make an organoid, those patients did poorly. Has nothing to do with the drugs. So if we can make an organoid in tan, the patients did fairly terrible compared to those where we failed. And so the samples where we failed, actually the genetics and the genomics of these patients had a more benign genome. A lot of them were wild type for P53 and a variety of other things, which is interesting. And this is the overall survival on the left, and progression-free is on the right. In the end, this is not a major thing, but of course interesting biologically, because it makes us think, what are the conditions that we need to support those other cultures? And so again, back to the point about, are they predictive? So we made the organoids and we carried out all of those assessments that we talked about. Part of it was to succeed in making the organoid, test the therapies as soon as possible so we can report the information back to the whole clinical team. We found, despite a variety of ways that we have attempted to improve things, it took us median 70 days from the sample acquisition to actually providing pharmacologic feedback to our colleagues. 70 days is pretty long. When you're sick with an infection, the doc wants to tell you within hours to at most 1 or 2 days, what are you infected with and how do we kill it? So if this is a bacteriology test, this would be like a mycobacterial test, right? Where it takes a long time to grow them out. So obviously this is something that we've been attuned to and we are focused on. So back to the point of when you get the culture, is it in fact predictive? So we did 2 ways. One was this empiric AUC, IC50 type way. The other is to develop transcriptional profiles, which I alluded to before. And by using the AUC empiric based way, it wasn't so bad. So we really looked at hypersensitive and hyper-resistant organoids to develop such an approach. And when we developed a responder signature, those patients that had a responder signature in their organoid and were treated with those therapies that they were most responsive to did better in blue compared to those that were non-correlated in black. Keep in mind again that we are looking at the organoids from the patients that in fact are doing worse because we weren't able to generate the organoids on the people who are doing better. So that looked promising. And we did the same thing about using RNA as opposed to the AUCs. And so the way that works is you take the top most sensitive, the bottom most resistant, and you develop a differential gene expression signature that then you can overlay onto patient samples. And that looked pretty good for one of the chemotherapies, which is gemcitabine with Taxol, less well for the other combination chemotherapy. So there's something there. The mechanism underlying that is still to be determined, but it looked like it could be reasonable. But all the hoopla today, for those of you who read even the New York Times or your own local newspaper, is that we have this new pill for pancreas cancer. The pill is a medicine that blocks the RAS oncoprotein, which is, again, 90 to 95% of pancreas cancer have it. And it blocks it by essentially, the main pill blocks it by actually reactivating the GTPase function of RAS, a discovery by Pirolito at Memorial. The concept of the compound came from Greg Verdine, chemist from Harvard, who's now in the private sector. And Frank McCormick's group at the NCI really got the RAS initiative off the ground with input from Keyvan Shoka and other folks. So when we look at the RAS drugs in that same collection of organoids, this is what we see. And the organoids that don't respond well to the RAS drugs tend to have wild type RAS, which is very interesting. Now, I alluded to this before. We used ATP levels to actually do all of our work. That's slow. And you can only do it once because you have to kill your sample to get the ATP. What about imaging-based approaches to try to get close to you terrific bioengineers that I'm listening to today? Well, here's our foray into that. And so we have a high-content imager, which we have acquired, and we have worked with the company that supplied it. We haven't invented necessarily a new method, but we have adapted what we have to it such that we can see the organoids, which are look green here. This is actually a dye that we can incorporate into the membrane. We've already shown it doesn't affect the proliferation of the organoids or response to drugs. And we've done a co-culture. And so this is the only way I could make it fancy for you all. And so we brought fibroblasts into the mix. Fibroblasts are the main cell type in a pancreas tumor. Macrophages are the second. The cancer cells are third or fourth. And in this case, the fibroblasts, the human cancer-associated fibroblasts are expressing cherry fluorescent protein. And again, the organoids first were incubated with a dye which goes into the membrane and doesn't affect the biology. And so this is what it looks like under the microscope. And this is what it looks like if you fix the samples and stain it with hematoxylin and eosin. And if you go use an antibody to your cherry, this is what you get. The reason why I'm walking through all of that is, what about the drugs? Because you can look just at the green cells and ask, how are they doing? And we've done some experiments showing that the microscopic approach and the AUC ATP approach are highly correlated. So when you look at Taxol, you get the same IC50 between the red curve and the black curve. In this case, black is just organoids and red is co-culture. But if you look at GEM, you have a push to the right. It takes slightly more gemcitabine to kill the organoids in the co-culture. But look at the RAS drugs. This is one such example of a RAS drug, a couple-fold different, like threefold. We've seen up to 10-fold different with the RAS drugs. We're currently trying to figure out, is this pharmacokinetics, amount of drug available, or is it pharmacodynamics, response of the system to the drug? We don't yet know. We tend to favor the latter based on some preliminary data, but that's still in the works. So my foray in invading your part of the session is we've done a co-clinical trial. It's not a prospective trial. We've done a co-clinical trial. You can do it. The time to analysis of the culture screening was 70 days. And to me, that's too long. The number of patients we could actually generate these culture on was not enough. In this case, it was half. I'd like to get it to 70 to 80% because there's a lot of clinical tests where 70 to 80% is acceptable. And we have a real issue, which is the people who are doing better, we can't even get their organoid, at least in our approach to this. And so we were learning from that and iterating upon that. We think high content microscopy actually could be one path forward. I saw all kinds of exotic approaches you all are using, and that's what we're trying to go into more now. Co-cultures with the fibroblasts, just that one other cell type, I think is going to turn out to be very interesting for us. And we might be able to, again, study initial response as well as adaptive type responses. And it is, from my simple-minded standpoint, a new approach and method. It's not, a mouse. It's not in the patient. It's outside the patient. And, actually, I have this. I'm not actually trying to hire people, but I am trying to hire people who want to help us move this forward. We're not a company. And we're not a bunch of bioengineers. I went to school with bioengineers, and I love them. But Cold Spring Harbor, we're mostly a biology place. And these are some of the pictures of the people who drove it. Amber Havowski has been a terrific postdoc. This is Jen Knox from Toronto, who's the co-PI in the study, and Fayez Noto, who's our bioinformatics. We get all kinds of funding from federal agencies in this country, as well as in Canada, and Stand Up to Cancer and the Lustgarten Foundation. I look forward to participating on the panel, and I hope I have something of relevance to add to it. Thanks.
Thank you. Thank you for the nice talk. Now we're gonna move to the panel discussion.
Very much. I'm going to share a slide sort of highlighting the key questions we have for our panelists. But while I do that, there's a quick question in the chat about how long can we preserve the chips after generation of organoids? If yes, what temperature will be favorable? Sort of a brief question, but then I'll share my slides.
I think this question is for Dr. Agbo, right?
Is it? How long can you preserve the chips? I'm not quite sure I understand the question, so I don't know. I mean, if the question is how long the organoids live, that depends on, you saw brain organoid data when they actually become ready after 200 days. But the chips don't reduce or prolong the lifetime. It's driven mostly by biology. The chips are quite agnostic. So I'm not sure if I understand the question.
I think that's probably what the question meant, just in general, and the answer is always it depends. probably for these systems when you set them up, but general temperature for viability, I think. Thank you. Right now we have some key questions, so I'll get us started with the first question, and sort of we'll take turns with my colleague Youngjun. The first question we wanted to pose to the panel was, so we heard a lot of potential gaps and opportunities from today's presentations, and as we come to the end of the workshop, what are the most significant infrastructure gaps and or opportunities which are the most urgent to address or implement? So kind of leave it to the panel. Anyone like to take a first stab at it?
Maybe I could go first. I mean, I think there are a lot of gaps, right? And it's hard to cover all of them. But I think during my talk, I kind of mentioned that we generated the world's largest human iPSR biobank, and we shared them. And I think in our experience, the really entry barrier for many investigators is getting access to healthy lines, and more importantly, getting access to disease lines. And a lot of times they just don't have access to these. And that's part of the reason why we created this biobank, and we just give them out. And then hopefully that kind of decreases the entry barrier. And then once people get in, they need to learn how to differentiate. They need to learn how to propagate, how do you generate organoids, what are the assays that you need to characterize them, what is the stressors you need to add. It's a whole bunch of other stuff, but I hope I kind of convince you or address some of the critical issues related to entry barriers for getting these disease and patient-specific cell lines.
Dr. Wu, for that comment, and your presentation was very thorough in discussing this. Others from the panel like to add?
I think the infrastructure that's required to do these models is not insignificant. And I mean, you're listening to world experts who have devoted a lot of attention to these model systems. Some of them are, engineers, some are biologists. We've tried to train all kinds of folks in the cancer research community how to use organoids. And so we just, almost similar to what I've heard today, we open our doors to them. They can come and spend time in my lab shadowing my postdocs, spend a day, spend a week, sometimes they spend a month. And then they take all this knowledge back to where they are. Because the conditions to do this are so expensive, most people are shy. But you're talking to a few sites where you have a big developmental biology program and terrific scientists where they hopefully are sharing the costs of the reagents and the expertise. But again, disseminating the concepts are non-trivial. That's where the government could be helpful, since you are the ones who proclaim the NAM statement. So anyway, there's my response back to you. But I think everyone on this call is very excited to tell people how to do this and to collaborate with them, et cetera.
So much for that comment, Dr. Wixwell or Agarwal?
My one observation is that, at least as far as I have been able to detect, there has been very little interest by NIH or FDA in supporting hardware to enable the long-term perfusion of either organoids or organ chips. And that's a problem, because that means that there are several companies that are producing hardware, but that may not be optimized for some of the things that need to be done. And it's clearly not general purpose and actually fairly targeted. So I happen to think that there needs to be an investment by NIH in the sensor, actuator, and control approaches. I just haven't seen it. And the proposals that I've seen on developing organ chip platforms from NCATS were multi-organs. And I think there are enough problems with multi-organs that it's not clear that that's the best way to advance biology. My fundamental view, as I try to get across in the talk, is I think from mechanism of action and efficacy, 3D biology in either organoids or organ chips, preferably perfused systems, is fantastic. But the hold on the entire field by the pipette community and pipette industry, not community, pipette high throughput screen industry is blocking progress.
So much for that comment, and that's why we're having this workshop. So thank you.
That's why I agreed to speak.
And those kinds of efforts are fundamentally so non-appealing to study sections. If you try and propose the hardware development as the goal, it's just generally doesn't fly well with the standard NIH study section. So there must be concerted RFAs just for that, what John just said.
And the difficulty is that I don't know the extent to which the program managers understand the hardware need, because to some extent, the organ chip community has oversold itself. Particularly the commercial organ chip community has oversold itself. I have no idea whether the organoid commercial community has oversold itself, because I'm not following organoids. As I've said, I've written for a fair number of years, vascularized organoids will end up replacing organ chips. But it's one thing to vascularize an organoid, and it's another thing to connect it to the world or to another organoid. And that happens to be a very hard thing to do. And I think it can be done, and it will be done. And there are a few groups that are doing it, but not from the point of view of producing a commercially viable, scalable platform that meets David's concerns for being cost-effective and easy to use.
That point, and I think that kind of leads us to the next question about complementary approaches. You prepped us well for that one, Youngjun.
Yes, I think, yeah, we heard this today that this famous quote, all models are wrong, but some are useful. So in your opinion, what would an ideal integrated research framework look like? And what is currently preventing us from getting there? So that's the next question. And I think, Dr. Wu, you mentioned that there's 4 key platforms and so integration of all this together and maybe with others like animal models. So do you want to give your opinions?
Yeah, so yeah, I'm happy to. I mean, I think I kind of mentioned the 4 key platforms, using iPS cells as a platform to help you validate the genetic variant that you got from, next generation sequencing. And you can also use AI to help you with the drug screen. So in the context of a drug development, I think, part of the thing that we get fooled is that, when I was doing my PhD, you create a transgenic mouse, and as long as it's novel, the mouse has a phenotype, you get a Cell Science Nature paper out of it. But when you stop and think about it, that mouse, if it's overexpression, that overexpressed gene is like 1,000-fold higher. And outside of the cancer context, most other diseases don't have a gene that overexpresses 1,000-fold. And then when you coming with a drug, the drug block that gene or protein and it comes down, of course, you're going to see a good phenotypic change. Of course, you're going to say that, oh, the drug worked, but then when you go to clinical trial, it fails, right? So whereas in humans, there's going to be a lot of variability. I didn't have the data to show it, but hopefully I'll show it later. If you take a drug and you give it to me, how I respond to drug will be very different from the rest of us. And the genetic variability of drug response, in our experience, actually is much bigger than the technical variability of, one postdoc to another postdoc, or technical variability one day to another variability. And so if you can imagine that if you're a drug company, you got 10 drugs, and you like drug number 1, another company has the same 10 drug, they like drug number 3, it's because they're using different cell line. And I do hope that, the complementary approach would be to take all this large biobank, do a clinical trial in a dish, and then validate your lead compound. I'm not advocating completely get rid of animals. I'm just saying that before it was 100% animals. Today it's probably 90% animals, 10% NAMs. Hopefully 10 years from now, you'll be 95% NAMs, 5% animals. In which the last part, you still do it for some of your PKPD studies. Just make sure you're not seeing any surprises. but upfront, you could do most of your work in using human relevant models. Yeah.
Thank you so much. In the interest of time, I think we can sort of, all of these are related to each other. So the next question is thinking about predictability. Where are the current models succeeding? So we heard a lot of examples of where they're succeeding, but where are they still falling short in predicting human disease? So I think I would sort of build on that complementary approach. So if you have any additional thoughts on, opportunities here for further development in the infrastructure.
The industry perspective is sort of missing. We've been part of funded consortium where industry has been brought in. Certainly the tissue chips consortium would have a lot of input from industry where they would open it up to even the data, intramural data in a safe space for failures that could be replicated in chips. But once you're out of that funded network for the normal investigator, it's really hard unless you develop those organic relationships with industry. And so perhaps NIH could enable a space where you could, ask industry, A, what's needed, what's the benchmark, but also are there any intramural studies we could replicate in chips that, failed humans but passed animals?
Supportive infrastructure, I'm hearing.
I have a question which I think is probably directed to Dr. Wu, and possibly others. To what extent is the differentiation of iPSCs to get a targeted cell phenotype, dominated by industry versus academic labs? And to what extent is there general purpose automated hardware to do this other than the Molecular Devices Cell Explorer?
I think, John, with regard to the hardware, typically it's hard for academia to buy these hardwares, right? Some of these hardwares could be the cheap ones, quote unquote, the cheap ones is $250, right? The expensive one could run to $1 million to $2 million. It's just out of the question for us. And that's why for most academic labs, we just resort to postdocs or technicians differentiating these cells, doesn't matter what cell types. And I think for industry, whereas they're able to do that, yeah, they'll buy these robots that help you change media that have a differentiation protocol built into it. So I think it's a cost issue really in terms of implementing hardware, implementing robots into the differentiation process.
So I appreciate that, but the question is, to what extent is that cost barrier delaying work because of the fact that the academic labs either have to do it by hand or find a commercial partner who can afford one of the automated machines?
I think in the ideal world, if we have a robot in my lab and it's $2 million robot, it's working 24/7. Yeah, we're gonna have a productivity, 3 times, 4 times less better, right? Than staff.
You don't have one, is that right?
No, we don't have a robot because I think I visited NCATS and they were showing me some of these robots and it's $2 to $3 million at each. And most academic labs just don't have the resources to buy it. Yeah.
You and I should talk.
Yeah.
Yeah.
So actually that's bring to the next question. So what kind of collaboration models are most needed to move the field forward? Anybody want to chime in about this collaboration?
Well, I mean,
I think yeah, go ahead, David.
I mean, you're doing fabulous. I think these models can be used for basic discovery. You heard about Dr. Wu showed terrific examples of essentially understanding germline genetic diseases that could affect the heart, possibly other tissues of your body by making these isogenic cell lines and making iPS cells from patients that have these various syndromes. I personally think that if these models could be brought into trials where novel agents are being tested and actually using the approaches of the other 3 speakers that I heard today to look first for toxicity. Traditionally the tissues that we worry about right away are heart, nerves, and nerves. Of course, the other end organ, kidney, liver, et cetera, lungs. But if we could use these methods to look for acute toxicity and then some measure of efficacy, it could help guide, drug development. And the one cell type I didn't hear about in this talk, and probably others talked about it today, were nerves. And a lot of our drugs don't cross the blood-brain barrier, and so it's non-issue, but some drugs do. And that's one thing, but actually my point was going to be something else, which is I think innervation of normal tissue is a big part of their biology. And I think it's a hard problem. I don't know if any of my colleagues on the phone have figured out how to add nerves into their culture, meaningful nerves. I'm talking about peripheral nerves. Particularly on the vasculature, they play a big role, but even epithelial tissues are innervated. And to me, that's a challenge, but that means it's an opportunity for the trainees of the labs on the line and for the field writ large to think about. I'm a cancer guy, so I just want to make the cancer go away. But a big problem I have with the drugs is the drugs are either toxic or, quote, don't work well enough. And I do think the models could be great. And I'm hoping that people on the phone who make these better models than I have would be willing to think about cancer in addition to the more complex conditions that they're working on.
Yeah. And maybe to piggyback on David, we do a lot of work related to cardiac organoids and looking at cancer-induced cardiotoxicity. But with regard to the nerve, it's a common issue that we see in our patients, right? They have peripheral neuropathy, and it should be possible to generate iPSC-derived peripheral nerves exposed to chemo and see what are the side effects. But I do think that, again, piggyback on David, that I do think that, for example, some of the key splashes recently with some of the gene therapy trials, for example, the Duchenne's muscular dystrophy with Sarepta, in which patient died with acute liver failure. There's also Intellia, this is for ATTR amyloidosis. Also patient died in their phase 3 CRISPR-based gene therapy trial. Several trials that were on hold because of the liver death. Traditionally, you just inject a mouse and show there's no liver toxicity with AAV, and they go to clinical trial. I do think in the future, you should be able to take this AAV or lipid nanoparticles and put it on the liver organoids of 100 different patients, just so that you make sure you're not getting surprises in humans. And this is something that I think the gene therapy field can incorporate immediately. Yeah.
Thank you very much for your feedback. I think we are running short on time, but really value your input and great comments across these areas. Some were highlighted in the training and the potential next steps. So thank you for your input. I'm not sure, Youngjun, if you have any other comments or questions.
No, thank you. Yeah, I think we should ramp up.
Thank you very much to everyone. So it's my pleasure to give opportunity for closing remark to Stephanie Murphy. Stephanie Murphy is director of the Division of Comparative Medicine at ORIP Poughkeepsie. Her division oversees a number of large, high-impact research model resources in portfolio of research grants, which aimed on development of new animal models and as well as other research models. I also would like to mention that her division is actually managing for the whole NIH Human Tissue Organ Research Resource Store, which is very highly valuable human tissue resource. Stephanie, please, podium is yours.
Thank you, Oleg. Well, as we conclude day one of this workshop, I would like to extend my sincere thanks to our keynote speaker, our presenters, panelists, session chairs, moderators, attendees, and the workshop organizing committee for an outstanding day of scientific exchange. Today showcased the remarkable innovation occurring across the biomedical research community as investigators develop new tools and approaches to better understand human biology and disease. While we heard about a wide range of technologies and model systems, several overarching themes emerged that cut across all of today's discussions. First, we repeatedly heard that the future lies in integration rather than replacement. While discussing organoids, organ-on-chip systems, stem cell models, artificial intelligence, computational approaches, or model systems, speakers emphasized that each offers unique strengths. The challenge and opportunity is to bring these approaches together in ways that generate deeper biological insight and more predictive science. Second, today's conversations reinforce the importance of research infrastructure as a scientific enabler. Advances in technology alone are not enough. Progress depends on access to high-quality biological resources, shared platforms, interoperable data systems, benchmarking tools, validation frameworks, and community standards that allow discoveries to be reproduced, compared, and scaled. Third, we heard a strong focus on building confidence in new approaches. Across multiple sessions, speakers highlighted the need for rigorous validation, reproducibility, transparency, and evidence-based benchmarking. As these methodologies mature, establishing confidence in their performance and applicability will be essential for broader adoption and impact. A fourth theme was the growing importance of collaboration across disciplines and sectors. The challenges we are addressing span biology, engineering, computational science, data science, medicine, and technology development. Success will require continued partnerships among academia, industry, government, nonprofit organizations, and resource providers. We also heard that developing the next generation of researchers and ensuring broad access to tools, data, and expertise will be critical to sustaining progress and fostering innovation in this rapidly evolving field. Collectively, today's presentations demonstrated that we are entering an exciting period in biomedical research, one in which advances in experimental systems, computational methods, data science, and AI are creating new opportunities to understand disease mechanisms and accelerate the development of interventions that improve human health. On day 2 of this workshop, which will take place on Friday, June 12th, we will build on these discussions by exploring the development, validation, scaling, and translation of NAMs, advances in automation and AI-enabled discovery, and emerging in silico approaches, digital twins, and data-driven frameworks that can further strengthen the biomedical research ecosystem. We look forward to continuing the conversation and identifying opportunities where research infrastructure can help bridge disciplines, connect communities, and accelerate scientific progress. On behalf of ORIP and the organizing committee, thank you again for your participation, your insights, and your commitment to advancing research infrastructure in support of human health. Have a wonderful evening, and we look forward to seeing you on day 2.